A very good morning to one and all. Today we'll be starting with the session 3. Before that, I would like to make one announcement. The posters which were displayed yesterday is being kept in the reception hall. Students, kindly collect your respective posters, please. There is a slight correction. The students, kindly collect your posters after the tea break in the afternoon, because again there is evaluation is still is going on. A very good morning once again. I welcome you all for the second day of the Student Symposium on Genomics-based Next Generation Crop Improvement Approaches, organized by the Division of Genetics IARI in collaboration with DuPont Pioneer. Now we will start the session 3 on applications of genomics. Yesterday we were fortunate to hear the lead lecture from Professor B.D. Singh, who is very familiar among the students of agriculture through his books on genetics, genetics, plant breeding and biotechnology. Today we have the equally eminent, very familiar among the students through his books on cytogenetics, that is Professor Pushpendra Kumar Gupta, who is very familiarly known as Professor P.K. Gupta. Professor P.K. Gupta extensively worked on different areas of plant cytogenetics, induced mutations, quantitative genetics, utilizing both classical and molecular approaches, mainly in cereals and legumes. Professor has more than 50 years experience of teaching and conducting research. He is a fellow of National Academy of Sciences, India, National Academy of Agricultural Sciences and Indian National Science Academy. Today, Professor will be enlightening us about the plant breeding in genomics era. So please.
Distinguished guests, I see many senior faculty members and scientists from IARI and elsewhere in the audience. It's a great pleasure for me to be here this morning to deliver a lecture as requested by Ashok, who has been very dear to me for many years now. He asked me to deliver a lecture on plant breeding in the genomics era. I modified it. Plant breeding in post-genomics era, because I think the genomics era is almost over. The first genome was sequenced in 1995. It was a bacterial genome.
So you can come
and
here.
the first hair plant genome of Arabidopsis was sequenced in December 2000. And after December 2000, during the last 15 years, all major crop genomes have been sequenced. In wheat also, which is the largest genome, most of the information about genome sequence is already available, and a high quality genome sequence of wheat should be available next year or by 2018. Therefore, I call it post-genomics era, and you will see that I will try to tell you how genomics information which is already available will be utilized for plant breeding in future. I learned that majority of what I am going to talk about has already been covered yesterday in different lectures. Therefore, you may treat it as a summary of the lectures which were presented yesterday, because I have seen the different topics and I have covered all of them very briefly, just to summarize them. How to operate? Yes. This is the outline of the lecture. I will spend a few minutes on talking about challenges, hunger, nutrition and climate change. Then I will briefly talk about traditional methods, non-traditional methods and new methods of plant breeding. Briefly about molecular markers which have been extensively utilized during the last 15-20 years. I will also talk at some length marker trait associations which have been discovered following two major methodologies, interval mapping and association mapping. And you will see that in interval mapping and association mapping, I will mostly concentrate on the recent developments, because these technologies have been modified almost every year. New methods, new softwares are available every year. Then bioinformatics is going to play a major role, because we can use bioinformatics to discover genes, develop markers for plant breeding. Then a few slides on heterosis breeding, because new methods have been developed in heterosis breeding also. I will not talk about cytoplasmic male sterility restorer system, but the new methods which have been proposed in recent years. And then only one slide on combining, how can you combine transgenics with marker assisted selection. I will not talk about GM crops, because that is a topic which requires a full lecture, and will restrict myself to molecular breeding, that is marker assisted selection. Challenge 1, climate change, must have been talked yesterday. We have great challenges because of the rise in temperature, different patterns of rainfall in the future years. The carbon dioxide will increase, which could have been beneficial for photosynthesis, but because of other factors like increase in temperature, this benefit of carbon dioxide increase will be offset. We will not be able to utilize it. There will be heat waves, drought and floods in years to come, and then there will be new pests and diseases emerging which will have to be dealt with by the plant breeder. Then challenges 2 is population explosion. You can see that the present population may be about 7.5 million or billion or 7 billion. Expected population we already know about 9.5 billion or 10 billion in 2050, and then hunger and malnutrition is going to be a major issue. This is the slide which I have taken from an editorial published only day before yesterday in Current Science. I learned that Dr. Swaminathan has already covered this particular topic, therefore I don't have to explain what Anthropocene era is. And in this particular concept which Dr. Swaminathan has presented, he has talked about farming system for nutrition, naturally biofortified plants and gardens of fortified crops, and also talked about community hunger fighters which should be trained in order to look after the malnutrition and nutrition problem which is existing almost throughout the world. I don't have to explain them because Dr. Swaminathan must have talked about them. Non-traditional methods, you can divide them in two. The classical non-traditional methods, mutation breeding, which has been an important area of research in order to develop a large number of mutant varieties. Thousands of mutant varieties have been developed. They are available on the web, on the IAEA, International Atomic Energy Agency. Then cytogenetics have been used, but not as extensively as mutation breeding. Polyploidy has been used, for example, TTKL crop, new crop has been developed. Haploidy has been used in order to develop the crops utilizing the double haploids. Alien genetic variation transfer has been utilized, particularly in case of wheat. Large number of genes have been transferred from alien species. And then balanced tertiary trisomics, which I teach to my students, was another technique which was used in 1960s, 70s, leading to release of a variety, Humber variety in USA, which was later on became susceptible and therefore could not continue. So you can see that mutation breeding and cytogenetics we have been teaching to our students for the last about 50 years. But more recently, the other 4 which are listed here, the development of molecular markers, the development of GM crops, BT cotton, BT brinjal and GM mustard, you must have been hearing about it. Then gene targeting, another very important area. There are different techniques, different nucleases, CRISPR-Cas technique, and then heterosis breeding. In addition to marker the cytoplasmic male sterility restorer system, new methods have been suggested recently which will be used in future for development of hybrids. Some major technologies which have been used during this period in post-genomics era, development of molecular markers, genome sequences have been discovered for majority of crops, genomics information is available. Next generation sequencing during the last 5-10 years is being used extensively for almost all programs which involve marker trait associations. Allele mining is being done by tilling. MicroRNA, competing endogenous RNA and long non-coding RNAs, this is a subject which has emerged during the last 5 years and is going to be a very important area of research which will be utilized in plant breeding. Epigenetics is another very important recent area in which we try to examine the pattern of DNA methylation utilizing different techniques, MSAP and MEDIP, and histone modification using the chip technique. We are already using ourselves in collaboration with Dr. Prabhu to utilize epigenetics for disease resistance. Genomics, whole genome sequence and next generation sequencing for molecular breeding. Whole genome sequencing and next generation sequencing are being extensively utilized. Even in species where genome sequence is already known, we are sequencing thousands of new varieties in order to find out the genetic variability. Recombination bins have been identified in rice, particularly in China by Dr. Bin Han. He has published a series of papers where instead of conventional molecular markers, he is using recombination bins as molecular markers. And then if you have the genome sequences available, then you can develop in silico SSRs, SNPs, RADs, ISBP, CN, number of different types of markers can be discovered, although in our country we are not using most of them. Oh, okay. is there pointer also in this? Pointer? Yes, yes. And then you can also develop PCR based functional markers if gene sequences are known. You can develop the primers and you can amplify the gene in which you are interested and then try to find out the markers. Bioinformatics can be used for gene discovery in a crop where the gene is not known, if it is known in other crops. Then genes for microRNA, competing endogenous RNA and long non-coding RNAs, they have been discovered in number of crops during the last 2-3 years. And then epi-alleles also have been discovered. I will discuss this in some more detail, DNA methylation and histone modifications. Yes. Now I pass on to the methods which we are going to use for developing marker trait associations, and for this purpose you can see these are the different populations which can be used. You know, most of the students know about biparental populations which are used in QTL interval mapping and association mapping panel which is used in association mapping. But you can see here that there are a large number of different type of populations which can be used: near isogenic lines, introgression lines, advanced intercrosses RILs, backcross RILs, single segment substitution lines, chromosome segment substitution lines, and then multiparental population involving MAGIC and NAM populations. All of them are being used, but not much in India has been done in this direction so far. Also, I am committing a mistake. Yes, this is a very classical picture of QTL interval mapping, where you do the genotyping and phenotyping, and then you try to screen all intervals. This is a genetic map, and every interval of the genetic map between two markers is screened for the presence of QTL using expectation maximization algorithm and also likelihood ratio. And when you find a particular region of the genome where the log value is very high, in this case about 12, then it means that a QTL is present. This is a very simple one, a traditional one, more than 10 years old from our lab. This is another important picture which I want to show, because not many people in our country examine multiple traits together. If you have correlated traits, if you are studying many traits, then you should study the correlations, and the correlated traits then should be subjected to QTL interval mapping, because there are QTLs which control more than one trait and there may be pleiotropy, and therefore it is very important that every student, every scientist should examine multi-trait QTL interval mapping. There are facilities available in QTL cartographer and other areas where you can conduct multi-trait QTL interval mapping. You can see in this case there are two traits and you have the same QTL. Whether it is due to close linkage or pleiotropy, that has to be discovered. Another slide from one of my students, Vijay Gehlot, who did his PhD with us. The purpose of showing this slide is that large number of epistatic interactions are there, which many of us do not take into account. There are facilities available where you can use software in order to find out epistatic interactions, and sometimes these epistatic interactions contribute more than 50% of the phenotypic variation for a particular trait. Therefore, ignoring epistatic interactions will really give you wrong results. Method of QTL SAC, this has been suggested in order to reduce quantum of work, where next generation sequencing can be utilized. You can phenotype the population RILs, and you have then take pooled samples of DNA from the two extremes of the phenotype, and these pooled sequences are then subjected to analysis where you can discover SNPs. So you reduce the quantum of work by not genotyping all RILs, but taking only the extremes, pool of the extreme phenotypes, and then subject them to QTL sequencing. Rather than genotyping with a particular marker, you can sequence them and then identify SNPs, also in crops where molecular markers are not known. Another very important slide where you could develop RILs for epigenetics. This is DDM, decreased DNA methylation. This is a gene for decreased DNA methylation. It is present in one and not in the other, and therefore you can develop an RIL, and with the help of this RIL you can identify the genes which are affecting the trait through DNA methylation. Epialleles for plant breeding, DNA methylation, histone modifications, chromatin remodeling, RNAi, all of them have been shown to influence important agronomic traits almost in all crops. There are a number of examples I have listed here which have already been shown to have epialleles and influence the traits through epialleles. For example, lysine content in opaque to maize, reduced caffeine in coffee, fatty acid composition in cotton, carotenoid and flavonoid in tomato, low glutenin in rice, and drought and heat tolerance in grapevine. You can see some of our students are already engaged in this in doing in wheat. We are trying to identify epialleles in majority of cases, and when you start studying a particular character, then you find that almost all characters have an epigenetic component to control the phenotype, and therefore these discoveries really suggest future role of epigenetics in plant breeding. Genotyping using next generation sequencing, whether it is interval mapping or association mapping, in these cases you use these two important technologies. You try to find out recombination bins, as has been done in case of rice, where 150 RILs have been subjected to next generation sequencing, and with the help of sequences which were available and knowing the sequences of the parents, you can find out the recombination bins which are available in the recombinant inbred lines, and utilize these recombination bins as markers. Then genotype by sequencing, this has become very popular during the last about 4-5 years, discovered, suggested in 2011-12, and now almost even in India many people are using this technology where bar-coded multiplexing of genotyping is done, and you can outsource this and get the data and utilize this data for the purpose of genotyping the population even when you don't have the markers. SNPs will be discovered automatically after genotyping by sequencing, and that can be directly utilized. Our students are already utilizing this particular technology in finding out marker trait associations. But this technique of recombination bins has been used to the best of my knowledge only in China by Bin Han in Shanghai, and I have not seen any other paper on recombination bins. This is the paper, recombination bins and sliding window approach, which was used by Bin Han, this is the Huang in 2009 and then 2010, and number of papers have been published where these are the two parents, you have the sequences of these two parents, then you develop the RILs and you try to find out the recombination when two adjacent markers derived from the same parent are not present together, it means recombination has taken place. In this manner, hundreds of recombination bins were identified and utilized for identification of material. This is association mapping. You take the germplasm, genotyping and phenotyping, and after genotyping and phenotyping, either you can use candidate gene approach or you can use genome-wide polymorphism and association mapping approach. This is the classical approach, but number of different models are available in association mapping, and therefore majority of us normally use this model where we use Q and K matrix in order to find out the association mapping. But I will show in some of the subsequent slides that these methods have become outmoded because there are serious problems of error and false discovery in this particular case. Therefore, you have to be extremely careful in using these two approaches, candidate gene approach and association mapping. Problem of low frequency of alleles, I have been talking about it, but many people don't care about it. You see, when you do association mapping, then we ignore about 5% to 10% of the data because the particular marker allele is present in a very low frequency. If a marker allele is present in less than 5%, that data is eliminated because of statistical reasons, because you cannot really utilize that. And therefore, during the last 2-3 years, considerable work has been done on these rare alleles, because these rare alleles may be the most important, because the desirable character which you are looking for may not be present in high frequency, and therefore marker alleles also will be present in low frequency. Therefore, these marker alleles, one of my students identified these marker alleles and tried to analyze the data, and then she discovered that these marker alleles are very important and they should be subjected to future studies utilizing these rare alleles. Post-GWAS, we are writing a paper at present on post-GWAS approaches. What after GWAS? GWAS, you have identified the markers, but what after that? This is a diagram which shows what can be done after that. You can map these markers on the genomic regions with associated markers. You can use genomic sequences and ESTs and eQTLs in order to fine map the genome, identify the candidate gene, then score and select the genes, prioritize the candidate gene which you have found, and then conduct functional analysis, tissue expression, gene set association, genetic architecture, trait development pathway. This particular post-GWAS approach is already being followed in a number of cases, but not unfortunately in our country. This is another study which one of my students conducted, Vandana Jaiswal. She published this work in PLOS One last year, where she took a gene for grain weight, and this gene was already available, the sequence was already known. Therefore, she designed the primers, and about 250 genotypes she sequenced. This gene was amplified by PCR, and then amplified product was sequenced, and then she identified SNPs. In this one gene, she identified 4 different SNPs, and she found, when she compared this with the phenotypic data, she found that only one of them was the causal SNP. Others were not the causal SNPs. The last one is the causal one, where allele A is higher 1000 grain weight and allele G is lower 1000 grain weight. And therefore, she conducted further expression studies utilizing this particular data, which has been published in this paper, and I am not showing. She also developed cap markers utilizing this particular SNP, which is the causal. You see, in most of the cases we don't care about the causal gene or the causal markers, because for a plant breeder it is not important. You want to have a marker which will allow you to select the phenotype, and therefore it is not really important. But for further detailed studies, you have to know the causal polymorphism. Marker trait associations in post-genomics era. This shows that how should you consider the recent developments in both these areas, interval mapping and association mapping. In interval mapping, some of our students have conducted meta-QTL analysis. For example, in pre-harvest sprouting, there are 150 papers published and more than 150 QTLs reported in the literature on all the 21 chromosomes of wheat, and therefore when meta-QTL analysis was conducted, out of 150 QTLs we could discover only about 8 or 9 meta-QTLs which are considered to be more reliable and more robust for the purpose of plant breeding. So this meta-QTL analysis should be conducted. Not many students in India do it. Main effect, we should distinguish between main effect and epistatic QTLs, because epistatic QTLs may be present which are not between the QTLs showing main effect. You can also have epistatic interactions between QTLs which don't have any main effect. Therefore, you miss all of them. And it is very important because there are large number of genes which do not have any main effect, like transcription factor, microRNAs. They don't have any direct effect, but they contribute through epistatic interactions. So, in large number of studies conducted in our lab, we have identified epistatic QTLs, and as expected, we found that number of these epistatic QTLs are those QTLs which do not have any main effect. So, this I want to emphasize therefore. Then eQTLs, I have not seen any study from India on expression QTLs. Then epiQTLs, I told you that epi-RILs can be developed and epiQTLs can be identified. This also, some work has been done abroad, but not, we don't even talk about it in India. Also, you should realize that in interval mapping, we always use biparental populations, and there are serious drawbacks of this, because you are addressing only the phenotypic variation which is present between the two parents, and therefore limited genetic variability is being examined. Therefore, you have to use multi-parental populations, and there was a workshop in England on this 2 years ago, and a series of papers have been published on multi-parental populations in genetics and G3 journals during the last 2-3 years, where it is emphasized that from biparental population we should move to multi-parental populations in order to capture the entire genetic variability which may be present in the population. Magic populations have been developed in large number of cases, in case of wheat, rice, all of them. Association mapping also. We are moving forward by newer techniques. Almost every month I find a paper on new softwares, new methodologies which are being developed. You see, one of the drawbacks of association mapping is that you discover marker trait associations. Large number of them, more than 50-60%, may be false, and therefore when you apply correction, then you lose even the real ones. So that is the problem. False positives in the beginning, when you apply correction you get false negatives. So what to do? That is a serious problem. So you have to move from population based to family based association mapping, from genome wide association studies to joint linkage and association mapping, to post GWAS era, which I already mentioned briefly, and then in the recently one of our students published a paper on single locus single trait analysis to multi locus mixed model and multi trait mixed model. This is what I have been emphasizing during the last 4 years. Important techniques were published in 2012 on multi locus mixed model and multi trait mixed model, and this has not been used by many. The paper which we published this year by Vandana Jaiswal, the same lady, she utilized these two approaches. We took the help of Institute of Agricultural Statistics, Agricultural Institute. They helped us in conducting this analysis, and therefore you discover large number of new marker trait associations with this particular approach, and these particular approaches also tell us about the drawbacks of the kind of genome-wide association studies which we have been conducting, and therefore we are currently writing a paper on association mapping in the GWAS, post-GWAS era. These are the basic techniques, common method for backcrossing, both for single gene transfer by backcrossing and gene pyramiding, where you can have 3 donors and transfer the genes from all the 3 donors to the same through marker assisted selection. These are different approaches. Marker assisted backcrossing, simple. Marker aided pyramiding, you can take number of 3 donors and then utilize all the 3 in order to transfer number of genes. And then marker assisted recurrent selection and genomic selection. Marker assisted recurrent selection, I know Dr. Prabhu and his group have already utilized it at IARI. Genomic selection, I don't see any paper from India so far, but some work is being done on genomic selection, and it is a very powerful approach. The only problem is with the prediction models, which are being refined almost every year, and therefore I am sure that in future the prediction models will be refined to the extent that this technique will also become very, very useful in future for molecular breeding. Mass for complex traits. You see, majority of the studies which we have conducted and utilized for plant breeding, they are for simple traits like disease resistance, where we are transferring single gene or pyramiding a few genes. But what about the complex traits? The complex traits like drought resistance, salinity, heat tolerance, they are difficult to deal with because there are large number of genes involved in these, and then we always use surrogate traits which we identify and utilize, and therefore in all these cases there is a serious problem. Although major genes have been identified for drought tolerance and salinity tolerance, and they have been utilized in marker assisted selection, but much more can be done. Complex traits are controlled by few major and many minor genes with low heritability, and therefore you have to capture these minor genes, because major genes are already, most of them are already captured in conventional plant breeding. So if you address the capturing of these minor genes with the help of molecular breeding and marker trait association, then this can be another very, very useful approach. Marker assisted recurrent selection and genomic selection deal with complex traits, but there is hardly any ongoing program in India. Breeding for complex traits, number of them, biotic and abiotic stresses. Yes. nutrient use efficiency. Then there are polygenes, including genes encoding for transcription factors, two-component system, non-coding RNAs, and epigenetics. This is rather very important for us, because we are using all these approaches in our lab in order to find out new markers which are responsible for traits which normally we do not care for. Can bioinformatics help? Every student in my lab devotes 50% of their time in bioinformatics now. The reason is that you can mine a number of genes with the help of bioinformatics. A gene may be known in rice, a corresponding gene in wheat can be discovered through bioinformatics. One of my students has done this for grain weight. All genes known in case of rice have been isolated and sequenced. They have been used by her in order to find out corresponding genes in wheat, and you find that homologous genes are available. Then you can develop markers once you know the genes. Then one of my students used two-component system. I will very briefly mention about it later on. Two-component system, which is a very important system in order to address the abiotic stresses. Transcription factors, we have recently written a review published in Theoretical and Applied Genetics, how transcription factors can be used for drought tolerance in case of wheat. MicroRNAs, I published two papers in Current Science on microRNA and competing endogenous RNA, which can be utilized for the purpose of molecular breeding. And now one lady is devoting her time on long non-coding RNAs, which are also believed to play a very important role in plant breeding. Then another area which is very important is the study of biosynthetic pathways. For example, starch biosynthesis, there are a number of genes involved. You can study all those genes, try to develop markers for those genes and utilize them for molecular breeding. We are handling one important gene in starch biosynthesis in wheat, AGPase. I will talk about it briefly later on. This is the two-component system. Paper was published in Functional Integrative Genomics. Two-component system was discovered in case of bacteria initially, where you have histidine kinase here, and this histidine kinase triggers the response regulator here, which then transfers the information to the gene, which has to be activated. In case of higher plants, there are three steps. There is histidine kinase, then histidine phosphotransferase, and then response regulator. So this student Vijay Gehlot, he identified a large number of genes belonging to all these three steps, and then tried to conduct wet lab studies to characterize, validate these genes, whether or not he took two varieties, one drought tolerant and the other was drought recessive, and utilizing these two varieties, he demonstrated that this system is relevant and can be utilized for plant breeding. These are the genes, a number of histidine kinase gene, then histidine phosphotransferase gene, then response regulator genes. Large number of these genes have been discovered, but not in many crops. For example, in rice it has been done, in wheat we have done it, in soybean a foreign group has done it, but in India nobody has done it except our lab. Then number of students are involved in bioinformatics in my lab. Transcription factors, number of transcription factors have been discovered, genes for these transcription factors, microRNAs for disease resistance and drought tolerance, and nitrogen nutrient use efficiency, 17 genes for transcription factors for drought, 11 genes for transcription factors for nutrient use efficiency, they have been discovered, and sequences have been retrieved and molecular markers have been developed. You can see here what I am talking about, the transcription factors. Oh no. Yes, this is a recent paper published only this year, about 1 or 2 months back, where it has been demonstrated that a particular transcription factor modulates jasmonic acid and salicylic acid in disease resistance and therefore can be utilized. And just to remind you, the submergence tolerance gene in case of rice, which has been extensively utilized, I read a paper in plant physiology, it controls 900 genes. It is a transcription factor gene. So we should be able to discover genes for transcription factor which may be actually influencing a large number of genes. This is a paper for Arabidopsis NAC transcription factor, which controls disease resistance. MicroRNAs for drought tolerance. There are 434 drought responsive microRNAs from 11 plant species which have been published. And utilizing this information, one of my students tried to discover microRNAs for drought tolerance in case of wheat. And there are 3 papers published from University of Delhi South Campus recently, where it has been shown in case of rice, as you can see here, identification novel microRNAs from drought tolerant rice variety Nagina 22. They have published 3 papers during the last 2 years, where they have shown that there are specific microRNAs and their genes which are differentiating this particular variety from a drought sensitive variety, and therefore these microRNA genes can be utilized for plant breeding. You can go through these papers. This paper was published in Planta 2015. This was published only last month, Scientific Reports August 2016, and they have another paper in press. Yes, this is a diagram from one of our review which we have published in Theoretical Applied Genetics last year. Starch synthesis, crystal structure of AGPase and conserved residues. AGPase is a rate limiting enzyme, very important enzyme in starch synthesis, and its structure has been studied in great detail in number of cases, particularly in case of maize, but we try to study it in case of wheat, and a large number of amino acids residues have been identified which are involved in heat tolerance. Therefore, you can use site directed mutagenesis in order to change these amino acids one by one and then develop heat tolerant varieties. So, our main objective is, through the study of this particular enzyme, one of my students spent about 6 months in Kansas State University recently in order to identify these genes, these amino acid residues, expression studies, and we want to utilize this AGPase through molecular breeding in order to develop heat tolerant varieties. Heterosis breeding, you see, normally we use cytoplasmic male sterility restorer system, but there are problems of restorer genes, how much restoration is there, whether restoration is available or not. Therefore, number of methods have been suggested where genetic male sterility is being utilized. There is a disadvantage with genetic male sterility because you have to screen out the male sterile plants for hybrid production, but methods have been developed. The first 2 points here I am mentioning is that hybrid maize, hybrid rice, and now number of hybrid varieties have also been released in Europe, and the methods which have been used, they are different. Development of heterotic groups in major crops, this is another very important issue, because in maize we have these heterotic groups, but they have not been developed in many crops. Like in case of wheat, we do not know heterotic groups. In case of rice, there is 1 or 2 papers published on heterotic groups, and therefore you should understand what these heterotic groups are. I don't have time to explain this, but all students, MSc PhD students, should read what heterotic groups really means. Heterotic group does not mean combining ability. You can't identify heterotic groups by combining ability. There are definite pathways, pipeline for development of heterotic groups. Use of Barney's bar star system for hybrid development, cytoplasmic male sterility restorer and other systems are not available. This University of Delhi South Campus, they have used this Barney's bar star system, in case of mustard. Then this is seed production technology which has been developed by DuPont for heterosis breeding. Number of proposals as a member of RCGM in DBT I have examined where they use this particular technology. I will briefly show it later on. What does it mean? Use of marker assisted selection for improvement of parents. This has been done in case of maize. This has been done in case of bajra in our country. Possible uses of apomixis. This is another very important area. May not be feasible in the near future, but in long term basis, everybody is talking about the possibility. Imran Siddiqui at Hyderabad has done some good work in this direction, and possible uses also of reverse breeding. Very briefly, rice seed production technology. I don't have time to deal it in detail, but there are 3 genes involved in this particular case. in order for the maintenance of male sterility, so that although you are using transgenic material, but your final product will not be transgenic. This is the beauty of this. Therefore, no regulatory problem will be there. This is another paper which was published only recently, where we are using split Barnase. Barnase genes divided into two has been carried in provector, and with the help of this, you see, you don't have the problem in this particular case of having a marker in order to identify the male sterile plants. In case of GM mustard, for example, Deepak Pental has developed, he has to use a marker, for example, glyphosate for herbicide resistance in order to kill the plants which are not male sterile. So, problem of selection of male sterile progeny for female parent is really a problem which has been overcome in this particular case. We are using split Barnase. When split Barnase is present in two segments, then it will not produce male sterile plants. Another paper, very recent paper, 2016, you can see where this gene editing, nucleases, CRISPR technology has been used in order to attack this particular gene and produce male sterile plants. Reverse breeding, plant breeding for heterosis. Those of you who are not aware about it, you should know that we can select an F2 plant which is better than all other F1 plants, and then go back and develop the parents by a particular series of techniques. In this particular case, Ashok knows about it, and if you do not know, you have to read about it. Then comparing the transgenics, conventional approach, you can see transgenesis, where you transfer a gene from particular bacteria or other species which cannot be utilized by conventional plant breeding, and then cisgenesis, where using transgenic technology, genetic engineering, but utilizing a gene which is present within the genetic variability of that particular crop or related species. Summary: plant breeding in post-genomics era will change traditional plant breeding. Traditional plant breeding will continue, but it will be supplemented by these new approaches in a big way in future. Marker assisted selection will be increasingly used for selection in future. Improved methods of interval mapping and association mapping for marker trait association will be used. Heterosis breeding, new methodologies have been developed which will be used in future. And transgenics may be combined with marker assisted selection in order to produce better genotypes for farmers. I think that's the last slide. Thank you very much for your patient hearing. I try to keep to the time, but if there are any questions, you are most welcome.
Good morning, sir. I just want to know, in this molecular era of molecular breeding, does cytogenetics or its new technique have some relevance? If yes, then what are those?
No, don't confuse between molecular breeding and cytogenetics. You see, cytogenetics, I have been a cytogeneticist for 50 years, but I don't think cytogenetics really delivered for the purpose of plant breeding in a big way. Cytogenetics really failed, because I remember when I went for doing my PhD, there was a separate lab for synthesis of new species and for the transfer of alien genetic variation for the improvement of crop plants, and that lab was closed after 40 years. So cytogenetics conceptually is very important, but when it comes to using it in practical plant breeding, you have serious problems. You see, triticale, for example, we thought is a crop which will perhaps replace wheat, but it could never do it. Years and decades have been spent in the development of that crop. It is grown in some marginal areas, but could not compete with wheat.
So
there are some applications of cytogenetics. For example, development of seedless watermelons, seedless guavas, triploids have been used for that purpose. Triploid ornamentals have also been produced, because in that case you will have to produce the triploid every time. You will have to go to the seed producer, take the triploid seed, or instead, in case of ornamentals, for example, if you have produced triploids, you can grow them, propagate them through vegetative means. So there it has played a role. But otherwise, most of these techniques have been initially demonstrated to be useful. For example, balanced tertiary trisomic, I told you, it has been commercially demonstrated that a variety can be developed, but no further one. So don't confuse between cytogenetics and molecular breeding, because in molecular breeding we are developing markers and there is hardly any cytogenetics involved because it is immaterial on which chromosome it is present.
And sir, as you talk about the transcriptional factors, does these transcriptional factors affecting the genome, this
Which conceptual factors?
transcriptional factors?
Transcription factors, okay.
Yeah, these transcriptional factors have any role in hindrance, I can say, in developing genetic modified, because if those transcription factor have some effect on the main gene, and
You see, transcription factors, if you know the molecular biology of transcription factor, the same transcription factor can influence a large number of genes. I gave you the example of submergence tolerance gene. It is a transcription factor gene which controls 900 genes. So when we talk about complex traits and we say it is controlled by a large number of genes, if you can discover a transcription factor which controls all these genes, then you don't need many genes. One gene will be enough if it is a transcription factor. So this is an area we have written a review actually which came back to us for revision number of times, but ultimately published in Theoretical and Applied Genetics. There are a large number of transcription factors which have been discovered and play a very, very important role in drought tolerance, heat tolerance, almost in all traits. All traits. You see, at the molecular level, whenever you are thinking of a character and you are thinking about a particular situation where new genes have to be activated, then transcription factor will always be involved. MicroRNAs will always be involved. We have ignored it because we didn't know the methodology in order to discover. But now because we know the methodology, all these different technologies, transcription factors, microRNAs, competing endogenous RNAs, long non-coding RNAs, they are going to be used in a big way in future. I gave you example of 3 papers published from University of Delhi South Campus, where in Nagina 20 rice they have discovered the drought tolerant microRNAs which have been shown to play a major role in drought tolerance.
Thank you very much.
Yeah, good morning, sir. So I want to go back to previous question about cytogenetics.
Yes, please.
Good morning, sir. Actually, I want to go back to previous question regarding cytogenetics.
Yes.
So
yes.
every tool or every discipline has its limitations as well as its potentialities, and that is true for genomics, post-genomics, previous era, and cytogenetics also. It has not completely failed. We are having LR9, LR19, LR24, and if we see the wheat varieties, large number of varieties are having LR24 genes. Large number of genes have been transferred using cytogenetic tools, and still many genes are coming by cytogenetic techniques. Genes are being transferred.
Yeah. I take your question. Yeah. You see, I have been asking people, can you tell me an example of a wheat variety where systematically an alien gene has been transferred leading to development of a variety? All varieties where alien genes are there, they are all by nature, random. No planned experiment you can show me where an alien gene has been, you plan an experiment, you start with a gene and transfer it leading to development of a variety.
Yes,
there is no example.
Yeah, it was not a gene, it was a trait.
I have published a detailed comprehensive article in a book which is recently published by S.N. Rayana and his students, where I have covered this. There are large number of varieties and large number of genes, alien genes which have been transferred. So the role of alien genetic variation in plant breeding is demonstrated, there is no doubt, in case of wheat, in case of rice, but in majority of cases you start with a cross and then you really depend on chance. You cannot plan. You see, when I was a student, my thesis was on rice substitution lines in wheat, and it was considered at my supervisor had a table in his room, 21 wheat chromosomes and 7 rice chromosomes, and he wanted to produce all the 147 wheat rice substitution lines, and he used to make a cross when one substitution was over. But all that, you see, in Cambridge also work was done on wheat rice substitutions. E.R. Sears did, Manitoba, these were the three most important centers. But they all discontinued this work. You see, substitution lines and addition lines for alien chromosomes, large number of them have been produced, but none of them has been systematically used for transfer of genes. You just make a cross and then look around, whatever you get. There were, you see, in 1960s there were experiments conducted where a particular gene from agropyrin was transferred, a particular gene from cicale was transferred, but none of them led to release of a commercial variety. These were sort of proof of the concept. They demonstrated it can be done. But when it comes to actual developing a commercial variety, you still have to depend upon chance.
And
You see, cytogenetics, I have been cytogenetics, people call me that I work against cytogenetics, but the fact remains that cytogenetics could not deliver. Cytogenetics is utilized by nature to give you better products. But as a cytogeneticist, you cannot replicate what nature has been doing. You see, we wanted to do, for example, in wheat, I remember my teacher of plant breeding in 1964, he delivered a lecture in the first International Wheat Genetics Symposium, the first paper in the symposium, where he described that it was a mistake on the part of nature to utilize the D genome. Therefore, we should reconstitute the D genome. And one of his students developed methods where D genome had few chromosomes from one species, few chromosomes from another species, and few chromosomes, all desirable chromosomes from 3-4 different species. You are reconstituting a genome. Reconstitution of a genome, this paper was published in 1965-66 by one of the students who used to teach me, Laurie Evans. But what after that? None of them could give rise to a variety.
no, so these are not giving a variety, that I agree with you, but these are providing a variability, a usable variability which breeders can use.
Oh yes,
Yeah.
variability.
They are
Alien
providing
genetic
a
variability
they
is
are
a,
providing a
is a very important
Yeah.
source.
Yeah.
You see, alien genetic variation is a very important source which has been utilized in conventional plant breeding, and it will also be utilized in genetic engineering in future. Cisgenesis, intragenesis, they will be. It is a very, very important source. There is no doubt about it. So you have to examine and work on alien genetic variation, find out genes. Dr. Dhaliwal and his group, Parveen Chuneja in PAU, they have been doing it. They have been maintaining alien species in a large number, and they have identified a large number of genes even for biofortification, which they think can be utilized. True, they are very important source. But the point which I have been making for decades now, and Dr. Dhaliwal sometimes does not agree with me, is that can you plan an experiment that I want to transfer this gene, and then it leads to development of a commercial variety.
Yeah, it's not a breeding tool, but definitely it is a tool.
It is
in the process
important,
of
I
breeding.
agree with
It's a
you, no
pre-breeding
problem.
tool. It's not a
It is
breeding tool, it's a
important, very, very
pre-breeding tool.
important. Alien genetic variation has been utilized in a number of cases, not only in case of wheat, in case of rice, in case of tomato, in several species, brassica also. There are a number of examples where alien genetic variation has been utilized.
Thank you.
बट सर जो आजकल मॉलिक्यूलर साइटोजेनेटिक्स टेक्निक्स हैं, डज दे हैव एनी रोल? और
You see, again, molecular cytogenetics, what does it mean? Molecular cytogenetics means that you are mapping a particular gene on the chromosome utilizing molecular means. And if you have done it, so what? You see, for fundamental, don't confuse between real plant breeding leading to development of commercial varieties and the basic studies. Molecular cytogenetics is a basic tool. It generates knowledge and very important knowledge. There is no doubt about it. But can you use molecular cytogenetics, prepare a plan of work where molecular cytogenetics will be directly used and lead to development of a variety? That perhaps is not possible.
Sir, you have told us about EP alleles and EP reels and all.
Happy.
So, okay, sorry sir, EP alleles. So with that I want to ask one question is that EP alleles are developed by changing some kind of chromatin modification or DNA methylation,
Right.
or some kind of that. So how that is going to be stable over generation? That means, is there any kind of transgeneration inheritance?
They are stable. You see, the epi alleles, for example, DNA methylation. There are 3 different mechanisms of DNA methylation. One is methyl transferases enzymes are there. There are genes for methyl transferases. There is decrease in DNA methylation, genes are there. And then there is RNA dependent DNA methylation. These are the 3 techniques. But all these 3 techniques ultimately boil down to a gene which synthesizes the enzyme which brings about methylation. Therefore, in actual practice, when you will do plant breeding, you will really use that gene which is responsible for methylation of other genes. You see, the epi-RILs which I have shown, in that particular case, you are not really utilizing a parent which has DNA methylation, but along with DNA methylation, you are using a parent which is a mutant for the gene which causes decreased DNA methylation. So you have to, in all these cases, whether you are talking of microRNA, whether you are talking of long non-coding RNAs, whether you are talking of epi alleles, ultimately you have to identify a gene and the marker for that gene. So we are not really looking for DNA methylation and utilizing it. You are actually looking for a gene which is causing this methylation. And if that gene is there, then methylation will be caused. So you don't have to worry about it. All these epigenetics, you see, there is no doubt about it that epigenetics is transgenerational. 10 years ago, perhaps we were talking that epigenetics exist in the same generation. But epigenetics is transgenerational. It was discovered as far back as when paramutations were studied in case of maize, and it was shown that it is not mere, you see, paramutation, sometimes we used to say that it will disappear in 2-3 generations, but this, it might, but then it really depends upon a mechanism which we earlier did not know and now we know. And because we know the mechanism, you can capitalize on the causal factors, you can capitalize on the genes which are responsible for causing epigenetic changes.
So friends, as you all know that this program is live and I'm getting some queries from the other stations, and I just thought of bringing some to the notice of Professor Gupta. There are 3 questions which I have received from Professor Jaishankar, Telangana University in Telangana. And the students are asking question that the one is, what is the prospects of apomixis, fixation of heterosis? Question number 1.
Apomixis I don't think is going to be feasible in the near future.
Okay.
No doubt about that.
Second question is on is there any product for developed using marker system?
No, but
And
genomic selection I cannot say. Genomic selection has been more useful in case of animals, but marker assisted selection we have number of examples even in India.
marker assisted recurrent selection. Marker assisted recurrent selection and
Maybe
genomic
Prabhu
selection.
should answer that question.
The marker assisted recurrent selection, not recurrent selection.
I think Prabhu should answer that question.
We have developed the marker recurrent selection products now in wheat for drought tolerance and heat tolerance, and they are in the coordinated trial now, in the first year of trials.
No, I think private industry is utilizing it. In case of maize, more than 20 genes have been transferred in case of maize using marker assisted recurrent selection. That is a powerful approach, there is no doubt about it. And genomic selection also will be utilized in the future, there is no doubt about it. Because genomic selection is based on the concept that every marker must be associated with a gene, minor gene, and therefore you have to take the breeding values of alleles of each marker and then add them up and use it as a selection parameter. It will be done. The major problem with genomic selection is the prediction models which have been proposed. How accurate those? Accuracy of prediction models is being tested. Almost every year a few papers appear on this, and I am sure within 2 or 3 years we ultimately will have a prediction model which will be fairly accurate, and then genomic selection will certainly be utilized for actual plant breeding.
Thanks. I think Raman wants to add something on that. Before you do that, one more question that probably also you can address simultaneously. is on any product from site-directed mutagenesis.
Yeah, regarding this marker assisted selection, it is being utilized for development of the sclerotinia tolerance in mustard and canola in our programs. Marker assisted recurrent selection.
So in the interest of time, I'll keep it very short. So I think the markers, I mean, whether genomic selection or the whole genome prediction, is it really resulting in product development? There is a resounding yes from the industry side, I can tell you. And this is not anymore considered as a method which is different from your usual regular pedigree breeding method or any other conventional breeding method that you follow. So this is very much part and parcel of your regular breeding program. Using markers is a very routine, mainstream technology, and it has already resulted in a huge line of products. But they are not going to be branded as marker-assisted selection products, because in each and every genotype that we deal with, the line that we deal with, it has already been genotyped, and we are making use of that information in multiple different ways. In some cases, specific markers; some cases, it's whole genome markers. On site-directed mutagenetics, I'll perhaps if Amitav is here or no, it's Sreshti. Is it about genome editing or? Yeah, okay. So Amitav is here. He has just come in. So Amitav, the question is, this is a live question from Telangana University, our Hyderabad. So the question is, are there any examples of products being developed from site-directed mutagenesis? Sorry to put you in a fix. You have just come in.
Sorry, yeah. Yes, there are a couple of them. So there is a mushroom that has come to market. You know, that's from CRISPR-Cas, and there are a couple of others almost there. You know, so there are.
CRISPR-Cap is different.
Yes.
Talk of site-directed mutagenesis. You can also classify this as site-directed mutagenesis, but actual site-directed mutagenesis means that you are replacing one amino acid by another amino acid.
Yeah. So, sir, SDN 1, 2, 3, as you know that, you know, so this is there. You know, there are many examples of SDN 1 which are almost there, and 2 and 3 are also in the pipeline. You know, so
I know about this AGPS gene which I have talked about. There is a group in Florida who have done site-directed mutagenesis and demonstrated that it can lead to heat-tolerant maize, but not leading to a variety, commercial variety. Yes, please.
Sir, a wonderful summarization of the different tools which can be utilized for plant breeding. Like with your experience and the expertise in this area, like there has been a conception among the young group, like the gene editing techniques would be playing a major role in the near future.
Oh, no doubt
Where
about
do,
it.
where
No
do
doubt
we
about
see?
it. In India, we have to develop the expertise,
Yes.
and this CRISPR-Cas technology is considered to be the easiest and the most convenient and the best technique among the three: zinc finger nucleases and TALEN, CRISPR. And I have been reading for the last 1 year. Every day there are papers on CRISPR, and CRISPR is going to win a Nobel Prize. There is no doubt about it.
Sir, where do you see, like in the near future, what are all the?
No, on CRISPR there was a paper published from Navi. If you have seen
Yes.
it, they have used CRISPR in case of wheat.
Sir, in this regard, with respect to especially training the students, where do you see, like what kind of training a student has to be
It's a
equipped with in
difficult
order to
question
be a
because I have
good?
never done CRISPR therefore. But training of a student should be done. You see, we are going to have a seminar on 23rd of November on this genome editing in NSC complex. We are going to discuss in greater detail about it. But I think abroad in many labs this is being successfully used. There is no problem about it. I gave you an example of genome editing for developing male style lines. That is also genome editing, and I am sure many more examples. I know that there are also examples of developing commercial varieties through CRISPR-Cas. I can't give you the examples right now, but I have 3 or 4 examples available on CRISPR-Cas where it has been used for development of commercial varieties.
Sir, I would like also to take this opportunity to talk about the Vaxicon that I talked yesterday. So in our company also, we have developed Vaxicon with CRISPR-Cas, which has been notified by USDA as not regulated. Since I spoke only yesterday, I didn't give that example. I was giving the mushroom example. Thank you.
Sir, we have started using the CRISPR-Cas technology in rice already for ABA receptor and some of the genes which have been shown to be negatively regulating drought tolerance. We have already developed the construct. We are doing the transformation in progress.
I'm sure it will be utilized. There is no doubt about it. You have to prepare a list of genes which you would like to attack and develop the constructs. On the face of it, it appears to be simple, but there must be, I'm sure, technique you have to standardize, and the students need to be trained in all these new areas. The students should be trained, and the students should be excited to undertake such research, as I feel excited in undertaking new areas like microRNA, epigenetics, and you see, I always like to move to a new discipline, new area, because it is so exciting to read it and then try to demonstrate that it can be used.
Sorry to intervene. We are all very excited to listen to you, sir, and all of us are excited and have many questions in mind. But but I request everyone, anyone having doubts, please interact with Professor P.K. Gupta. He will be around throughout the day. And let us continue with the next thing. Thank you, sir.
A huge round of applause to the Professor P.K. Gupta sir for a wonderful deliberation and also for answering the queries very enthusiastically. Thank you, sir. Thank you so much. We have the next speaker, Dr. A.K. Singh sir, who will be speaking about molecular breeding applications in rice improvement. Dr. A.K. Singh has made pioneer contributions in basics and applied research on rice genetics and breeding. His research has led to development of 13 basmati and aromatic rice varieties and a hybrid, which has improved the livelihood of millions of basmati rice farmers and won him national recognition. He has been awarded with prestigious Rafi Muhammad Kidwai Award and Dr. Subramaniam Award. He is a teacher of par excellence and authored a book on marker-assisted breeding. Sir, please.
Thanks, Bhuvaneshwari. It's a difficult task to speak after Professor Gupta. He is always ahead of time. For today's lecture, he came at 9 o'clock here. But anyway, that was my mistake. Friends, I'm going to talk to you about some of the work that we have done on marker-assisted breeding, leading to straight product development in case of rice, and more so in case of basmati rice. You know that traditional varieties of basmati rice, they are very tall, they are photoperiod sensitive, they are prone to lodging, they have a duration of about 160 days, and they yield about 2 to 2.5 tons per hectare, very poor productivity. And therefore, a program was started to combine the quality of traditional basmati into high yielding background. And the first variety that was released was Pusa Basmati 1 in 1989, which was semi-dwarf, high yielding and excellent grain and cooking quality. Later in 2013, we came out with another variety, Pusa Basmati 1509, which has a duration of 120 days, and it saves about 35% of the total irrigation requirement, saves a lot of water with very high productivity, and you can see the cooked kernel length is almost inch in length after cooking. The progressive development that has happened from 1930 onward, when the first systematic release of Basmati 370, one of the traditional Basmati variety, was done, had a grain length, cooked kernel length that you can see, and progressively in 1989 Pusa Basmati 1, then 1121 in 2003, the most popular variety even now, and 2013 the 1509. How, of course, at that point of time we did not have knowledge of molecular markers, genes involved, pyramiding of QTLs. Obviously, this is a clear example of pyramiding QTLs for grain length, for elongation and other characters, which has been achieved through conventional breeding approaches. But with the advent of these tools, now it is possible to dissect whether the genes resides, which parent is contributing, and how they can be better manipulated in the breeding program. So overall, what has happened, the duration of traditional varieties, which was about 160 days, has been reduced to 125 days in the dwarf varieties, and the productivity has enhanced from 2 tons per hectare to 6 tons per hectare, which is a remarkable gain, because generally the longer duration, higher is the yield. but we have reduced duration with higher yield. And this has resulted in the tremendous increase in the foreign exchange earning for the nation. Currently we are exporting basmati rice to the tune of 4 million tons of about 5.2 billion US dollar every year. So that has happened possible because of the improved productivity in these varieties. But there are a lot of challenges which are lying ahead of us, and these challenges include both biotic stresses and abiotic stresses. The answers in case of biotic stresses are far more simple and straight. Abiotic stresses are still complicated. Yesterday we had excellent presentation from Pioneer partners on phenotyping and precision phenotyping and high throughput genotyping, which will be the key to address to the issues related to quantitative traits and complex traits like drought, submergence and others. But I have some successful examples for biotic stresses where we have it has resulted into product development. The major problems in rice are bacterial blight, blast, brown plant hopper and sheath blight. These are the major biotic factors which reduce quality and yield. As far as bacterial blight is concerned, there are 39 genes known. Many of these genes, they come from wild species. Many of these genes have been reported into cultivated, but none in basmati rice varieties. They are all present in non-basmati sources. And so when we transfer genes from non-basmati sources into basmati varieties, the quality is impaired. It is distorted completely, and it takes a lot of time to reconstitute the quality traits through backcross breeding program. And that is why the marker assisted backcross breeding has come very handy in reconstituting the recurrent parent genome with additional trait from the donor. Now, since the viewership is a large community of students sitting in this hall and also outside, I would like to briefly tell you what are the major steps that are involved in marker assisted backcross breeding program, where you are trying to improve a otherwise excellent variety lacking in a specific character. The components are foreground selection, which refers to selection for trait of interest using marker linked to the gene of interest. It could be a gene based marker, or it could be the best case a functional marker, as Professor Gupta was describing, which will not lead to any problem of false positive identification through recombination problem. The second step in marker assisted backcross breeding program is recombinant selection. So when you are using a donor which is little diverse, and whenever a donor segment is coming with a desirable gene, there is always possibility of having a flanking genomic region, and this flanking genomic region can bring some undesirable traits, and therefore that has to be minimized to the extent possible, and this can be done by using the flanking markers to the target genes and looking for the recombination at the flanking marker loci for the recurrent parent allele. So recombinant selection, of course, we should there is a lot of discussion whether we should really invest so much of time on recombinant selection, and I would say that the experience varies from case to case, the cross that you are handling, crop that you are handling. Unless the linkage drag is a severe issue, we should not invest too much of time on recombinant selection. If linkage drag is obvious, then obviously it is extremely important. Background selection using a set of polymorphic markers between donor and recurrent parent, and these background markers can be used for recovery of recurrent parent genome. Now, there can be debate on how many markers should be used for background analysis, whether in general we say 5 to 6 markers per chromosome. In case of rice, about 60 to 65 markers will provide sufficient genome coverage to reconstitute the recurrent parent genome or not, are the questions which can be debated. Whether a chip containing a large set of markers, for example, SNP chip containing 50K markers in case of rice should be used for background selection, and what will be the cost involved in doing all this, are those the factors to decide about it. But most importantly, there is a perception that when we are using marker assisted backcross breeding using foreground, background selection, recombinant selection, probably we can do away with phenotypic selection. Our experience has been that phenotypic selection still remains extremely important, whether it is a question of target trait that you are introducing or the background trait recovery. What stage, to what extent it should be practiced is extremely important, and that needs to be discussed. As far as marker assisted background selection is concerned, we must understand that the markers would only help in identifying a genotype if it is there in the population. The markers will not create a genotype. Now, having that genotype with a set of say 60 markers in a population is a number game. Larger the population, better the chances to identify a marker or a plant which has maximum recovery of recurrent parent genome. If we are having large number of markers, say 50K chip, but our backcross population size is 100, the 50K chip is not going to do any good, because unless the recombinant is there, we cannot identify. So, this is one critical factor that we have to take into account. The phenotyping for therefore target traits, yield, quality, stress is extremely important. So, we started using marker assisted backcross breeding program to combine the bacterial blight resistance in one of our basmati variety, Pusa basmati 1, and the first product that we developed was improved Pusa basmati 1, which had the gene XA13 and XA21 in the background of Pusa basmati 1, and we empowered farmers to fight blight, and this variety was released in 2007 with resistance to bacterial blight. Further, the blast is another major problem which farmers have to use a lot of fungicides to control blast, particularly neck blast problem, and therefore we thought of first creating a set of isogenic lines for blast resistant genes, major blast resistant genes. There are about 103 blast resistant genes known so far. Again, they are all in non-basmati sources, not in basmati variety. Professor Kus had done extremely good job in creating isogenic lines for blight and blast in IR24 background and CO39 background, and therefore we thought we should have a set of isogenic lines carrying different major blast resistant genes in one basmati variety, and for that purpose we chose Pusa basmati 1. So, we took gene like PI1, PI5, PETA, PI9, PIZ and PIB. These are 7 major blast resistant genes, and we followed the marker assisted simultaneous but stepwise backcross breeding program to develop 1 gene, 2 gene, 3 gene, 4 gene, 5 gene and 6 genes pyramided isogenic lines in the background of Pusa basmati 1. And one of this line, you can see how these isogenic lines for the students' purpose you must see. In the middle you see Pusa basmati 1. The flanking rows are isogenic lines which have exactly the same height, maturity and other characters, but they have got resistance to blast disease. One of this from this project, we see here the point that I was emphasizing, that the donor line that you see with red kernel there, IRBL9W, this carries PI9 gene. But this has got red pericarp, which is undesirable character, chalky grain, bold grain. On cooking it swells, it doesn't elongate. And when you make a cross with this with basmati genotype, the all characters they get disturbed. So, repeated back crossing is required and phenotypic selection is required, and you can see the improved lines. They have got excellent grain and cooking quality, as good as Pusa basmati 1. Now, one of this you can see here the reaction of these lines. The Pusa basmati 1 at Uniform Blast Nursery in Milan in Himachal Pradesh, this is completely susceptible, everything is killed. But the same Pusa basmati 1, when added the PI9 gene, this is completely resistant. And this PI9 gene for some reason has been found to be very, very effective in different areas. We have tested in the Uniform Blast Nursery in Hazaribagh in eastern India. Then we have tested in the hilly region in Almora. We have tested in Mugad in Karnataka, but this is a very important gene which confers resistance to most of the isolate. Now, I was referring to background selection. In one set, we used background selection using about 65 markers polymorphic between the donor and recurrent parent. And when we compared the data of those 65 SSR marker with a 50K chip data, then we find that the recovery when we use SSR marker versus SNP chip with a large number of markers 50K, you have a overestimation of data that is presented by SSR, limited number of markers as compared to when you increase the number of markers, because you get real picture in terms of recovery of the recurrent parent genome. And this has led to this project has led to development of a new variety that we have released in 2016 through Central Variety Release Committee in the name Pusa basmati 1637. This is Pusa basmati 1 having PI9 gene, excellent productivity. We had a field trial on farmers' fields. You can see the crop is excellent performance, 6 ton plus crop in case of basmati with protection against blast disease. Further, we have added along with bacterial blight and blast a major QTL for sheath blight, which is a major problem in case of basmati rice in particular and rice in general. Then likewise we have two more varieties, Pusa basmati 1121 and Pusa basmati 6, which we are improving for blight and blast resistance with different gene. And this paper came in plant science where we brought in the gene for bacterial blight resistance XA13, XA21 in the background of 1121. This variety has also been identified and next year it will be released. The reaction to bacterial blight isolates from different areas and also to blast it provides very good resistance level. And the grain and cooking quality that is reconstituted similar to the recurrent parent. So this variety in the background of Pusa basmati 6 with XA13 and XA21 has also been released in 2016. We had simultaneous release from the a trial. We have separate testing for the mass derived product where the leverage is given. The mass derived lines are tested only for 2 years as against 3 years in the regular testing. And these two varieties from the same trial both have been released in 2016, one in the background of Pusa basmati 6 and other in the background of Pusa basmati 1 for blight, 1 for blast. Then a new gene has been recently introduced from Oryza nivara XA38, and we have along with this gene XA13 and XA21, now we are putting XA38 in the background of these varieties so that we get a wide range of resistance. So we have developed isogenic lines of 1121 carrying XA38. This paper came in scientific report very recently, and you can see the grain and cooking quality of the XA38 carrying lines in the background of 1121 is as good as 1121, while this is the donor XA38 that we have used in the breeding program, which is a non-basmati variety. Salt tolerance is a important trait, particularly in the state of Haryana, where underground water is brackish water and the crop establishment is badly affected. A major QTL saltol that has been transferred from Pokali, a land race from Kerala, has been mapped by IARI group, and we undertook this program. Professor Gupta was leading this program at DBT to transfer biotic, abiotic stress tolerance in many varieties. And now we have introduced the saltol through marker assisted backcross breeding in 1121. You also know that the transplanting is a expensive operation, and therefore farmers are slowly moving to direct seeded rice. Now when we come to direct seeded rice, there are three main culprits. The one is weed, other is iron deficiency, and third one is nematode. And therefore these three problems must be addressed if direct seeded rice has to succeed. And to address this problem, we had a tilling project in Nagina 22 background. We created a mutant in ALS gene, and this ALS mutant provides resistance to immediate hypergroup of herbicide. And you can see here that this is a STM herbicide tolerant mutant in Nagina 22 background, which after spraying herbicide is completely tolerant, while the variety Pusa basmati 1121 is highly susceptible, it is completely killed. Now this gene, we know this is ALS gene, we have mapped, we have identified the markers, gene based markers, and this we are using to transfer the gene through marker assisted backcross breeding to 1121, 1509 and Pusa 44, so that these varieties can be mobilized into direct seeded rice area. Likewise, we had a hybrid Pusa RS10 that was released quite some time back, which has been improved for resistance to bacterial blight and blast, both the parental lines. And one of the parental lines, the male parent, has been released as a variety directly as Pusa 1609, carrying gene PI54 and PIZ5. Then we have 1612, which has been released, and yet another product that has come out of this is Pusa 1592, and these are all three are Central Variety Release Committee's releases. This carries isogenic lines for bacterial blight gene XA13, XA21. Further, in non-basmati background, we are doing a systematic breeding program to bring in gene for resistance to blast diseases, particularly in case of BPT 5204, which is known as Samba Masuri, one of the most popular varieties in quality rice category. And it was improved for bacterial blight resistance by Directorate of Rice Research and CCMB in collaboration. We have added genes PI54, PI1 and PETA. This line is in advanced varietal trial too. This morning I took time to take to Professor Gupta to the field to show this material. So it will be released soon. Likewise, along with the blight, now the blast gene is also being put in Samba Masuri background, which will be shortly available. Drought tolerance is a very important trait, and fortunately in past few years a good number of drought tolerant QTLs have been mapped from variety like Nagina 22, EPO, Vairaram, and these QTLs, they are robust major QTLs which account for more than 10 to 15% of the phenotypic variation, and one of the QTL comes from Nagina 22, which is DTY 1.1, which we have transferred to Pusa Basmati 1 now. The idea is that by transferring these QTLs, somebody may ask a question that when you have irrigated ecology, what is the utility of having the QTLs in them? Our presumption is that if we can reduce the number of irrigation by putting these 1 or 2 or 3 QTLs for drought tolerance in irrigated ecology, rice generally is requiring about 22 to 25 irrigation per crop season. If that can be brought down to 15, that will be a huge water saving. So this has relevance. Likewise, a QTL DTY 3.1 from EPO has been transferred to Pusa 44. A major problem that we have been facing in case of basmati rice is Bakane, a disease which is caused by a fungus Zibberella fujikuri, and this fungus produces gibberellic acid. Because of it, the plants, they elongate and finally they dry. This is a picture taken on a farmer's field. You can see how badly the crop is affected. The field gets a very ugly look, and the productivity loss is about 15 to 20% in general. So we did a QTL mapping exercise, and we had one of our breeding line, Pusa 1342. You can see here, this is treated with the seed is inoculated with the fungal inoculum by pre-soaking for 24 hours, and this is controlled. So on inoculation, the susceptible parent is completely dead, while 1342, the resistant parent, there is no difference between treated and controlled. We had a recombinant and line population from 1342 into 1121 cross, which was used for QTL mapping, and we identified 3 major QTL, 1.1, 1.2 and 1.3, and also 3.1, a minor QTL. And these for the major QTL 1.2, we performed QTL-seq analysis, and this is identified in the same region, and based on QTL-seq analysis, we have identified a candidate genes, which is putative genes, and now we are verifying this using the RT data to verify the candidate genes. So now the marker is identified, and through marker assisted back cross breeding, we are mobilizing this QTL for Bakane resistance into all our basmati varieties. Professor Gupta mentioned about development of multi-parent population, nested association mapping population. My colleague Dr. Gopal Krishnan is working. We had, as a matter of fact, 3 common parents, only one is shown here, and 22, and we had 25 founders, and now we are in almost F5 stage with these 25 populations coming out of NAM populations. Once this is available in rice, we will use it for mapping different traits. Golden rice has been a very favorite project I have spent for more than, I think, 12 years now, starting in 2002, but it seems still a long way to go. We transferred this gene from a donor line in the K-bonate background, which was developed by Syngenta, using that as a donor in the background of Swarna, one of the most popular variety. And you can see the expression levels are pretty high, about 25 microgram per gram of endosperm is total carotenoid expression. But when we developed these lines and fixed them to homozygous state, we were surprised to see some major phenotypic aberration. So we found that the plants which are homozygous for the transgene, they become dwarf, tiny, and hemizygous and null plants, they are fine like recurrent parent. Further distortion that happened in the phenotype is that all the plants which are homozygous for the transgenes, they have got reduced panicle length, number of grains is drastically reduced, and the pencil exertion is badly affected, and therefore in spite of having high carotenoid content, this line cannot be commercialized used because of the poor agronomic performance. And now when we found this problem, one of my students, Harita, started working this as a PhD problem to find out what are the reasons why this kind of aberration in phenotype is happening. And she discovered that during the transgene insertion process, we know that transgene insertion is a random process. This transgene insertion had happened in the OX1 gene, and because of OX1 gene disruption due to insertion of transgene, the auxin metabolism in these transgenic homozygous line is badly affected. As long as the plant is hemizygous, you have one OX1 normal copy, and that provides sufficient oxygen for the normal growth and development of plant. But moment you self a BC3F1 plant or BC3F1, BC2F1 plants, and the transgene becomes in homozygous condition, both OX1 copies are disrupted and oxygen level is drastically reduced, which we further verified by chemical assay and also by RT data. And when this was brought out, the work on this particular event called R event was discontinued. We were working for last 7-8 years on this, and in 5 different countries, but the whole the work is discontinued. Now we have shifted to a different event called E event, and that will take its own time to come to a final stage. I would like to thank all my colleagues, Gopal Krishnan, who is working particularly on the Samba Masuri improvement, NAM population development. Prabhu is part of our entire program and has been doing a very great job on many policy issues, in particular registering GI for basmati rice, redefining basmati, and many more issues. Pralay is working on hybrid rice development. Nagarajan, Vinod, they are in off-season Aduturai nursery where we use our off-season crop. Dr. Twal, Rakesh, they are basically our seed scientists at our regional station. N.K. Singh, Mahapatra, T.R. Sharma from the biotechnology group, we had very strong collaboration. My student Ranjit, now he is a scientist with our group, has been working on transferring XA38, and now he is going to take up work more on transcriptome side. And Harita, she is working on the nutritional biofortification of rice, including iron, zinc, phytic acid, and several quality traits. Thank you all very much.
So Professor A.K. Singh, a very comprehensive presentation, and really commend your group for doing such excellent work on knot diseases by both biotic and epigenetic and rice. So my question here is, when you talked a little bit about drought, you talked about direct seeding, you've talked about weeds, and as a policy and in terms of rice cultivation with 100 million plus hectares across South Asia and the importance of this crop, where do you see this going if you are able to get drought tolerance plus herbicide tolerance into some of these elite genetic varieties that you have? Do you see that this will be the next source of increasing that area that Dr. Swaminathan talked about yesterday and getting more productivity from areas which are direct seeded or which may be purely rain fed? Thanks.
So the main issue is that this change in cropping system will happen for certain compulsion, and one major compulsion that I see is the labor that is involved in the transplanting operation, particularly in the states where labor is not available. For example, entire Punjab, Haryana, Western UP, I am talking of India, the transplanting operation is based on migratory labor that comes from Eastern UP and Bihar, which because of the local employment available there, the labor is becoming a major problem, and the farmers therefore have started slowly realizing the importance of this, and on their own they have started in fact practicing direct seeded rice, but they have to be supported by technology if this has to succeed. So I see a great future, and particularly putting in drought tolerant QTLs because still the often the comparison is made between rice productivity in China and rice productivity in India, and we stand nowhere because of the fact that in many fragile ecosystem, more than 50% area falls into fragile ecosystem, either suffering from drought or from submergence, and same area sometimes suffer from submergence, sometimes we suffer from drought at different stages. So in rain-fed environment, having drought tolerance is going to be the key to improve productivity from say about 7 to 10 quintals on average that we are getting, and we can bring it to about 12 quintals per hectare. That will be really good. So I see there is a lot of future for the drought tolerant rice, certainly.
Sir, thanks once again. As my colleague Anand put it, so it was an excellent presentation. So this is something I always tell everybody that in IARI, the teachers not only teach, they practice what they preach. So you see that the markers are being put in product development, and that's something excellent to demonstrate. So I'm just wondering, based on my previous experience working with the public system as well as the CG system, I have always felt students as well as researchers, we spend a lot of time trying to generate the data rather than analyze the data. So for instance, say, do we have a high throughput genotyping center in ICAR or even for IARI? So I just submit the samples, I get the data back, and I work. I spend a lot of time analyzing the data and making use of that data rather than trying to generate a lot of, that data. I end up spending a lot of time in that one. And another point is there an organizational wide or institution wide breeding principle in place for crops? Say, this is my hybrid rice, this is my hybrid corn, this is how I am going to breed. So what you are doing is you are actually eliminating that people dependency. So breeder may come in and he may go out, but then there is an organization wide breeding philosophy in place, so that you continuously monitor the genetic gains, you continuously you are able to deliver on what we are promising. So whereas I see that there are a lot of steps in the process wherein it's highly people dependent, so people come in and go, and along with them programs come and go. So we see a lot of gap there. So yeah, I'm wondering, it's not about the presentation, it's just a simple thought. Since you are our teachers, you have always answered us, so I'm just asking you.
Thank you, Raman, for that question, and it's a very pertinent question, and I have no hesitation in accepting the fact that most of our marker-assisted backcross breeding program, as of now is grossly restricted to marker-assisted backcross breeding, and in that also foreground selection more so. Background selection is added as a component many times. I'm not talking of the exceptions are always there, but as a routine I am saying this is what is happening, that you need to make sense of data in terms of publications and good papers and so on and so forth. The phenotypic selection and therefore has become extremely important in most of these programs to realize the product. But the stage has not reached, and there are several considerations in terms of facilitation of genotyping, as you said. By time we have nursery coming up, we decide that we do the analysis and identify the plants which are positive for the foreground, and then remove rest of the plants and so on. In foreground positive plants, background analysis should be done. Only those plants which are having maximum recovery should be taken to field and for backcrossing purpose. You plan for every stage, it does not happen, and finally you end up with making crosses at random on 20, 30, 40 plants, and then you get the data which plant is best to select for. So those are the real problems in terms of operations that need to be addressed as a system. We had a very massive program proposed, one consortia research platform on molecular breeding, CRP and molecular breeding. And there exactly our proposal was the same as you are saying, that we have a regional genotyping center, one in say in Delhi, one in Hyderabad, one in Bangalore, one in Northeast, and these regional genotyping centers The public sector institutions will provide the infrastructure in terms of equipment, facilities and other. We give this facility to a vendor who will employ their people to run the machine 24/7 and pay their salary and everything, and they will charge us also for the services that they provide, but they ensure that the machine is run 24/7. And we had some industry partners willing to come forward to give this kind of proposal. I hope that this kind of facility gets in place. Then only this will become a routine activity in terms of genotyping. Is a major constraint as of now, there is no doubt about it. It also many equipments, you see, one is that we have enough equipment. For example, if you take ICARI institutes in Hyderabad alone, and if all institutes, whether it is sorghum, rice, oil seeds, the facilities are put in one building, you will have maybe something like 5-6 sequencers, which are not used 10% of the time. So that is a problem of management to put it to maximum use, and something of that sort has to happen. As far as continuity of program is concerned, the lineages, pedigree of the material that is dealt with, of course, within program we have continuity. For example, my predecessor Dr. Swaminathan started this work, Dr. Siddiq joined, Dr. V.P. Singh retired, I have joined, now I have my colleagues Gopal, Ranjit, Harita. So they understand, because there is sufficient overlap period of 10 to 15 years working together. So that problem really doesn't come. We understand fairly well what material we are dealing with, and that is where, and wherever this kind of understanding is not there, you don't see that kind of progress happening. You see, IARI is not the only center working on basmati rice. There are 8 centers working on basmati rice in the country in the GI areas. But when you see in terms of product, it is 80% product from IARI, not from other centers. So that's a very critical, important point. Yes, sir.
To begin with, Ashok, I compliment you heartily for the excellent summarization of your work on basmati rice and other rices you touched on. You had been a very successful breeder before you used markers, and you are still a successful breeder when you are using markers. Before the advent of molecular era, the successful breeder was one who has the feel for the plants he was working with, and he could have a judgment of which plants are going to do what for him. Markers somehow give you that information through their association with the characteristics that you are doing. So 2 questions on this, possibly students would like to know. One, has the use of markers made you a better plant breeder? 2, in the future, will markers and molecular technology replace the breeder by machines and programs that make the decisions?
Okay. As far as adding efficiency is concerned, we have been doing fairly good in terms of product development before the marker era, but all the work that we have started on biotic stress resistance breeding, we virtually did not do anything on bacterial blight, blast and brown plant hopper, these biotic stresses, for which with the knowledge of markers, availability of genetic resources were available earlier also, but with the advent of markers, the dependency on phenotyping for environment, for hopper to come, for disease to come, those things reduced to a great extent, and this has really accelerated our incorporation. Trait correction program has been definitely accelerated to a great extent, use of marker, there is no doubt about it, and all these 6-7 varieties that we have released is product of that. Although I have a feeling that many of these varieties, you miss at one or the other points as compared to recurrent parent, which becomes a critical factor in deciding the success of the new variety. And one such example I will say, for example, this improved Pusa basmati, when we developed this variety, Gopal as a PhD program, he started working on this. When the variety, we were all satisfied with performance, coordinated trial released it based on performance, disease reaction, yield and everything. When it went to farmers, the farmers started complaining that this variety has got a little hard threshing, and a labor which is asking for say 3000 rupees for threshing its recurrent parent will ask for 4000 rupees to thresh the improved variety, because he has to beat with hand 3-4 times. Basmati is threshed manually, because if you use combine, the grains are broken because of combined pressure. So this simple trait of threshability was something that missed our attention, because we were not working at that level. Our threshing was based on, take single plant, beat with a stick. You don't realize if it is hard or loose. So that became a critical factor, and the variety did not really become popular as much as it should have become and replaced the recurrent parent. So these kinds of missing gaps are extremely important, that have to be looked into in spite of the facilitation that is given by marker. But certainly it has helped in product development. That's one point. And the second point, I don't think there will be a replacement of breeders with machine any time to come. I think that, but unfortunately, this is happening in the sense that with the markers coming into picture, the time that young generation is spending with the plants in the field is reducing. I'm not making generalization, there can be exception, but this is true that the time that the breeders are spending in the field is substantially reduced, and that is where the plants do speak but in whispers, and you can hear their voice unless you are in close contact with them. That is something missing with more of these tools coming. I think probably we will have to go to a level where the tools are so strong, so massive, that somebody yesterday was presenting data that you have 10 acres plot, and 100 in 10 acres plot you take the refractional image and you get the plant height data of 10 acres in 5 minutes, and much more precise and accurate. Now here we have to harvest 10 plants from each plot. Somebody had to sit on ground, measure the length, pencil length, plant height, and all those things. It takes lot of time, but if that kind of facilitation happens with accuracy, obviously it will facilitate the breeding program. So but as a country, I think we are still, that's one point that I felt has to be done a lot in terms of investment in infrastructure development, human resource development, computational aspect. We are lacking a lot.
So you have 3 questions coming from Angrao University. Dr. Balram, what event used for transfer of golden rice? Is homozygous condition transfer of the same event is showing the same effect in different backgrounds? That's the first question. Is phenotyping protocol standardized for neck blast? Second question. Third is any work initiated on breaking yield barriers in irrigated rice? Likewise, addressing problems of false smut and brown spot.
Great. So the first one was
first one is what event was used for transfer of golden rice?
Yeah, so the one which is not giving abrupt phenotype, I think he's asking that question. The previous one is R. Now we are using E event, but the expression level of E events are low. I'm not very happy with that per se, because R event has 25 microgram per gram of endosperm, and E event has only 11 microgram. Whether
he wants to find out whether it is background neutral or background specific.
I think it has been transferred to quite a few background using the scientists from Bangladesh, and also we have done in Swarna. So it seems to be background neutral, but per se expression is low.
And is phenotyping protocol standardized for neck blast?
Neck blast, yes. We have a project on incentivizing research in agriculture, in which neck blast was taken on priority, and 2 of our colleagues, Dr. Rathore from Palampur, he has been involved in developing protocol, and he has got fairly good development on this. We can share that information.
Next question is any advancement on breaking yield barriers in irrigated rice?
So as it was started long back by Dr. Kus using new plant type concept by introducing Japonica blood into it, and we have a network program in utilization of tropical Japonicas. Very good lines now in offerings with substantial increase in grain number. Hopefully we will have.
Any breakthrough in problems of false smut and brown spot?
these are the two major problems. I recently traveled, I was mentioning to you, in eastern UP, and it's a devastation, complete devastation because of false smut. No resistant source available so far, but this is also one problem selected in incentivizing research in agriculture project, and large scale screening is going on.
One more question has come in. How many basmati varieties were integrated with Saltol?
So we have done with Pusa Basmati 1 1509 and Pusa Basmati 1 3 varieties. But one problem that we see is that unless the reproductive, this is gene QTL Saltol confers salinity tolerance at seedling stage, and unless and until it is combined with the salinity tolerance at reproductive stage, per se it is not going to be much useful. So now we have identified certain donors for reproductive stage salinity, and we want to combine the both together to make it a commercially viable product.
Thank you.
Thank you. Thank you very much.
Ashok, this backcross molecular breeding has been questioned by many, in the sense, why can't you look for something which is better than your recipient parent? You see, as a plant breeder, you want to transfer a gene to a variety which is already known, high yielding and everything. For example, in your case you are transferring disease resistance, but in our case we transferred protein content, but during the period of reconstituting the recipient parent, it became susceptible. So you are back to another program for making it disease resistant. The question which was asked to me by Bikram Gill in China when I was making a presentation, he said, why should you reconstitute the recurrent parent? Why can't you look for something which is better than the recipient parent? If you do the conventional plant breeding, I mean, you don't have to reconstitute. Right. If you look in the same segregating population, there is always a chance of getting something which is better than the recipient parent.
That is true, sir, but the premise is that you have a variety which is widely adapted, agronomically superior, and lacking in one or two specific character which needs to be rectified. But if you can do that, you see, the principle of substantial equivalence, like in case of transgenics, also holds good with this, as long as your new product is substantially equivalent to recurrent parent. It doesn't become susceptible. If it has become susceptible, there has been something wrong in your procedure. Otherwise, if recurrent parent was resistant, why it should have become susceptible? That means you did not select for the genes for resistance. That is how it will become susceptible.
It became susceptible, the variety recipient parent itself became susceptible.
Oh, that became susceptible because a new race came, yes. But that will happen in pedigree breeding also. So in pedigree breeding,
you will keep on selecting, but the other traits also.
But it doesn't prevent, you know, you see.
In pedigree breeding, you will keep on selecting for everything.
So,
you are developing a variety.
So there is no restriction that you cannot select for a better phenotype than recurrent parent. Who says that? I don't know.
So why are you not doing this? That's the question.
No, because to do that you have to restrict the number of backcrosses, because you have to have donor segment. So you have to stop at backcross 1, then go for pedigree. Then you can do it. But then the other limitation comes that the product therefore developed will not be called as isogenic lines. It will be a new product and it will go for registration. So there is no restriction on that.
Ashok, you are in demand. There is one more question. What is the effect of zygosity in different backgrounds when you transfer a gene?
So, well, as far as the zygosity is concerned, this zygosity became a serious issue in this case because the transgene had disrupted a native gene. If transgene has not disrupted a native gene, then possibly zygosity would not have played such an important role. Is he asking a question that when transgene is homozygous condition, what is the level of expression? When it is in hemizygous condition, what is the level of expression? Yeah. Okay.
So speaking of the recurrent parent phenotype, as someone who's not very familiar with rice, I was struck by the ability to use recurrent backcrossing to get that basmati phenotype back. And so I was just curious, how much is known about the genetic architecture of what makes basmati rice basmati rice? Are there major QTL or is that highly polygenic? I was just curious about that.
So, well, I mean, it's again a complex trait. Basmati is just not one trait. To define a rice as basmati, it has certain parameters. The The first and foremost is that it should be aromatic. It should be aromatic. But all aromatic rices are not basmati. All basmati rices are aromatic. As far as genetics of aroma is concerned, it is governed by a major gene BAT2, betaine aldehyde dehydrogenase 2 gene on chromosome 8, and there is another gene on chromosome 4, which is the minor QTL. So these two, if they are addressed and the markers are available for both this, we can reconstitute aroma. Then comes the question of amylose content. The amylose content in basmati genotype should be intermediate, about 22% to get best expression in terms of quality characters. So amylose, we know there is a granule bound starch synthase gene, GBSS locus, and there is polymorphism for GBS locus between the basmati and non-basmati. That polymorphism can be used for reconstituting amylose. In addition to that, we have gel consistency, gelatinization temperature, and cooked kernel elongation, which is a very important character. The basmati rice must elongate twice its original length on cooking. If it is better, it is still better because you can make more plates from a limited quantity of rice that you cook. So it is good for the hotelier industry. So if you have say 4.5 times volume expansion, that rate we are still looking at, but cooked kernel elongation, that gene is known, QTLs are known. Also the grain length per se, there is a G2C change, which may A to C change that makes kernel long. If there is a premature termination of polypeptide, the kernel becomes long. If it is C, it will be A, it is small. If it is C, it becomes long. So this is a mutation that has resulted into gain of function, and such mutations can be created by targeted approach of CRISPR-Cas kind of situations. But the genetic variation is available for which the marker is known, and wherever for all these 7 characters that I have mentioned, for some of them markers available, we are using those markers for. But test of putting lice in eating it, because there is above all there is a mouth feel is a trait, and mouth feel is something which cannot be described. It will be something different for you and it will be something different for me. So we have to have a panelist, 5-6 panelists who understands basmati, and then they give their final judgments. Thank you very much.
Thank you, sir, for the excellent deliberation and answering the queries. Now we'll break for coffee for 10 minutes and kindly reassemble for the session to continue. Thank you.
I would like to request all the dignitaries, please take your respective seat. Very good afternoon and welcome back once again in this student symposium, Genomics based next generation crop improvement approaches. Now we are going to continue our third technical session, which is about application of genomics. In this first, I would like to invite our next and the last speaker of this session, Dr. Firoz Hussain. Dr. Firoz Hussain is a senior scientist and maize breeder in the division of genetics IARI. He has received his PhD degree from IARI in the year 2005 and is currently pursuing research in the field of nutritional quality and specialty trait in maize. His team is involved in developing the country's first beta-carotene rich maize hybrid, which is currently under AICRP testing, and today he will be delivering a presentation on genomics for nutritional improvement of tropical corn. Please, sir.
Very good afternoon. So I shall be presenting the topic on genomics for nutritional improvement of tropical corn. Now, students, you had an excellent 2 lectures by Professor Gupta and Dr. A.K. Singh, and you had beautiful interactions with them. Dr. A.K. Singh has expressed the challenge he had after the presentation of Professor Gupta. Now you understand my challenge to present after both Dr. Professor Gupta and Dr. A.K. Singh. But at the outset, I must say, I must sincerely pay my gratitude to Dr. B.D. Singh, whose book has at least inspired me to select genetics and plant breeding in the 4th year of our undergraduate program. And I'm sure all of us here, the students, they are still continuing. Your books are still inspiring the students to join in the field of both genetics and plant breeding, and same is the case with Dr. P.K. Gupta sir. His book has also inspired. On the other aspect of it, Dr. Babu has mentioned that we at IRI, the students are very lucky. We have seen Dr. A.K. Singh, Dr. Prabhu, Dr. Prasanna, Dr. T. Mohapatra, who have taught us, and we have learnt all those things by watching them doing the science over here. So I'll try to present the work which we are doing at IRI. So the micronutrient deficiency has been one of the important area which requires an urgent attention. World's out of 7 billion people, 2 billion people are malnourished, and 45% of the death below 5 years are linked to malnutrition. And malnutrition remains the #1 risk factor for the global burden of disease anywhere in the world. And it represents loss of 11% of the GDP every year in Africa and Asia. That's what the IFPRI report 2016 says. And these are the maps in which it shows that extent of vitamin A deficiency in preschool children. Darker the color, higher is the severity of the problem, and India lies in the severity zone. Here it is for the iron. Here also we are in the severity zone, and this is for the zinc deficiency, where our country falls under that severity zone. So therefore, micronutrient deficiency is not only a problem across the world, but our country too have a problem of micronutrient deficiency. Now we have a sustainable development goal which has been institutionalized in 2015 by a global community who came together and formed 17 goals for a global development agenda that needs to be fulfilled by 2030. And the core is to eliminate extreme poverty, hunger, and malnutrition. These are the 17 goals they have highlighted, and one of the goal number 2 is on hunger and nutrition. But one of the important factors is among each goal there are different indicators. Orange indicates the nutrition. So therefore, out of 14, 12 is the indicator which is related to nutrition. Like that, you understand, out of 11 of the goals, nutrition is the core to the solution of that problem. Now, regarding maize, we all know it is an important source for food and feed. It provides 30% of the food calories to the 4.5 billion people in 94 countries. World produces highest 1,000 million tons, which is highest among cereals. And in India and Asia contributes 1/3 of the total production. So Asia remains an important zone, and our country produces nearly about 24 million tons of grain annually. But contrary to the world utilization pattern in India and in Southeast Asia, the food is only for the 10%, but the majority, around 60% of the maize is used for the feed. But in Africa and in Latin America, it's something the other way around. But nevertheless, maize remains an important aspect of food and feed. We know that biofortification, so therefore we have different approaches for the hidden hunger or to alleviate micronutrient malnutrition. We have supplementation, commercial fortification, dietary diversification, and biofortification. But among all these 4, biofortification is the most cost effective, sustainable, it reaches target people in pure form. There is no involvement of the distribution system, which can make an important delay in delivering of those goods and can reach the poor quickly. So therefore, it is one of the viable options. And with this, in maize, a lot of nutritional improvement have been there, and I'll cite examples one by one. And if anybody talks about nutritional improvement in cereals, across any cereals we talk about, the quality protein maize will come as the first example. We know that we require around 30 mg of lysine, 4 mg of tryptophan per kg of body weight per day, but maize provides less than half of the recommended dose. And we know the famous mutant OPEC2, which provides the twice-fold of lysine and tryptophan. And because of this opaque nature, the mutant was named as OPEC2. And later on, we know the history. Dr. Wassel and his team in CIMMYT has introduced the modifiers and developed high lysine and tryptophan-based maize, which is popularly called as quality protein maize. As far as the history of India is concerned, Shakti Ratan Proteina, the 3 composite, which was released in 1971, and these are the first set of biofortified cereals or across any crops which has been developed through targeted breeding approach way back in 1971. The effort still continues. We have now around 12 hybrids in the name of Shaktiman series, HQPM series, and others. So we have now maize varieties which are high in lysine and tryptophan. And this Vivek QPM9 is the first example of market-assisted selection-based maize hybrids, where Dr. Babu himself sitting over here, he actually initiated the work at BPK's, and they have come up with the first market-assisted selection product in maize that has been released, and which has got 30% more lysine and 40% tryptophan. And this is the essentially derived variety having high lysine and tryptophan, and the original variety was Vivek Hybrid 9. Then after the success of this, DBT funded a network project under the name of Accelerated Crop Improvement Program, where Professor Gupta is the chairman of that committee, and several hybrids across centers were given responsibility to convert already available QPM normal hybrids into QPM hybrids by introducing OPEC2 allele. This is what we have done at IARI. We have converted 5 hybrids under that program, and out of 5, 3 of the hybrids have been put in national coordinated trial, and it is being tested as an EDV. And we are happy to share here that in every aspect we had a almost 100% to 50% increase of lysine and tryptophan in all these varieties. So therefore, it is now nutritionally more rich. And all these 3 varieties, 4, 8, and 9, were tested under ACRIP in Kharif 2014 and 15. and blue is the bar for the normal and red is the bar for the QPM. This is the yield potential we achieved under ACRIP, and everywhere it has been found to be statistically at par with each other. So therefore, that is the crux of marker-assisted breeding program, where we improve a trait by keeping yield and other characteristics as similar as that of the original one. We went ahead with another addition of gene, that is OPEC 16, that is present on OPEC chromosome 10. So far, all the QPM hybrids developed in India and elsewhere is based on the OPEC2 gene only. So we got this OPEC 16 gene from China, and Dr. Wenpan Yang is the person who has published this paper in 2005 and reported the benefit of this OPEC 16 allele, and it was said that along with OPEC2 and OPEC 16, it gives higher lysine and tryptophan. So we introgressed both OPEC2 and OPEC 16 in already available QPM hybrids, which is released, and this is what we are at our lines are now at finished product. And this is the normal line, which has got something around 0.12% of lysine. This is the original HKI 161, where because of the OPEC2 it is around 0.3, but these blue lines are having both OPEC2 and OPEC 16. So because of the introgression of both OPEC2 and OPEC 16, we have an increase of around 58 to 77% in lysine, and similar is the case with the fact that our tryptophan has also increased by 38 to 100%. So now we have a more enriched lysine, where we have more enriched QPM, where we have higher lysine and tryptophan by almost 40 to 60%. But the next key question is, are we compromising with the softness? Because the success of the QPM lied with the fact that if the grain needs to be harder, no farmers, no industry will accept the soft grain. So therefore, the key issue was whether OPEC 16 is inducing softness in the OPEC2 background. And this is what the example of original 161, and these are the three sister lines where the grains are pretty similar to the original one. Here all these have both OPEC2 and OPEC 16. Similar is the case with another line of HKI 161, where the three pyramided lines were pretty similar to that. Once we broke the grain, and here also we found that the orange one shows the hardness, then the white one shows the softness. So OPEC2 and OPEC 16, the hardness and the softness ratio remained almost same. So that basically indicates that gene does not affect the hardness or the softness. Maybe it is increasing the free amino acid level in the form of lysine and tryptophan. So this is a great boon to the breeder, that just by introgressing this gene in the OPEC2 background, we can enhance the lysine and tryptophan level by almost 40 to 60%, which was not at all possible earlier, because then in that case we had to track other modifiers, and there is no example on that aspect of it. We have also developed in our program some of the OPEC 16 line, only OPEC 16 line. We developed in earlier slide, we have showed OPEC 2 plus OPEC 16. Now these are some of the lines which are highly diverse in terms of color, shape, grain, the 100 kernel weight. And here we see that the OPEC 16 only has got higher lysine and tryptophan as that of the wild type lines. So now these are the novel genetic resources that we have right now, and these can be easily used in any breeding program across the country. This is what the graph we want to show, that the in normal case we have the bar is here around for the sample, and this is what the QPM level is, and these are the ones where we have more OPEC 16 lines. So it is almost at par with only OPEC 16. OPEC 16 is many cases is at par with OPEC 2. The second aspect which we are dealing at IARI is the enrichment of provitamin A, and we know that we have a white maize and we have a yellow maize. White maize is because of the gene called small y1, which is in recessive condition, produces no carotenoids, and this y1 is nothing but phytoin synthase, which is the first committed step, and that is the gene has been taken from maize to make a golden rice in case of for the golden rice aspect. And then we have, whenever the dominant allele is there, yellow one, we have the yellow coloration, and it segregates in a typical 3:1 fashion when we make crosses. But the next aspect of it is, now within yellow we have a lot of variations. Some are red, some are orange, some are yellow, some are light yellow. Now these variations in quantitative genetic terms is mostly additive. It is governed by many genes as far as the earlier information is concerned. So the breeder has to work with many genes in a program. But one landmark discovery has happened, which showed that some of the germplasm from Simit Harvest Plus, they have 81% of the carotenoid, which is equal to 15 ppm, as compared to the normal lines which has got only 3% of beta carotene, and in terms of value it is only 1 to 2 ppm. So majority of the lines have got 1 to 2 ppm, while some of the special lines have bred by Simit Harvest Plus have contain 15 to 20 ppm. The question is why? The reason is, as mentioned earlier, there are two landmark papers came in 2008 by Hodges et al., who has, by candidate gene based association mapping, they could identify there is a gene called LCYE which converts the flux from this side to the this side, right side. So therefore we have higher beta carotene. So that gene was found to be one of the important element in the pathway. And there's another gene called CRTRB1 where Ian et al. 2010, Dr. Babu himself is the co-author of that paper when he was in Summit, Mexico, and that was also found that by analyzing an association mapping panel of 281 diverse lines. So the point of the matter here is they have done candidate gene-based association mapping. They have come down to a place where they could identify the gene-based polymorphism which is responsible for higher accumulation of beta carotene. So from association mapping to a targeted region which is causing the gene to enhance the beta carotene has been identified. Once this has been done, the next step is definitely to introgress that gene in your best germplasm, and that's what we have done. We have converted 4 hybrids we already released. 2 are in the early group, 2 are in the medium group, and they have their different adaptability, and this is what the figure is. The Vivek QPM9, it has got 2 ppm, our improved version has got 21 ppm, 27 has got almost again 2 ppm, here it is 17, for HM4 we got 12, and HM8 we got 21. And here I want to mention again, this Vivek Hybrid 9 was converted to Vivek QPM9 by introgression OPEC2 at VPKS by Dr. Babu, and we at IARI further introgressed another gene. It's a classical example of incremental breeding in which we are adding one gene after another in the same alike variety. So therefore we have a beta carotene enriched Vivek QPM9 which is also rich in lysine and tryptophan. And this Vivek QPM9 we code it as APQH9 which is rich in both lysine, tryptophan and provitamin A. We put it in the RACRIP trial, and Kharif 2014 and 15 we evaluated at the specific zone 1 and zone 4, and we found that yield potential, blue is the original, red is the beta carotene rich. Everywhere it is statistically at par with each other. That essentially indicates that our background selection for the phenotypic characteristics, yield and other potential remain same because of the stringent selection which we could put there. But the point of the matter is, in the third year, which should not be the case, now it is being analyzed for the quality attributes. System could not analyze the quality during the last 2 years. It was evaluated only for the yield. Now it is for only being tested for the quality, and if everything goes right, we can have some good news. After introgressing CRTRB1, then we also selected that LCY should also be in the same background along with CRTRB1. In earlier example, we only introgressed CRTRB1. In the next example, we are trying to introgress both LCY and CRTRB1 with the intention that the entire flux will be diverted to this side, and the CRTRB1, the gene functionality is it does not allow hydroxylation of beta carotene into further component. So therefore, we have more accumulation of beta carotene at the end of the day. So by introgressing, we have targeted already released QPM hybrids, and over which we have introgressed both LCY and CRTRB1. Yes, and here it is the value of beta carotene, original HK161 in blue, and these are the system lines. We have achieved almost around 20 ppm. Here almost we have achieved around 15 ppm. So everywhere there is a quantum jump in the level of beta carotene. But the next question is, what is the effect of both the genes or the single genes in combination? And it was found that whenever we have a CRTRB1 favorable, the increase in beta carotene is very, very high. It increases from 1 or 2 ppm to almost 12 to 14 ppm. But whenever we are adding favorable LCY along with CRT, so the increase is only 1 or 2 ppm. So therefore, in a breeding program which can afford to invest time, fund, can grow larger population, we would suggest to involve both the genes for beta carotene enrichment, and if it does not permit, maybe one gene is sufficient to at least to go to a higher level. And these are some of the phenotypic characters, the original line, these are the converted line, and these are some of the phenotypic features which is very similar to the original line. And that is what the kind of stringent selection which we are doing here. And as far as the release of the provitamin A rich maize hybrid is concerned, Zambia has released 3 hybrids, Nigeria has released 2 hybrids and 2 synthetics, Ghana has released 1 synthetic, and they have 6 to 8 ppm of provitamin A, while their original normal maize is having around 1 to 2 ppm, and in fact in white maize there is nothing. So this particular this one has the release of this variety has led to a study which says the study was conducted among school aged between 4 to 8 years in rural Zambia, and children who ate orange maize showed improved night vision within 6 months. So therefore it is not only the development, it has been disseminated and it has got the effect at the rural level who had the problem of visions. So therefore from the development to the delivery, these kind of products are already available in the system. The next nutrition which we are targeting is vitamin E at the tocopherol. We know that maize is very rich in tocopherol, as same as that of soybean. But what happens is we have, out of total tocopherol, 80% is gamma tocopherol, whose vitamin E activity is much less than that of the alpha tocopherol. Alpha tocopherol constitutes only around 10%. So the geneticists were trying to improve the flux, to improve this catalytic step, so that we have more of the gamma tocopherol is converted into alpha, because by putting a more efficient gene in the pathway. So for that matter, Lee et al. In 2012 went for the genome-wide association mapping. The earlier vitamin A example was candidate gene-based association mapping, which has led to the development of the variety. Here they have done the genome-wide association mapping, and by which they have come down to this gene VT4. VT4 nothing but this gene which is converting gamma tocopherol more into alpha tocopherol. So this gene it was found that there are 2 indels, indel 7 and indel 118, which is causing the increase of alpha tocopherol from 7 to 8 ppm to almost 22 to 33 ppm, almost 3-fold increase, just because of the presence and absence of this indel. We screened our germplasm, large germplasm, 453 inbreds were screened, and we found there are only 4 inbreds which has got this favorable allele. So we now have the donor, and then we are using this donor in the marker selection program, where we are already improving the QPM plus provitamin A rich maize hybrid along with the VT4, that is the tocopherol. So therefore we are targeting to have the hybrids which is having not only higher lysine and tryptophan, higher provitamin A and higher tocopherol. We have also developed a sweet corn hybrid. It is in the 3rd year of trial. We called it as ASKH1, and it was found to be very promising in the farmers field. We know Aterna village is one of the very attractive place as far as the corn breeders are concerned. So we are also improving such kind of sweet corn hybrids with the nutritional quality. So with SH2 gene that is imparting sweetness, we are having CRT and VT4 and different combinations. So these products are in the pipeline. Now we have shown 2 maps that where it showed the severity of iron and zinc. Now in normal maize inbred, 25 ppm is the baseline. That is, normally we have 25 ppm, but our target is around 38 ppm. The Harvest Plus target, of course it is based on the consumption level, and it is a target for the Africa. It doesn't hold true for India, but nevertheless that can be achievable genetically. That's why the target was put. For iron, normally it is 30 ppm, but now the target is 60 ppm, almost double. But a lot of QTL mapping and genetic analysis have taken place, and it was found that it has governed by many QTLs, and they have a very minor effects, only 2 to 7% R square value, and many of the QTLs are background specific, so there is no universal QTLs. And on the other hand, the variation is not very high. To have a maize line consistently 60 ppm is a difficult job. And not only that, it is also having the problem of high G into E. Even though we have a higher zinc and iron, it is many times it is not consistent over here and over location. So it needs a separate approach. Maybe genomic selection and other tools can help to bring all small loci are having small effect to bring in one place, and maybe that could be a good approach. We are following a second approach, which is an indirect approach, to introgress LPA mutants. We know maize kernels contain 80 to 90% of total phosphorus is a phytic acid, and phytic acid is very much required for the germinating seeds. But the negative charge of phytic acid, it chelates iron and zinc, and therefore in the human and the animal gut, it does not allow the iron and zinc to be absorbed. So bioavailability is drastically reduced. Now we have the well characterized pathway in which phytic acid is formed. 3 naturally mutant, in fact induced mutants have been found, LPA1, LPA2 and LPA3, and they can reduce the phytic acid. LPA1 can reduce the phytic acid by 66%. LPA2 and 3 can reduce phytic acid by 50%. So we have a lesser reduction. So lesser reduction means lesser chelating and therefore higher bioavailability. And with this, this is what we are trying to show, that in direct approach, 30 ppm is the baseline. To go to 60 ppm, which is the target, will take a lot of efforts, different breeding tools, techniques, it will take more time. But even though 60 ppm is formed, we have only 5% of the bioavailability. That means whatever we consume, only 5% will be absorbed in our body. So therefore, we are essentially getting only 3 ppm. And based on 3, only 60 ppm target has been formed. But if we manipulate with low phytic acid, 30 ppm in maize line, if we induce LPA1 and 2, whose phytic acid is reduced by 50 to 60%, so there also we can get almost with, and that means the bioavailability increases by double, that is 10%, and then we are getting the same 3 ppm. The advantage here is we have only 1 or 2 genes, it's a recessive genes, we have the markers, it is easy to manipulate. So therefore we are converting some of our elite promising hybrids in the different maturity groups in which the different LP mutants are being introgressed. But this particular introgression is also not devoid of challenges. The moment the phytic acid is reduced, plant gets susceptible to different pathogen, abiotic stresses and all. But the fact of the matter is there is definitely variations, and there is a scope to select a line which is having low phytic acid but having better agronomic performance. So with that intention, we are going ahead with this program. In a nutshell, we are working to develop hybrids, not only QPM, then QPM with high amino acid with both the genes. We are working with provitamin A with QPM, again with 2 options, CRT and CRT-LCY. We have provitamin A QPM plus vitamin E, for all 4 genes in one place. We are also improving sweet corn hybrids with different genes for provitamin A, vitamin E and QPM, and of course we are targeting QPM hybrids for iron and zinc. And all these hybrids are elite hybrids, and they are in different maturity groups. We are not targeting any specific maturity group. We have early, we have medium, we have late, and all these, many of them are ready. It is in the ECRIP trial, and many of them are in the final stages of the in the pipeline, and which we are going to push it one by one whenever, based on seed production and other scenarios. Now, this is the challenges for any biofortified crop. Probably as far as the genetics is concerned, genes are available, tools are available. It's only the effort we need to put in to enrich maize with micronutrients. And the same effort can, is being done in other crops like rice, wheat, pearl millet, and and lentil, and many other crops. But just I want to say a few things which comes always in our mind: apprehension of low yield with nutritional quality. The moment we say it is higher in quality, it comes to our mind it is having lower yield. Same is the case with QPM. QPM is in the place for last almost from 1980 to 2016. The land, the area under QPM is now very, very minimal. One of the reasons that farmers, they say they get the less grain yield. The less grain yield is not because of the OPEC2 gene, it is because of the lesser diversification of the germplasm. Lesser effort have put in to improve the lines per se performance. We have categorically shown by normal hybrids and its EDV version of OPEC2 that there is no yield penalty per se because of the OPEC2 gene. The less yield potential is because of the genetic improvement the line is supposed to go through in last 20 years have not gone through because the efforts have been minimal. So therefore, all this quality which I have mentioned is nothing to do with yield penalty per se. Then second is, many times once we develop, go to the mandi to sell the grains, if we say it is high in provitamin A, we don't have any handheld meters which can say, okay, this is the level of provitamin A. Fortunately, we have a bricks meter for sweet corn, we have a bricks meter for sugarcane, where they go to the mandi, they immediately check, okay, this is the bricks, this is the value, let us take it, or if it is less, let us reject it. For this case, I think we need to develop some small handheld machine which can rapidly give some kind of an idea whether it is high, low and medium, I mean whether it is lysine, tryptophan, provitamin A or tocopherol or any other thing. Second is the dilution of nutritional quality due to xenia effects, because all these genes are recessive genes. The moment it is grown in the field, there is a possibility if the other farmers are not growing these nutritional quality hybrids, so the dominant gene will come and at least will contaminate the bordering areas and the quality will be reduced. So therefore, we need to take effort to convince farmers to go for a village mode where all the farmers in a village grow a typical type of quality of maize. Same kind of efforts are being replicated for baby corn and sweet corn in Eterna village and another village nearby on and around Delhi. Second is there is always a resistance in accepting altered phenotype. It happens in Africa. They always like white maize. The moment the Simit people and their country people went with an orange maize, they initially did not accept. They had to go through lot of extension surveys, lot of field demonstration, lot of convincing effort. They had to talk to village leaders. It went through a different aspect. Then only it has become a successful. So therefore, at one go, if the phenotype is altered, many times, even though we say it is nutritionally good, at least the farmers and many of the village consumers, they have the inhibition. Should we go ahead with the cultivation of this hybrid or not? So that is an issue. Then lack of awareness on nutritional benefit. We had a brainstorming session in last year. We found many of the poultry growers we have found that 60% of the maize goes to poultry. Many of the small poultry growers, they are not aware of QPM, although they are adding synthetic lysine, but they don't know what is QPM. So therefore, there is a need for making people aware of the nutritional quality, then things start happening. We also need to have the organized food processing industry. We had the people from Kellogg's and other companies, other food processing, maybe porridge and others. If they can be convinced, if they can be taken into consideration what is their criteria, what they want, accordingly these kind of breeding efforts can lead to a better nutritious food which can be easily marketed. And definitely we need a policy support without which we cannot do anything. We maybe a premium price, maybe an integration of nutritious crop in integrated child development program, midday meal program, in other aspects. The strong policy support will definitely help in disseminating this biofortified maize. Now, this is a very important message from which we found in IFPRI 2016, which says that the prevention of malnutrition by any means, whether it's biofortified crop or anywhere, it delivers $16 in return on investment for every $1 spent. So that is what the impact we have if we spend on nutrition, whatever is the criteria. Biofortified crop is just only one option. So that is what the impact of nutrition has, that if we can improve, that's what we can have the gain. And therefore, why not biofortified crops be popularized? If this is what the data, then why not? Now I stop here. I would like to thank my council, Indian Council of Agriculture Research, Director IARI. Prabhu sir is here, who has been the Joint Director Research and has been the head under which we initiated the program. Tremendous support, complete free hand, do this but accountable, deliver it. That is what the support we are getting it from him. We are equally thankful to Dr. A.K. Singh, who is the head division of genetics. In fact, whatever we are doing, providing tremendous support every time, suggestions, and that's how we are going ahead. We are thankful to Dr. Gupta, who is the coordinator for DBT program, under which many of the varieties have been developed. So we are thankful to him. We are thankful to IMR Winter Nursery, where rapid generation advance we take one season here, another season in the winter season at Hyderabad. We are thankful to Almora Uchani centers, their hybrids are being converted. Thankful to Simit Harvest Plus. Dr. Babu, when he was in Simit, he helped us tremendously. He provided the germplasm donors immediately whenever we asked. So thank you, sir. And thanks to ICR CRP for the funding. Thanks to DBT and DST for their generous funding in different projects. I sincerely thank the entire maize team. Dr. Vignesh, he has been the person who has developed the provitamin A version in his PhD program. Dr. Rajkumar is now our colleague. He has developed both LCY CRT. And then we have other members of the team who are the part of the entire maize team. Thanks to technical staff, students Sarika, who worked on OPEC 16, Rajkumar is a student, worked on OPEC 2, CRT and LCY. Then Abhijit is working on VT4. We are also thankful to the SRFs who have been the bench strength to our program. Achal, then Abhijit, and all these people. So thank to all of them. So thank you very much. So maybe.
Thank you, sir. Thank you for enlightening us about how we can use the genomics for improving our nutritional quality of the tropical corn. The session is open for discussion.
Yeah, sure, sir. Can anybody give a mic to RMC, sir?
At the outset, I compliment you for very nice presentation.
Thank you.
On enriching the nutritional quality of maize. As you said that there are a number of challenges, number of regions for not being acceptable as yet, and you also said the area is very, I am really concerned about it that it has been a long period when Vivek 9 was developed with all the good qualities and so forth. When we look at the scenario country-wise, it has not really made any impact as such. Now, other thing you said was that the higher yield in case of the QPM as compared to the non-QPM may be another factor because of narrow genetic base present in the QPM material, because we had very limited QPM material obtained, and from that the entire program has progressed. So what are your plans for really improving this? I mean, you know that it's narrow genetic base, so what are your, because I didn't see any presentation, your future line of work. Suppose if you want to take some work to improve the gains, yield gains of the QPM, what would you like to do in this particular case?
Yeah. Actually, I deliberately did not show the slides of the things which we are doing in the back. It's not only the markers selection program which we are carrying out in the program. We are also enriching our germplasm so that we have a better lines, new lines. Professor Gupta has said, why to improve our existing line? Why can't we have a new lines with a better trait? We are basically having the same scenario like any maize breeder will do. We have some information of their heterotic potential, and therefore we are trying to improve within groups by pedigree selection the yield performance of this line, so that we do not mix up the heterotic potential of the two groups, and by pedigree selection and even by introgressing new lines we are improving the yield potential of it. That is number one. And second thing is, even a new line which is developed through non-conventional breeding program, that is being immediately entered into the marker selection breeding program, because that has already achieved the enhanced yield part of it. So we are following basically these two approach to have our lines enhanced for the yield potential. Yes, sir.
You see, one problem that you face in the case of quality improvement is the economic aspects.
Right.
The farmers will not grow a very high quality grain crop unless it pays, and it doesn't pay. You know it. You have already said it also.
Right.
In Mandi, if you go and tell that this has wheat variety with 15% protein, he doesn't care for it. And the farmers also do not gain out of it. So why should he grow it? Unless you combine it with high yield also.
That's what exactly I said. The hybrids which we have converted, it is actually in the medium maturity group hybrids we have converted, that is having 6 to 8 tons per hectare. Now, the private sectors, and normally they are basically concentrated in the late maturity group where the yield potential is around 10 and 10 to 12 depending upon season and locations. So the thing which we got is that since OPEC 2 per se has no yield disadvantage, why can't it be put in the best of the varieties whosoever develops? And even though it doesn't give premium, what is the problem of disseminating such kind of maize?
No,
because
you have not understood. No, I get your point.
Yeah.
There is no harm in developing varieties with high quality. But the point is, why should a farmer go for it?
Yeah, exactly.
Unless there is some advantage somewhere, and the best advantage is yield from farmer's point of view. If you can carry more quantity of grain, whether it combines with the ordinary levels of zinc and iron or whatever it is, then, or it is high, he is not going to get a premium on it. So if the farmer has to be attracted, he should adopt it, because we cannot force farmer to cultivate any particular variety, even if it is very rich in micronutrients.
Yeah.
That is our problem.
Yeah,
and
exactly.
That is a general problem everywhere. You mentioned about bricks, and you said if it can be done with bricks, why it cannot be done for zinc and iron? The question is that bricks sugar production is affected, and therefore the factory is prepared to pay more for it. But for high protein wheat or for high lysine maize, nobody will pay for it.
No,
that is the crux of the problem.
No, that's what I am trying to tell, that suppose option number 1, farmer is convinced he is going to the mandi to sell this high biofortified maize. The mandi people who is going to buy these large quantities, they need to be ensured that it is rich in quality. Because any farmer can go with a normal maize, maybe mixed up maize, unknowingly, knowingly, there should be some kind of small, gadgets which can
That's
directly
all right.
tell.
I mean, you are making suggestions without considering their practicability. You do not know how the farmers' produce is sold in the mandis. You know, there are different heaps of materials.
Right.
You are a farmer, I am a farmer, he is a farmer, and we will have 4 different heads. Then there will be brokers, dalal, as in Hindi we call them. They will come, just a minute, they will come and fix a price. They will give a better price if there is a greater shine, if the grain is bolder, then they might offer a better price. But if somebody says it's a high protein or high lysine, nobody will bother about it. Yeah, please listen to me. And ultimately what will happen? That all 4 of us, all our produce will be pooled. And that some of them may be having high lysine, some of them may be having low, some of them may be having just average. So ultimately the fact is actually averaged out.
Yeah.
So these are the practical problems, and there has to be a system. Government cannot create a system. We do not have a kind of political system where you can force the farmers to cultivate a particular type of variety. That's not possible. It's only if the farmer gets an incentive in some way, either by yield, high yield combined with high nutrition quality, then it will definitely work.
Yeah, so that's what exactly I said, that this quality attributes has got no yield penalty, whatever trait we mentioned. So therefore, there we can develop high yielding maize varieties along with the quality. That is number 1. Number 2, whether farmer is to be convinced or not, that we need different programs that I have mentioned. We need a strong policy support, premium price, everything I have mentioned. A breeder cannot probably look into those aspects. We at one side said we have a problem of micronutrient. At one side we say there is a health problem. At one side we say we have the option to alleviate that. The third option is to implement that. There are different aspects involved in that. Policy matters definitely is one of them, and I personally believe without policy support, without the support of all these events which probably I mentioned, maybe I missed 1 or 2, why can't we achieve that? It has been achieved in other countries. Maybe scenario is different, but the option is definitely available. And another key point is, I personally believe if we don't go to the policymakers with a ready-made material, they are no longer considered. They say you first develop, then come, then we'll see. Now here we can say we have a ready-made material which has the potential. Now you see for the country to do that. At least we can do that, at least for the breeder side.
No, I think Professor Balram Sharma's point is extremely valid. There is no two opinion about it. Unless the product has commercial significance, it's not going to move. And that's very, very valid point, well taken. But what I'm saying is, it's also important for us to keep assessing what is the commercial requirement, right? The initial excitement was for OPEC 2. Yes, it led to doubling of lysine and tryptophan. But then when we went to a stakeholders meeting with all the poultry industry guys, I think the one thing that we learned from them was that the most expensive amino acid for them to supplement was methionine, neither lysine nor tryptophan. They can be cheaply supplemented, whereas it was methionine whose the synthetic supplement of methionine was very expensive. So they were asking us, can you develop a material which is rich in methionine? Then I will take it, right? So I think it's very important for us to keep tracking that requirement, be it the poultry industry, I mean, or any feed industry, or the farm. I mean, farmers, biofortification is a very sensitive topic, right? I think we cannot totally give this to the farmer as well. So he is not going to be completely understanding the significance of this product. So somewhere we need to create that awareness. At the same time, an excellent success in terms of producing a product, it also has to result eventually in adoption in the farmer's field. So I think we need to understand that critical gap, and from that point I think Professor Balram Sharma's point is very, very valid, and I appreciate that. Yeah.
There are questions from students, no, maybe not the students, faculty from Andhra Pradesh University. What is the effect of biofortified crops on pest scenario? Will it lead to usage of more agrochemicals to control the pests, because they may be attracted to the added protein?
Well, that is a possibility, but there is no scientific study available. Dr. Babu, you can supplement that whether any biofortified crop is becoming more susceptible to insect pests. I personally feel there is no relation to that, and that is a random event. If at all it happens, then
no, I think until now at least we have not seen any negative effect of biofortification.
Okay, next there is another question. What can be the genomic tools available to break the linkage between softness and QPM?
Well, this has been one of the challenging area in QPM, because whenever the OPEC 2 goes, we need to select for the modifiers, which essentially makes the soft kernel into the hard endosperm. But there is no question of breakage, breaking the linkage, because it's the pleiotropic effect of OPEC 2 and high lysine tryptophan, on the other hand the softness of it. So we can never break the linkage, but we can improve the scenario by introducing the modifiers, which has essentially been done by Dr. Vassal and his team. So now we have very good genetic background like HKI 161, CML 161, 163. They are very good modifiers. The moment we bring modifiers from them, our grains are almost as similar to that of original maize. And another is OPEC 16. We have shown many OPEC 16 lines are as good as OPEC 2 lines, but they don't have any effect on kernel hardness. So those mutants definitely can be used for that purpose.
Okay, microRNA and genome editing can come very handy here.
Yeah,
unless same amino acid sequence is involved in both the cases.
Right.
If there's any diversion of any of the amino acids, then you can use any of this. Yeah.
Right. Now I have one. Want to know from the private sectors. These are the questions regularly asked. What happens to biofortified maize or any crops whatsoever? Why it is not going to the people and all that. I think we all are convinced these quality attributes is nothing to do with the yield per se. So therefore, there is no linkage between quality and yield. We need to only bring the high yielding genotypes with qualities. Now, private sectors are working basically in the late maturity groups, and they are releasing hybrids which has got other attributes like stiff stalks and other things. What is the problem? If any germplasm being or any hybrids being marketed by the private companies, why can't they have any of these quality attributes as a baseline? Everywhere they develop, they should have these characteristics, and therefore, whether we get a premium price or not, at the end of the day, whosoever consumes will get the higher nutrition. The point of the matter is what actually is the problem, why it is not being, because we are not only selecting genes for one or two traits, we are also selecting genomic selection, so many genes. Selection of gene is not a problem. This is only the integration until there is a negative effect of it. So can you just tell the audience, including?
No, I think it's a good question. I have also been thinking on the same lines, but then, in the private sector, I feel any additional thing is an additional investment, and anything that happens on an industrial scale. So it's not easy to introduce a donor carrying provitamin A or quality protein maize, and then keep it maintained in your breeding germplasm is an extremely resource demanding task. That's why Harvest Plus came in. Not that all the usual regular breeding programs are going to tackle this kind of traits, and that's why they exclusively formed Harvest Plus. And because of that needs that kind of dedication, focus. So in the private sector, that decision has to be taken. That is actually a very, very resource demanding exercise, and unless there is an assured market, or unless I am going to add clearly commercial value to that product, it's going to be extremely difficult to take that step. But I would request Anand to add further.
Yeah, so Babu, just thanks. So I'll provide a slightly different perspective on this, and one thing that people are looking at it, especially in most places where corn is grown, is any of these, be it starch, protein, oil or sugar, as yield in terms of area. So I may have a variety that has any higher protein within itself, but on a per unit area my protein recovery has to be at a level which is acceptable. The second thing, most of the users, especially for corn, is industry. So for example, Cargill and other companies which are doing it, they have a process of separating or partitioning corn into different components, where their starch, oil, everything is extracted independently, and the optimization of the grain in terms of its use is maximized. And so in that whole context, when adding a certain component decreases or the value on the other, is something that they're looking at it. So from a end user industry perspective, which is 80% in the case of ADT and Cargill, you showed global production, 80% of this is with big producers like this, that are end use producers. That is where that is who drives what the product is needed. And so that's where some of this demand for what kind of grain is needed, what kind of quality is come. So it addresses these end users, the major industry, then it will definitely be disseminated.
Yeah, exactly. We need, at one side we have a micronutrient deficiency, another side this scenario you have said, maybe we need to look into that.
Just to add, Suguna, the feed industry guys, so they were very much interested in going for the QPM varieties recently. When we were engaging them in discussions, they said that we can assure certain 2% or 3% premium price for that kind of product. So if that kind of demand comes in, then perhaps it makes sense.
That's why awareness and all this.
Right.
Thank you for
There is one announcement. This is regards to the students. We have about 25 participants coming from outside the IARI from various state agriculture universities. In the valedictory session, one of you may have to come and give the feedback, and also one student from IARI. So I request whoever will be giving the feedback, please give your names in the reception section. Okay? Thank you.
Thank you, ma'am. With this, we are completed with our third technical session, and let's get started with our next technical session, session 4, which is about interactive workshop with a student on commercial plant breeding. I now take this opportunity to call upon the stage our first speaker of the session, Dr. R.S. Mahala. Dr. R.S. Mahala, a research director for millet and breeding operation at DuPont Pioneer. Dr. Mahala has developed a number of industry-leading pearl millet hybrids that are very popular among the farmers throughout India. Dr. Mahala specializes in commercial plant breeding, wide area testing, farm mechanization and seed production. And today he is going to tell us about how is the success defined in the commercial plant breeding. So please welcome Dr. R.S. Mahala. Please.
Distinguished professors and our dear teachers and dear students, good afternoon to you. I'm the last one, I think, in this series and I'm going to speak more about commercial plant breeding. And I feel very fortunate to be here, and this is my favorite subject, talking about commercial breeding. I started my career as a commercial plant breeder about 31 years back in Pioneer after graduating from Pantnagar, and I have worked as a millet breeder for almost more than 20 years, and after that I started guiding my colleagues about how to perform commercial plant breeding actually in the organization. So I think you heard a lot about since yesterday till this forenoon about the latest tools and technologies, and what I am going to cover is from the commercial perspective side, how the plant breeding is practiced in a commercial breeding organization, and how to use these tools and what is required and all that. So if we talk about commercial plant breeding, in the topic itself it says three things, plant breeding, and then another word used here is a commercial, which means business, and the third factor here is success. So unless it is successful, it's not considered as a viable business. Three topics we'll talk, and I have tried to put this in a very simple form in these 5 broad topics. What is commercial plant breeding all about? And impact of commercial plant breeding, that what impact it has created in all these years, and how the commercial plant breeding is evolving going forward, and then what it takes to become a commercial plant breeder, especially for the students. It's very, very important to just get some snapshot about this, what it takes to become a commercial plant breeder, since you will be starting your career. And then some of the key learnings from some of the senior plant breeders in a private sector, what they talk about the commercial plant breeding, their experience. So I think it's going to be very interesting. Please, and it's not a high-tech thing, it's a very simple presentation. I have tried to compile everything in my own style, and sit back, relax, and I think it's lunchtime, you must be feeling hungry, but have some more patience, and we are just going to finish this in maybe 30 to 40 minutes time. I think it's very important before we talk about plant breeding and commercial plant breeding and everything. Mendel. So Mendel started, opened the door in the field of the biology. And whether plant breeding is the basis for plant breeding is genetics, and basis for genetics is the Mendel's principles. And since then, a lot is being done, and I thought I should put this slide, my first slide, when I talk about the commercial plant breeding. And Mendel discovered, the 2 principles here, the principle of segregation and independent assortment. And the work was published in sometime 1854, and but later it was rediscovered in 1902. 7 monogenic traits, and that was the beginning of the things. And today, it is totally, the changed world. So let us talk about plant breeding. I think this is a standard one, and all of you know. But since I'm talking the subject, I thought just let me include this as an introductory slide. And when we talk about plant breeding, what is plant breeding? It's basically called as art and science. So art and science, for changing the genetics of the plants in order to produce desired characters. Desired characters vary depending on the need. Art and science, but these days the plant breeding is now evolving much faster, and it's becoming less art and more science, because a lot of science is used to practice plant breeding. And if you look at how it evolved over time, it has taken many years, right from starting from domestication, and then some of the selection practices, a lot of those things happened in the early days, and then it moved to the controlled mating. The controlled mating here, several breeding methods were developed, like population breeding and pedigree breeding and hybrid breeding. Am I audible to you? Okay. So, and then after that we moved on to monitored recombination, where the markers and selections on what you heard is being practiced now, genome selection. And some of the crops like corn and canola and all that, some of the students asked the question whether markers are being used in creating successful products. So double haploidy and markers and selection, pedigree selections, all these things are being used routinely nowadays, like a factory. So it's not that it's not being used, it's being used already, and it's very successful. So in nutshell, if you talk about plant breeding, what we say is plant breeding can be accomplished through the many different techniques, okay, ranging from very simple techniques of selection and crossing to the molecular tools. This is how the plant breeding is. So let us talk about what is commercial part in this. So why we call it commercial. So somebody has defined it, like Bernardo in 2000 says that he added one more word in this. It is science, art and a business of improving plants for human benefit. The plant breeding has become business now. And whether it is not only plant breeding, breeding has become a business like in animals or fishery or poultry or anything. So that's how it is. And if you understand it fully in another way, is a commercial plant breeding is a business of science of crop improvement. And it's the basis of sustainability and it is a foundation of our global food security. That's how the plant breeding is now in the current age. So from a plant breeder's perspective, how do we, exactly understand the commercial plant breeding or successful plant breeder or successful plant breeding? We generally say that, okay, we say that if we make any genetic gain, that means we are successful, we are making progress. And this is the favorite equation of a plant breeder when he talks about what he does. He does most of the time is selection, crossing, evaluation. And this cycle goes on year on year, and then you keep making progress. So the genetic gain is one of the way to monitor the progress, and how to, there are some of the components that we need to understand in the commercial plant breeding, in genetic gain, where you see there are three things here. One is the genetic gain is understood like genetic gain over time, and the key parameters in these components are selection intensity, selection accuracy, genetic variance, and number of years per cycle. This process, these are the four components. We will talk about more in detail about these things, how these things are practiced in a private sector kind of thing or commercial plant breeding, because these are very, very important key elements to manage the genetic gain. Genetic gain is not the only the thing in a commercial plant breeding. There is a much more beyond that, and that's what is all about commercial plant breeding. Now, if you look at here, I just try to put a simple graph here that in any organization or any public sector institute anywhere, when we run the breeding programs, we look at genetic gain, we make progress year on year in a particular crop. And this all depends on the internal, competency of the organizations. How you make this graph, whether more steeper, by using all tools and techniques. So different tools and techniques are used depending on the capacity, competency of the organization. In terms of to increase the selection intensity, some matrices are used, some tools are used, selection accuracy, some tools are used, and then genetic variance, creating for the genetic variance tools are used, and then years per cycle, reducing the invert development cycle, the tools are used, DH kind of things. So there are various tools which are used to increase the genetic gain and make it much more steeper. So what success means in a commercial plant breeding organization. So I'm coming now to the commercial plant breeding organization, what success means. And here, from a breeder's perspective, when you ask a breeder about the objectives, he will talk about mainly these four categories. Generally we say, I'm working on these breeding objectives. And mainly these are the yield, could be the grain, fiber, fodder, and then it could be in terms of quality, maybe milling, cooking and keeping, and then agronomics could be maturity, growth habits, dormancy, photosensitive. It all depends, and stresses may be salt and temperature, standability, many other things. Broadly, we can classify this in a four category. Breeding objective, anybody talks about the four breeding objective, and then under these categories. But in a commercial plant breeding, we need to look beyond again. It doesn't, end here. What is next? Next is, are these breeding objectives addressing these concerns of these three stakeholders? Unless these three stakeholders are not addressed, whatever we do, it's not successful, particularly in a commercial breeding organization. So there's a good interaction happens between the organization and a consumer, what he wants, and based on their breeding programs are tailored. And here the organization looks for profit and sustainable growth. Also the grower who buys the seed from the organization, he also looks that his crop productivity goes up, his cost of production goes down. So the products have to, fit into that kind of criteria. And unless that doesn't happen, a grower, maybe it's a seed grower, unless his seed yield is not much, he may not take up your production. So you have to compete in the marketplace, and you have to address these, these issues, the needs of these three stakeholders. Now, there is something beyond product again I'm talking about. You may develop a product, it may take 10 years for you. Is it sustainable? No. In a commercial breeding organization, it may not be sustainable, because marketing people, they need products every year. They are competing, a lot of other competitors in the marketplace. So they need product every year, year on year. And a breeder has to deliver those products. Now, the critical factor here is not only the product, it is beyond product. What are those? It has to be developed faster, because they want to be first in the market. It has to be cheaper for the grower and buyer. Otherwise, product will not be successful. Then superior, of course, genetic gain or superiority, whatever we talk about, then has to be addressed. So this is what is the scenario in a commercial breeding organization, where the product development also has to look into these things also as a breeder, that these issues are addressed. Now, success depends only here on how best these needs are fulfilled. So what are the tools and techniques you use to be faster, cheaper, and superior in the product perspective? What impact it has created over the years. So I have just put some of the slides here just to see how the plant breeding has created impact. This is an example of corn. Corn example is quite popular. So if you look at historically, single cross, double cross corn, and then the single cross corn here, the bushels per acre, the progress has been 1.8 or than 2, which is very steep. So most of this is it's a US example, a great success story about how the commercial plant breeding changes the lives of the people and creates impact and helps in producing more food. The another one is India specific. Here the maize yield, if you look at here, 31 and then 63. This is double cross and then move to single cross. That's how the progress is so rapid. A great success story. One more here, look at here. In case of rice, now moving from varieties to hybrids, and as we move more and more towards the hybrids, the progress is going to become more steeper. There's one more example I have put here is pollen millet. See here. Actually, the seed industry and the commercialization activity started sometime in late 90s, late 80s, 89 or sometime, 87, 88 or sometime, where the more private sector industry got into the business, and that's how they started getting more hybridization, and then a lot of progress happened all these years. So up to here it was a down yield impact, and after that, a lot of progress has been made in case of pollen millet also, increasing the productivity. This is the case of cotton, where the trait was introduced BG1 and BG2 somewhere this time, but still after that also nothing new happened. BG1, BG2 were there, but despite of that graph is going up. That means people say that there was not much of genetic gain, but actually genetic gain has happened in cotton as well. The graph shows that technology introduced that in 10 years, despite of the technology part remaining same, still genetic gain happened. So if you look at overall scenario here, this is a snapshot of the share of the private sector, though it's an old slide, but share of the private sector in this case is something like 80s and 86 and 80s, and then in this graph 91, which is really indicating that good impact has been created by using successfully the commercial plant breeding in the private sector also. How commercial plant breeding is evolving now, if you look at, there are three things which are very critical in this to look at. The breeding is now being practiced in a much more sophisticated way, and then also the germplasm, how it is being utilized, and then how the farming systems are changing in the whole scenario. So let us look at this. Yesterday also we saw this funnel. Lot of tools and technologies are used to create, as much more variability in the funnel, and then reduce the time to ensure that product delivery, in-bed development is faster. And for that, all the tools and technology, whether it is SNP markers or chromosome information and phenotyping information, all that is being used. and breeding is not being practiced now the way it was being practiced in the old days. So, and look at the germplasm now. There are most of the big organizations are using germplasm, exchanging germplasm very aggressively, and they are tracking and then classifying and utilizing in a very, very professional manner and very strategically to develop the best product. Also, things are changing. Farming practices are also changing in developed countries and as well in India also. If you look here, the cotton, the kind of cotton we used to grow now, in going forward in the future, the different kind of machine harvestable things are going to be in marketplace. Same with the sorghum, and same way example like high density. So a commercial breeder, the message for the students here is that a commercial breeder has to think always 10 years ahead. What is going to come in the market? Accordingly, he has to develop breeding program and plan things, and based on that he has to put the product development strategies in place. Otherwise, if you keep working like this kind of cotton, suppose, and one company comes in the market with a machine harvestable, overnight your company will collapse and your program will also collapse. Nobody is going to buy your products. So this is the something which always you have to keep your eyes open in the commercial breeding. Now, it's becoming more complex and multidisciplinary. How? Three things again. What the every time, three focus area is the time, cost, and precision. How to increase the precision and get the more success rate, and increase the success rate and get the best product. How to reduce the cost of product development cost and running the organizations more efficiently. How to develop the products much faster, reduce the time. These are three focus areas again, where in a commercial plant breeding, the focus has to be. Now, what tools and technologies should be used for that? So this already has been covered. I'm not going to talk much about, but there are numerous things. A lot of tools are being explored and used. Some are developing, many are already used, and depending on the situations and depending on the priorities. Not necessarily that in commercial breeding, in each and every crop, all these tools will be routinely used. Maybe next 15 years also, some of these tools might not be available to smaller crops even. It all depends on the cost of the using tools and efficiency and other things. But yes, large crops, maybe some sophisticated tools will be used. And see here, like here in vitro techniques and molecular breeding, mass, GBS and CRISPR-Cas is coming now in future. Phenotyping tools where drones and other things are being used to reduce the cost and increase the precision. There may be environment modeling, simulation and all that. These are the kind of things which are going to be used more and more and routinely to increase the precision and bring the efficiency and also reduce the time. And DH is already a successful story and example in canola and corn, and routinely it's being used. So what success means here? Success means here is the ability to transfer the genetic gain into commercial products in the most efficient way. That's what is called success here. And as I showed this graph again, depends on the how steeper you make it or how fast you make it. It all depends on the internal competency and efficiency of the organization. And what tools they use, apart from the biotech and other support tools, the breeding programs in a commercial organizations are run very efficiently in a very professional manner. There is a fixed amount available for certain things based on the objectives and priorities of the organization. And then the breeder has to plan everything to get certain success number of a hybrid every year. Like I want one hybrid to be released every year. How many hybrids I should make in top cross stage, top cross 2 stage, initial hybrid trial 1, 2, advanced hybrid trial, and finally release 1. So when I start my program, I need to know precisely how many hybrids I should make right from the beginning. I should know how many testers I should use, how many locations I should use, and how many plots, what should be my selection intensity. Ultimately, the success program, it will be evaluated based on the cost per inbred, how much money you invested developing successful inbred, how much money you invested developing successful hybrid, because every rupee counts. So every rupee will be, it will be spent, will be evaluated whether the business is profitable or not, and how much we are spending and how much we are earning. Then apart from, and this is used to achieve, this whole funnel thing, the inbred development process. So this is about a little bit about the future outlook of plant breeding industry, how it's going to look like in future. Key things here, global seed markets have grown, the billions of industry now being invested in this. And it's now being integrated, the technology. A lot of new technologies are coming every day and they are being now integrated. Apart from that, the changes are happening very fast and these changes are driven largely by the technology. So because of that, it's very rapid. So the young breeders have to keep themselves updated all the time. The new technology and tools are coming. Intellectual property rights are becoming now everywhere very important, and the plant breeding industry now is investing a lot and trying to develop the best materials and protect the germplasm and all that. Private sector is investing heavily in these things. Genomic maps will be available in future, already available for many crops and important crops, and many more will be available, more refined maps in fact. And then functional genomics is going to be used routinely, and genome editing, which is upcoming technology now, might even make many other things obsolete very fast. So that thing also is getting now into the mainstream, and maybe in 10 years the things will change very dramatically. And then a lot of software tools are available nowadays, statistical tools and everything is being used. The breeding by design, prediction, and then how to predict that what material should be, germplasm should be crossed with what so that you get the best inbred, what cross should be made, what hybrid should be made, what cross should be made so that you get the best hybrid. So these are called the prediction tools. So softwares are available, they analyze the historical data, and based on that you decide, okay, this is the material that you should cross to get the maximum probability of the success. And the life cycle of products will be shorter because industry is looking for every time to offer the better and better product much faster to the customers. So and then what it takes to become a commercial plant builder, I just put it in my own way. What I say is, like, commercial plant building is like a journey from simulator to real time flying. I see the way you learn, like on a simulator initially, you practice alone, practice it how to fly, then you fly with the experienced pilot. And after the experienced pilot, you become a professional and hardcore fighter and then win the game. Okay, the bidding is also something like that. You hardcore commercial plant builder, if you want to become, first you do some studies through your thesis research, or maybe initial days you learn a little bit about the fundamentals and basics. Then you go and learn with the experienced plant builder, and you are done. Then you are ready to work as an independent, as a hardcore commercial plant builder. So it takes long time, a little bit. But yes, if you have determination, this is how the route to go. So the summary of this is, say that graduation is not the end actually. If you get a PhD degree or a master's degree, it doesn't mean that you are ready to become a plant builder, commercial plant builder. It's just a beginning actually. So you are learning the basics, and after this basics, you have to learn a lot more, and to be aware more about the new and newer technologies which are right now probably you are not aware, and you don't get opportunity to hands-on training and all that. Maybe going forward in the future, you should be ready to learn more, and then you are going to, but basic thing is you should know the fundamentals and basics of the plant breeding and genetics. So expectations from the young breeders. Young breeders is, I would say, from the students who are just graduating from the schools. This is what is the expectations. This is should be ready. Molecular information, must be prepared to handle molecular information, and then field information, data note taking and all that, large data sets and high performance computing kind of things. Then you should have, good about the knowledge of the genetics and phenotyping, and then how to handle the germplasm, the knowledge of the germplasm, and different environments where you are going to evaluate your materials and how you are going to evaluate. But beyond that, in a commercial plant breeding, the one major difference is you also must be aware about the business, how the whole business runs. So you should be knowing about the business plans and the business strategies and then objectives, vision, and what are the legal and regulatory requirements, because you should not use the germplasm just like that. You should have proper documentation and how to use that. You should know all the legal requirements. Sometimes they may land in the trouble if you use the germplasm wrongly and illegally. The basics of the seed production is very important. Sales and marketing, what they want, because if you may make a very good hybrid which is extraordinary, but if it is seed parent is not really giving good yield, farmer is not going to take up production, or if it is nicking is not matching, he may not really take up. So company has to produce the hybrid has to spend more money because yield will be less. So product margins will go down. So it's not only the product making, but how to make a successful product and address what I said is those three stakeholders, organization, and then grower, and the end user, the buyer. So let us look at what it says that traditional skills are the only minimum entry requirement. Understand the basic of business is very critical. How business is run, I just put one snapshot here. The marketing process, this is how the organizations run. They will have a proper business plan and objectives, market strategies, and then they will sell the product by using different tools and which we put it simply in a MBA graduates, they put it in a 4P, like a product, price, and promotions, and the place. So all these things together, product is only one element in the whole system. Now if you look at the product, there are a few more things here we need to know. Look at here. It is the performance and feature which is the only part that we play the role as a commercial planners. But there's a much more in a commercial organization which you see here is, the quality, packaging, warranty, services, pricing strategy, retail, discounts, advertising, promotions, distribution channel, coverage, inventory management, warehousing, addressing the issues. There's so much. This is how the business runs. So the role of product development is this, but most critical. If this is not there, nothing else can be done. So it's the most important. So a commercial planner has to know this whole model, how it works, and what is his role, what he is supposed to do to make it successful. Builders should also know about who are the players, because they will be launching products every year better and better. They are also competing, so keep your eyes open. World is changing, things are changing on daily basis. So you need to know where I stand and how I'm going to compete and making it the business sustainable, because it's very difficult. Not that. Sorry. No, I just put the key players as something like private sector, right? Because I'm talking about some commercial breeding. And there are some of the learning from commercial plant breeders. So here is some of the quotes I thought we had a very senior director in DuPont Pioneer who we asked him to share when he was retiring to some of his experience, and he put these 10 commandments, like some of the things which he says there should be no ego, when you should not have any ego, because plant breeding is performed, product development is performed as a team. You are not alone, there are other team members also. You should not have very high ego that I am the only one and rest is nothing. So unless you are a good team worker, this thing is not going to be successful. For young leaders, when you become breeder, make sure you park your ego aside and you are fully committed to the profession and do it as a team worker. This message is typically I am putting this slide for students. And the next one is journal plan is the key, because without journal plan nothing is successful. So make sure you use the journal plan very aggressively, evaluate it properly, and share it with your other partners whosoever like in multiple programs. So exchange it and share it and use it more aggressively. Advanced degree do not make a breeder successful. So degrees, as I said in the beginning, is a starting point. That doesn't make you a qualified breeder right now. So it will take some more time to learn from a real guru. So keep in mind that you have to learn a lot when you start doing as a breeding job. In breeding program, look forward, not backward. What it says is don't try to catch hold of and hold on the one particular material. Don't become so emotional that I will use this material, this always. Sometime, holding a lot of old material and you don't throw it, your breeding program will become unmanageable, size will so big. You should be very good in discarding the material. A good breeder is the one who discards more so that best material is retained. Use that approach. Be ruthless in discarding the material if it is required. Listen to the production and sales team. As I said, the production is very important. Sales team is, unless you don't understand what is required and you are working on different aspect, marketing team wants something else. It is never going to be successful. Always listen to them. They will give the feedback and set your criteria, what they are expecting, what level of yield, what level of maturity, what level of traits. defensive trade, diseases and all that. According the program has to run. And production people also, they'll tell you how much cereal minimum they have to. So you have to develop input line seed parents which should be minimum at that level to make it sustainable. So it's beyond the product development, it's beyond the hybrid, many more things to consider. Don't worry about job grades. Sometimes there will be situations where a breeder job is to make some selections, certain decisions, but during the peak season, the bagging, pollination, seed packaging, there are so many other things where we should, as a breeder or a program leader, should join hand with our research associates and junior scientists, also help them as a good team leader, because sometimes workload, he has to join hands and do work. So don't worry about that, I am so and so, so I will not do. Seed packaging is not my job. In industry it doesn't happen. I have done many times bagging and pollination with my colleagues. And respond promptly to the request of others, because many of the people, other programs, they'll be looking for the germplasm more aggressively, there are cut-off dates, planting time, so always respond to them timely, share your material, data, information so that they can make faster decision. Say thanks to open, be a good team leader, good team worker, and always acknowledge what has been being done by your team members. Give breeders room to express their own personality in their breeding program. This is very important, because being you as a leader, nurture talent in your own team and train them being as a group, and so that you make a very good the trained professional in the program, and program never suffers and product delivery will never be impacted. That ultimately helps the country, helps the farmers. So train more and more people, being if you are experienced person, so that it benefits the country and the farmers. So this is the last slide. Common myths about reality about the career in agriculture. Some of the common myths people talk about is advanced degree take too much time. We should not go for PhD. We just should somehow get a job. And sometimes the research careers are not very rewarding. Some people say private sector jobs are not really not very rewarding and not very stable. Anytime they can be fired and all that. You have to work too hard in private sector, too much work. So I would say. Actually, it's not true. The reality is very, very different. The spending time on PhD, it broadens your horizon, and that's fact. You get much better opportunity if you go for higher education. Research careers are some of the most rewarding. Yes, being a research professional, you get most satisfying job satisfaction when you develop hybrids and when they are grown on millions of hectares in the country. And when you go around and see in the farmer's field its product which is developed by you, it gives so much satisfaction you cannot imagine. Private sector offers immense scope for quality research. There are a lot of latest tools and technologies. What is shown yesterday and till today morning doesn't mean that you as a breeder have to run the whole show. You are not going to run everything. There are professionals available, different expertise available in the organization who are going to run molecular data for you or analysis for you, and then provide information to you to utilize in your breeding program. So you are not alone, there's a whole team. So no need to feel very scared that there are a lot of other tools and technologies are coming and probably research is going to be too much of work and too difficult to deal with. It's not that so. The breeder is supposed to do breeding job, and he's provided all the support by different expertise, different team members. How hard work is the only way. Yes, that's required, whether you work in a government job, public sector or private sector, everywhere work has to be done. And today, nowadays, I am seeing many of my colleagues, they are working even much more than me in the public sector. So don't think that anywhere the work is less. Work is everywhere. You have to work hard to make progress, and it's very important. So with this, I'll end my presentation. I would say I worked for almost 20 years as a hardcore millet breeder, and could succeed in developing many good products because of hard work and because of good support from the organization. And many of you may not know that pearl millet developed by Pioneer last, I think, almost 1/3 of the Indian continent, 1/3 of the hectares of India, the Pioneer pearl millet is being grown. And last 25 years we are leaders in that. So the commercial breeding is a lot more fun. And I wish you all the best, and you people are the best to judge what career to choose and what to do. But there's a lot of fun in this, and once you start enjoying this, it's not very difficult, it's very easy. So thank you so much for your time.
Thank you, sir. Thank you for this wonderful talk, and I'm sure this is going to help our student a lot in making their career in successful commercial plant breeding. As this is a student symposium, the questions from students are highly appreciated. Please.
There's a question from Dharwad, University of Dharwad, student from Dharwad. Being key player in the society, why most of the private companies are not interested in developing commercial and commercializing hybrids of pulses and small millets, which are potential of nutritional security in India?
Good question. I think I'll put my perspective on this. Generally, most of the commercial organizations, they are, it's a business. As I said right in the beginning, the plant breeding is business nowadays, and they look for where there are more business opportunities. And these smaller crops, number of acres are very, very small. Actually, the organizations want to get into this, but actually the opportunity is very, very small. The kind of money they have to invest in terms of research activities, that hiring people and the resources and everything, they don't get returns back on that. And that's one of the reason it is not on their high priority list.
Throughout your presentation, I was wondering whether you should really add this adjective commercial plant breeding. Can you make a distinction between plant breeding and commercial plant breeding? Should we teach our students plant breeding and then tell them the difference between commercial plant breeding and plant breeding? Dr. B.D. Singh may have to add a chapter in his book now on commercial plant
breeding. So, well said. Yes, sir. So well said. The breeding is a breeding. Breeding is breeding, and breeding will breeding and breeding will remain plant breeding all the time. Only thing is whether breeding reaches to the customer through a commercial organization. Yeah.
Why call it commercial plant breeding? You are conveying a wrong message to the students to make a distinction between plant breeding and commercial. Plant breeding is always commercial.
Yeah.
Plant breeder always has to keep in mind the commercial aspect.
Yes. So.
Dr. Mahala has already associated commercial plant breeding is beyond product actually. We have to associate the product superiority with the cost of production of that particular product or hybrid. We have to look into the placement of that hybrid as per our company's plans or strategies. We have to look into all our commercial plans as per the companies, where they want to put the product or hybrids. So it's what I'm trying to say. It is developing the products.
You are talking about something after the product development. That is marketing.
Sir, there are several examples in corn actually. That Anand can also tell about that. Cost of production of certain hybrids was very, very high. The product was very good, excellent, but the cost of production was very high. That didn't went to the commercial market. There are several examples in the corn.
So it's a very valid point, sir. Breeding is breeding. I think the intention of the speaker here was to say that additionally, what are the students needing to learn in today's educational institutes and today's curriculum to be industry ready. I think that is his intention when he coined the word commercial plant breeding, but it doesn't, the intention I think is not to say that the breeding is different, and breeding is fundamentally we learn from all of you and what we have learned in our graduate school. So that's not going to change. It's trying to get ourselves, if so somebody wants an industry job, what else do you have to be thinking of? I think that's his intent. Thank you.
Right, I think well said. Yes.
Hello, sir. Hello, sir. I have a question.
Okay.
So,
okay.
the plant breeding is a number game. So in segregating from the germplasm to the final inbred lines, so how you are handling segregating population, the numbers, and also the methodology that you are following to handle the segregating population and the selection, how you are going to affect the selection in the segregating populations in early generations.
I think there are, first thing is the size of the breeding programs, varies organization to organization depending on the organization's objectives. And each organization have got, refined the breeding programs depending on the success they have seen, and those numbers are not fixed all the time. So there cannot be exactly like number, okay, this much if you do, this much success you will get. It all depends on the what kind of germplasm you are using, how you are advancing your material in top cross levels, what kind of testers you are using, and then how many plots you need, how many testing locations you need to become successful. So different organizations use the different schemes, and it's difficult to say that exact, okay, this is the number we use. But yes, every organization has got a plan, how many products every year they want to release, and based on that they design, customize their whole breeding program. No, generally most organizations hybrid breeding program, pedigree methods and all selection methods are same they are used, but matrix varies. So the breeding programs are based on a fixed matrix, and these matrix numbers vary organization to organization.
Sure.
Yes, sir.
That was very nice presentation and very effectively communicated.
Yes, it's working.
Throughout your presentation, I was delighted that it showed the importance of plant breeding activities, the plant breeder, and also the challenges the plant breeder has in a commercial organization. But in one of your slides towards the end, what I could see was that plant breeder was very small guy in a very large team, and he has to listen more than he is listened to by others.
Yeah, sir, I just wanted to give an overview to the particular students sometime, that because to make the product development process successful, ultimately it has to reach to the farmers. And often, you might have come across sometime in your career time and all that, you might have seen that there were wonderful products developed, but they never reached to the farmer level. Either they were not producible, or they had high yield but they had some of the other problems like lodging or disease or something, because they were not fully evaluated. And breeders programs, even I have heard, collapsed completely because the breeders did not have the good information about from the what customer needs, and they were running the programs, and end of the day they could never succeed in developing the products which could finally farmer could accept. So this is very important. I thought it is a good opportunity to give this kind of overview to students, because when they start their career, it's very important for them to understand that they need to know the customer, they need to know the farmer, what is required and how to develop that. Thank you.
I have one question. We teach to students about breeding cross-pollinated crops, breeding self-pollinated crops. So when we teach, the students have a question in their mind, and it is also a public opinion also, that the breeding about cross-pollinated crops, especially hybrids, the public sector is lacking and private sector is ahead. So what, as a commercial plant breeder working in the private sector, what do you think that what are the lacuna the public sector has, and what are the advantages you have? And we want in terms of the team working on the plant breeding, the number of people assisting, and right from your product development to distribution of the seed to the farmers. We just want to have a
good question. I think I would say it's difficult to answer what lacuna public sector has, because I have never got an opportunity to visit public hybrid breeding programs and look deeply into that. But yes, I would say, based on whatever experience I have in my organization, I would say what is more important is to know what is required first. It works reverse. We should not start some breeding program just with what we assume. We have to listen to the customer, so we should know the first information from the farmer or end user what they want, and then based on that we should plan all our breeding strategies. And bidding strategies we have to plan based on the timeline, the cost, and then right germplasm, and then the correct process, the right process. If we use the right process, right metrics, and a perfect timeline and target, clear objectives, I think bidding program will definitely be successful. This is what I do. I listen to the sales people, I listen to production people, I listen to farmer, I visit, make very frequent visit in each and every year in each and every state, wherever the crop is being grown, and talk to the farmer every time, see what new challenges are coming. Accordingly, my team, we customize our bidding program, make some changes. Like example, the blast is becoming a very serious disease in pearl millet now. Okay, but the good organizations or smart breeders, you will be surprised, already started working about 7-8 years back itself, making the bidding process, screening the material, putting that in the bidding program, and they are coming up with the new inbred lines. And products are available in the market and getting more available. So 10 years ahead, you have to always keep thinking what will be required, or at least 7-8 years, and what I should make the changes, identify the gaps in the program, where are the gaps, how I fix those, how to accelerate the bidding program, number of processes should be more or less, or how many locations, and how to evaluate. So robust process and the good vision, good clarity about what is required is very important.
And what would be the size of your team?
Very interesting to we are just 10 people, including myself, and 3 breeders and some RAs and temporary staff. And we are running huge program, 3 breeding programs and 15-16 locations like that, and many activities, I would say. But it's very well managed and very effective. So I would say for a program, I think 10 is a very good number, not a big, to run even 2-3 programs.
Thank you.
Sir, I have a question like that. What may be the effect of government regulation in setting up the objective for a breeder? Like if the government has said that, like I can say the example for in corn itself, that if the government has said that for corn flakes or any corn item, the protein percentage or amino acid content percentage should be like this. So what will be your response immediately? Because you told that that a breeder should have to deal means up to 7 or 8 years before. So for that immediate regulation, what will be your response? So how you will deal with that?
I don't think anybody gives any immediate target that within 2 years. Everybody understands bidding, how much time it takes, and professionals know it very well. And those who are advisors on the government panel also, they also know that how bidding works. So it's not that just like overnight they'll say today you do it. It doesn't happen that way.
Okay, sir.
Okay.
I think it was an excellent presentation, Dr. Mella. I think the product development as well as the tech. Just to ask one query. When you talk many times we will be that. Just to reflect upon that, hybrid itself was one of the important development, is it that, as you know, and if you can say that the product development in hybrid maize was a complementation of public research as well as the private, because a lot of work was done by the inbred line development which would not have been sustainable as them on their own. So there was a good complementation when even 100 years back. So that time there was a good at least rapport or at least involvement at that stage. So now currently I think a lot of water has flowed, and you have indicated there is take away home message for both the public sector. So currently I think there is also involved new thinking in the public sector, because even 10 years back we didn't much think of complementing. We are thinking of MOU. Now there is a MOA, that there is a lot of business coming out of it. So in this context, how you perceive the complementation between public-private sectors? Particularly your company.
I think if I understood the question, I would say how the public and private sector is going to work together. I think one of the best, most successful model right now everybody is talking about, and private sector is also coming forward and they want to work together, is public-private partnership models. And it is much more evolved in many other countries where they have, the universities have got their legal departments, and they take care of that thing and then work together as joint projects kind of thing. And I heard here in India also now a lot of private sector companies who cannot afford certain tools and technologies, or doing their own research, they want to partner and come forward. Either they want to sell the product developed by public sector, or they want to have some kind of joint kind of research where they can come up with the products and then sell in the marketplace. So public-private partnership is going to be an excellent model going forward probably. I think some more policy refinement and maybe some more clarity is required on that at every public sector institute level. And if that becomes more and more stronger, the partnership is going to really become very, very strong. And I think that's the future.
A very big hand to Dr. Mahela. We all know break for lunch, and we'll gather after 30 minutes. Because we're running short of time, we request all of us to have a quick lunch and come back.
ಊಟ ಬೇಕಿಲ್ಲ ಒಂದು
ಅವನೇನು ಮಾಡ್ತಾನೆ अच्छा मेरा वहाँ से एक मिनट एक और चेयर लाना पड़ेगा ना नहीं
Also not here.
You had your lunch?
No.
No?
It's okay. I'm not sick. It's okay, ma'am. It's okay. It's
No,
okay.
no, no.
It's okay, ma'am.
खाना भी नहीं खाया
Please. Actually, I had very heavy breakfast. That's why I didn't eat. No, please. Today's last day in MS. Okay.
I am going to be standing here and so we take out my name and come back.
Okay. So no one will hold the chair.
No need. I'll stand.
Sorry? I want the
Welcome back once again. Let's continue the session 4. Our next program is panel discussion, where various eminent personalities, both from public and private sector, are going to discuss about are you seed industry ready? This panel is chaired by Dr. B.P.Pal, Professor of Genetics, ex-Vice Chancellor, University of Delhi South Campus, and the other panelists are Dr. R.M. Singh, former Professor and Head, Department of Genetics and Plant Breeding, Banaras Hindu University, Varanasi, Dr. K.V. Prabhu, Joint Director of Research, IARI, New Delhi, Dr. Anand Pandravada, Research Director for Corn, Asia Pacific, DuPont Pioneer, and Dr. Amitabh Mohanty, Senior Research Manager, Trade Discovery, Optimization, DuPont Pioneer. This panel would be facilitated by the Dr. Raman Babu, Technology Deployment Lead, South Asia, DuPont Pioneer. I would like to request all the dignitaries, please come on the dais, sir. Thank you.
So very good afternoon, friends. Thanks. I think Dr. Prabhu is here. Sir, please. So I think welcome to one of the most important sessions of this symposium. Okay. This is basically, I set this up. I wanted to get some very good feedback from the students. I really request you to open up. This is the session where we would like to hear from you. Right, so the purpose of this are you seed industry ready session is threefold. One, we would like to encourage very active participation from students, right? Please don't be intimidated by all the technology things that we are showing. I mean, these are all, after all, applications. The kind of education that you gain in universities, in prominent institutions like IARI, it essentially sets you up to face the world. So you don't have to worry about it. These are all just application part of it that we are demonstrating here. And number two, we also would like to know, are you equipped enough to perform some of the functions routinely that are carried out in the seed industry? So we would like to know. I will just briefly go through four or five slides to ask you some questions, and there is nothing right or wrong answer, so please don't feel shy. Whatever you know about that, please open up and let us know, and we would like to know what curriculum addresses these essential functions that are performed in seed industry. So we are joined by a host of very renowned experts on the stage here. We have Dr. Professor B.P.Pal, Dr. K.V. Prabhu, Dr. Amitabh, Dr. R.M. Singh, Dr. Anand Pandravada. So between them, there is tremendous teaching experience and there is tremendous seed industry experience. So I am going to go through a set of slides for first say 5 to 8 minutes. We'll keep it very brief, and then I would request students to open up, and then in the end I would like to request the panel members to speak each 5 minutes or 3 minutes each, depending on the time we have. Okay, so let me go through the slides. So I am asking you a very, very simple question. Okay, there is no biotechnology involved in this. There is no genome editing. There is no genome-wide association analysis. There is no genomic selection. Okay, and so please don't worry if you don't know or if you can't perform this function. I just wanted to encourage interaction on this point. So can you evaluate a trial of 100 maize hybrid entries in 10 locations in Kharif season in South India, let's say, and select the top 5%. So is this an exercise that sounds okay for you? Can you do this? How many of you can do this? Suppose we give you a data, right? Grain yield data for 100 maize hybrid entries in an Excel file. You have 10 columns, let's say locations. Under each location you have 2 or 3 reps, whatever it is. You have the data with you. Can you select the top 5%? How many of you are confident that, I mean, this is something, if you join on seed industry, you will do it every day on a regular basis. Am I right, Anand? So this is something you will perform every day routinely in your analysis. So how many of you think that you are confident to do this? You have done this, and there are courses in your curriculum which helps you do this.
Those who give the answer will get the job. That's what he is offering.
Those who give answers, I can assure you that I can call you here and ask you to demonstrate. Sorry, I am kidding, yeah. So please feel free, and if you don't know, that's fine. There is nothing wrong in not knowing this. So how many of you are comfortable? Nobody? Sure? I'm sure some of you must be doing some analysis with your data.
One day I believe with it is that you all raise your hands.
Okay, so let me just go ahead with. So if you ask me to do the way, or only me, anybody in the industry, or anybody in the public sector, all the teachers, all the researchers in IARI or in any other agricultural universities who run All India Coordinated Program, they all do this. Okay. So what do? We generate an augmented or partially replicated design, right? Can you generate a design using any software? Can do, right? So you can lay out a row column structure to enable spatial analysis, collect data digitally, identify and eliminate bad locations. Suppose if you have 10 location data and you want to identify bad locations from there, what statistical parameters do you use to identify those bad locations? And identifying the spatial variance structure and using a mixed model, something like ASRML, can you adjust those means? Elucidate the environmental correlation. Suppose you have 10 locations, can I take average of all those 10 locations, or should I consider some kind of grouping before I average my values? Blues and blubs. How many of you have heard these terms? Blues and blubs? There are at least 4 or 5, right? Can any one of you explain what is a blue, what is a blub? Why do we need blub over blue? Just, I mean, you can say whatever you want and nobody is going to judge. Just feel free. Well, the basic purpose of this session is to make you speak. Okay? So, yeah, please. Can someone get the mic? See, the basic purpose of this session is to know what the seed industry is doing, whether you have the required skill sets currently to do this. So if we identify some gaps, then perhaps that is an entry point where we can start pushing that, these are the things which are routinely required, and the curriculum is not addressing that. Can we?
So, blue is something basic linear unbiased estimates.
Very good.
And blobs is basic linear unbiased predictions.
Right.
So what is the meaning of that? I don't know that.
Very good. Very good. But you know the abbreviation at least. Okay, next. Anyone else? Okay, so I am not going to discuss any answers here. So I want you to know that these are the relevant questions to seed industry, and I would encourage you to go and read about these aspects. Can you use the mic? Okay, okay. Okay, fine. So, and then you construct a selection index based on what you have estimated and select the top 5%. Okay. So, actually one of our objective of this symposium, we wanted to also demonstrate how we do this, but I am not sure whether we have time to do that, and also there is some issue with connecting our laptop because you cannot use the external laptop here right now. So, but I just want you to understand that this is something very, very essential, very, very basic. There is no technology advancement here, right? So, we have been doing this for the last 100 years or so, and this is all being done by your own teachers, your own researchers in your institute. So, please go and talk to them. Okay. So, I just wanted to emphasize that quantitative genetic skills are very, very vital, and I recollect whatever Dr. Prabhu taught us in terms of elements of quantitative genetics or advanced quantitative genetics every day in our work. Okay. So, this is something very essential. So, how do you analyze a multi-location trial? What are spatial analysis? What are considered fixed and random effects? Are these questions sound familiar to you, or are you totally new, or have you all gone molecular? Yeah. I suspected it. So, in our eagerness to adopt modern breeding technologies, have we forgotten these basic fundamentals? But these are the things which will really help you in your everyday function when you join seed industry, right? How to calculate covariate adjusted grain yield. Suppose you know that anthesis date is influencing your grain yield trait in your trial. How can you adjust grain yield based on plant height or plant anthesis date? How to estimate the tester effects if you are working in a hybrid crop. So, what I wanted to know is what courses in your curriculum familiarize you with the above concepts. So, at least in IARI we know that there are certain courses that I have personally taken. I am sure all currently the students are taking, but what about other institutions, students from other universities? Do you think there are courses which deal with these concepts? Students from other SAUs are encouraged to speak, please.
Advances in quantitative genetics.
So, you are taking this course?
Yes, sir.
Okay, very good. How about others? You are from? Students, can you introduce your name,
Agriculture
your
University, Pusa, Samastipur, Bihar.
Very good, thank you. Students, please introduce your name briefly, where you are coming from. If you are not familiar, if you do not have such courses in your university, please speak
Hello,
up.
sir. I am Sneha Adhikari and I am from Govindvallabh Pant University of Agriculture and Technology, Pantnagar. And sir, we have studied advanced experimental statistics, and right now we are studying advanced experimental design and advanced biometrical statistics.
So, do you think the content of those courses enable you to perform this analysis or not?
Yes, sir.
You can do this?
Yes, sir.
Right, very good. How about others? State agriculture universities. I know we have students from PAU, MPAU, Udaipur, Jammu Kashmir, Himachal. So, how many of you are comfortable with hands-on data analysis? So, I know that you have taken some courses, so you may be at least familiar with the concepts. How about data analysis? What kind of software would you use if you have to generate blobs, or if you, any software that you are aware of which can help you do this?
Sir, SAS software is we are just learning in my institute, sir.
Okay. So is there a person to whom you go to perform the SAS functions, or students perform themselves?
Sir, teacher performs, sir.
Okay, so there is a statistics department,
yes,
and you go and
yes,
get that done. Okay. Only usual suspects are speaking up. No? Okay, so there is, so I think, as I understand it, it's also one of the good representative group. And this also, sir, I think Prabhu sir, I would like to emphasize that other universities, quantitative genetics courses are something not taken very seriously, and the hands-on data analysis should be part of these courses, and we are not talking about any advances here. It's very simple basic stuff. So how about adapting yourself to the modern breeding programs? So yesterday I was talking to, we had lots of talks on all the modern breeding programs, right? So did you feel comfortable with all those talks? I think I know that some of you came and told us, very frankly, that sir, some of these things we don't even understand, everything is going overhead. So the point, like each speaker has only 30 or 40 minutes, and he's not going to make you completely understand all those things, but the point is to induce curiosity in you. Suppose if you keep hearing the same word again and again, whole genome prediction, genomic selection, all that stuff, so you are going to go back and read. I am sure at least 10% of you are going to go and read those topics. There are plenty of papers to understand. But that is the basic purpose of this symposium, to induce curiosity in students, and we want you to understand what are the currently relevant areas where you need to focus on and where you can build expertise. A lot of people come and ask us, sir, can you take us in your company, right? So I was trying to ask you back, suppose if I have 100 CVs, what is the differentiator for me in that CV? If all the 100 CVs are more or less same, and one guy is claiming that I know Python script writing, or I know how to generate blabs, or I know how to select varieties confidently, perhaps that is a differentiator there. So I think you will have to make some extra effort to learn those things, but you also should know what to learn. So that is exactly why I thought we should discuss this. The whole symposium you would have learned, what are the relevant topics currently. So how many of you have seen SNP data? How many of you have handled SNP data? If you are not handling phenotypic data, if you are not handling SNP data, then, and if you are a plant breeding student, there is something wrong. How many of you have seen SNP data?
Sir, I have done some created data with association mapping and QTL mapping.
Very good.
And use the TESEL or cartographer, but just try to mapping the QTL.
Excellent. Yeah.
But I just want to do QTL mapping with biofortified zinc containing rice, sir. But as a first year student of the PhD, sir, I was thinking that I can do it, sir.
Excellent. I am sure you will be able to do it. Once you have that interest to do, I am sure you will be able to do it. Anyone else who has seen SNP data? Please. Yeah, we would like ladies students to speak.
Good afternoon, sir. Myself Divya Sharma from B.P.Pal University, and we are having GBS data, the SNP data, and that data we have used in TESEL software for phylogenetic analysis, and we are also doing association mapping analysis using that data in TESEL.
Excellent. Very good. So we have more students on the modern breeding. The conventional breeding, we don't have many. Okay, good. So recently we were invited for a Vice Chancellor's conference in Hyderabad. So all the Vice Chancellors were present there. We were asked to make a small presentation on what are the seed industry expectations from universities. So they wanted to design curriculum based on feedback from seed industry. So we made a very short presentation to them. I just thought I will just go through this very briefly. So a plant breeder in today's world, we saw that, right? Dr. Mahala presented it very nicely. So he is in the center, and there are a lot of other disciplines that work around him, right? So in the center means he has to listen to everybody. So you should know a little bit of biotechnology. You don't have to perform all those functions, but you should know that how a biotechnologist can help the breeder, how a computer scientist is going to help the breeder, how pathologists, all others. So it is not anymore a single discipline. So it's highly complex discipline, and you should know what other disciplines can contribute to plant breeding. And this is all again only technical expertise. So you need business acumen. You should know what will sell in the market, and you should have some legal knowledge. whether you are infringing upon unintentionally on someone else's rights, or if somebody is infringing on your rights in the market, you should know as a breeder. And also you should work in a team, right? You should work in a very multidisciplinary team, and the breeder is the one who will be expected to take the lead on a crop improvement program. So you should have all the people related skill sets. There is no opportunity for you to get worked up. You have to be diplomatic, and you have to move around in the team very efficiently, right? You just see, plant breeders employed in the public sector in the US, only 27%, whereas the remaining 73% of the plant breeders are employed in the private sector. I don't think this picture is very different in India also. All the students nowadays, where are they going? Well, the ARS positions are very few, right? Where do you end up? Mostly in the private sector seed industry. So it's very important that you learn what is the industry doing, and if you are aspiring for a career, better be prepared for it. And we did a small analysis, like where are the gaps in education and training of plant breeders. So education, as they say, is very strategic, and it instills broad knowledge across crops. So you don't educate a person to become a maize breeder or rice breeder. You educate a person to become plant breeder, right? The other thing is training, which is very tactical, and it instills very specific domain knowledge, be rice breeding or molecular breeding or any specialized domain. So for a wholesome training, you need both good education and good training. Both are equally important. So education, you are getting it from your university, from your SAUs or wherever you are, right? IARI or all the institutions are doing the job of education. Whereas training part, which is also equally important, that's again are totally dependent on these universities. And I know that there are refresher courses being offered, but these are all mostly for mid-career professionals, those who are already employed, and they just get their skills refreshed. So whereas if there is a student who wants to go to a seed company and he wants to enrich himself, so where does he go? So that there is a gap there, and there are many players in this training domain, as I put here, ICAR, SAU, CGIAR, and there are international organizations, and private sector is also a very central part here. And what I believe is the feedback from private sector can make the programs run more efficiently, the training programs and other things, so that they can customize to the needs of the customers. I think one thing that private sector brings to the table is product orientation, which is very, very important, on the ground knowledge, and very, very closely aligned to the needs of the farmers, and it performs cutting edge science, right? We have been discussing all the latest advances. So a decision is taken on making use of this tool, then it's done. It is implemented, routinely implemented in the breeding program. So these are things that the private sector can bring in any of the breeding programs. So I was just wondering, look at the courses on the left side. There are plenty of courses on IT related fields, Java, Oracle, .NET, Hadoop, Python. So if somebody wants to learn, even a person like me who is not into any of IT sector, I can go and join a program in any of the market institutions, and I can, within 3 or 6 months, I can become proficient in any of these fields. But that is not the case with respect to these set of courses, right? Suppose somebody wants to get some hands-on knowledge on hybrid seed production, spatial analysis and blobs, double haploids, precision phenotyping, big data analysis in agriculture, whole genome prediction. Where does the person go? Genome editing and applications, crop modeling. So these are important areas that we need to address. All the players that I mentioned earlier need to come together and address. And currently I know that universities, institutions are offering refresher courses here. But what I believe is the things that you see on the left side, they are all demand driven, right? There is plenty of demand to take these courses, so they are in the marketplace. Whereas these courses are currently mostly supply driven. That a faculty is there who is trained on genome-wide association analysis, so we run courses, refresher courses on genome-wide association. Whether it is relevant in the marketplace or not, we do not know. Right, so that customization needs to happen in institutions. And what possible role for private sector? This is my last slide, and then I'll open this up. So what possible role for the private sector? There is clearly, you know, as I told you, the 3 points, product orientation, very closely aligned to the needs of the farmers, the cutting edge science. All that can be brought in with the help of the other players, and we need to make continuous engagement with the regional and global academia. And also I would like to, you know, just mention two evolving effective collaboration mechanisms. So one is the public-private partnership. Somebody talked about it earlier. I know that CG institutions have been running few public consortiums where germ plasm exchange and all the information exchange happens. That have been very efficient. And also there are some open source innovation platforms. So what do I mean by this? You know, recently DuPont Pioneer has been engaging with the global academic and public sector research institutions. So DuPont Pioneer has entered into agreement with CIMMIT on a very important cutting edge tool, genome editing. So DuPont Pioneer is offering this expertise to CIMMIT to fight a lethal disease in Africa called maize lethal necrosis. So DuPont Pioneer will offer this expertise to CIMMIT. Similarly, it has collaboration with IARI. DuPont Pioneer has offered on genome editing aspect, and I know that Dr. Amitabh is championing some of the efforts between ICAR and DuPont Pioneer on the same genome editing. So these are open source innovation platforms, excellent opportunities for the private sector and public sector to come together and deliver on what the smallholder farmers need. And proactive participation in modern curriculum design. This is something I am very personally attached to, and I feel there is excellent scope here. As we saw today, you know, there are some of the critical gaps, and that needs to be filled. And we need to support the establishment of regional and global centers of excellence. I mean, the private sector, whatever possible support it can extend to the public sector institutions, that needs to happen. So let me finish with that one, and I would like to hear from some of the students, and then if there are no more comments, then maybe we will open this to our panel members. Any student who wants to speak? Well, there is something very little about this. I just wanted to mention a few things about this symposium. This is a student symposium, and we have done this in many universities, and it's all completely driven by students. So please don't hesitate. Just speak up, and you don't have to worry. You will not be judged. There is nothing like good doubt or bad doubt. A doubt is a doubt. Somebody mentioned, I think it was Amitabh who mentioned earlier. Right? Yeah. So do you think there is a gap, or you think you are fine with what you are learning?
Hello, sir. My name is Navdeep, and I am from Palampur, Himachal. As you are asked about the quantitative genetics and whole, we are still in the initial phases. We still read the advances in quantitative genetics, and we haven't read the advanced softwares there. And we are similar for the SNPs data. At least I haven't, in our department, we haven't yet reached to the SNPs level. We still struggling with the SSRs and PCR markers.
That's fine, completely understandable.
So where is the gap, and this public private partnership or these things, I think Himalayan states are still far away, so we haven't yet any collaborations of these kind of collaborations there. So thank you very much.
Thank you very much. That was very excellent feedback. Anyone else? We may have to make it obligatory that one person from each university will speak. Yeah.
Good afternoon, sir. My name is Rudraj. I am from BHU Varanasi. I have a small doubt, sir. From 1.5 days we are talking about molecular markers and genomics, sir. But students, mainly I think in this age, they have to concentrate on the conventional breeding. Along with that, they have to have the knowledge of the genomics. Most of the students, what we are seeing means we don't have the proper knowledge of the field. We are mainly concentrating on the wet laboratory data, the data which we are taking from the field experts or the field assistants. The students who are not going to the field, they are just taking the data from the field assistants, laborers, and they are generating those data. So how can you say that students should rely only on the data?
So the data is not taken by the student?
Yes, sir.
That is a serious issue. At least in IARI, I know that will never be allowed. Student is directly involved in whatever he is doing. No, I think it's a good observation. Whatever you, we go by what you say.
No, we don't take it otherwise. But the point he's making is very relevant.
Right, yeah.
Because most students are interested more in learning because they don't feel excited in going to the field and measuring routine observations of height, weight, or this flowering, or things of this kind. Maybe in the sense of learning, achieving something by using molecular tools, etc., or software, and
they are overexcited.
So that's obvious. But then, as a researcher, there should be no alternative to real hard work in the field.
And just to add, I think the observation is right that with the focus on these modern tools, the emphasis on conventional plant breeding techniques, what you do with pedigree selection, what you do with population selection, what you do with recurrent selection, and the ability to know when and what you use where is something that is very, very fundamental. If you didn't know that and you had all of these technologies, I mean, the breeder, that person will not be able to do anything with it. So that this is still the fundamental, and so I think the point is very valid that the emphasis has to be on these aspects to make sure that this is done first before the second thing is done. And every student should make sure that they are getting this information first hand.
Very, very good point. Yeah. No, I used to, sir, I don't know, Prabhu sir remembers or not. So we took elements of quantitative genetics and advanced quantitative genetics. So after the 2 courses, I went and asked sir that, sir, we wanted to learn QTL mapping, but you didn't teach us QTL mapping in this course. So he used to smile at us. Today I realized why he was smiling. Because what you need to learn is the principles, basics and fundamentals. How allele frequency changes, how your linkage happens, how linkage disequilibrium happens. If you learn the fundamentals, any modern tool, learning that modern tool is nothing. So it's very easy. It will come to you very naturally once you are through with the fundamentals. And that is exactly what Dr. A.K. Singh used to do. Teach the very basic fundamentals. And we had the freedom to barge into his room anytime and ask as many questions. So he used to be very open, so he was the constant victim of all the students. So,
thank you.
So maybe like to
it was very, very good. He said it very well. So the important point is when you get into modern tools, which are just translating the information on the material, nothing else. Whatever be the technology or tool which you are going to use in the name of DNA-based data. But what is important is the material on which you get the data. And development of material is pure classical fundamental plant breeding. So that cannot be say put to a second or back burner, because that is the one that burns first. Rest of the things are only generated by that flame. Okay, so working in the field, understanding the crop, understanding the trait variation and the way it adjusts to the environment as it changes are all things which should be inculcated in the plant breeder as soon as he enters in improvement territory of that plant breeder, of that crop. So therefore, nothing can replace that knowledge. That is only by hard work in the field. As Dr. A.K. Singh said in the morning, the plants whisper. He said they talk. Okay, Dr. Borlak said they whisper. You got to go so close to listen to their whisper. That is possible only when you spend more time in the field. So there should be no alternative to the hard field work. Then comes the rest of the things. Okay, including the when you cross. If you by mistake take a twig from the next plant or the next line, you believe in on your data record it will be cross of A into B. The inbred could have been actually D, because the branch would have come from there, and the pollination that actually happened is the D into A, and what you're analyzing is polymorphism between A and D, something on the DNA which has come from some other line. So there is going to be no match between the data you collect and the analysis you do, the molecular analysis, isn't it? So it is very important. You don't disconnect yourself from the field.
Thank you.
Amitabh, want to comment?
I'll emphasize on two things, ingenuity and attitude. What we need to have is a positive attitude, that if you want to learn, you can learn anywhere. One of the person who talked about that, oh, we are doing this, what is next? How do we apply, right? When you interview, and believe me, we have interviewed thousands of people, okay, thousands of candidates. And what we look into is not really what they have been trained, right? You can train people, but you got to have the right attitude, right? If you have attitude of learning, if you want to do something, you want to learn something, rest things become secondary. Tell me what is there, that we spoke that we could not learn. Everything. There was somebody who was like, first to do it, right? And then we followed. Or maybe one of us did first something, right? So what is needed for to be successful in life, is attitude. So if you have the right attitude, if you have ingenuity, for example, somebody said, first of all, please don't get my comment wrong. I strongly believe that you got to spend a lot of time with your plant. Even today, I spend a lot of time in my area, looking at the plant, how they phenotypically look different, But if you don't like, so suppose you don't like that, to do every day, what you can do? You can fly a small drone, like all these small toy drones are there, right? And with one of these softwares, it's very basic, elementary, knowledge. You could write a script to analyze that data. It's not a rocket science, right? But you could do it. But then again comes attitude. That you need to know, how to measure the plant data and whatever you're doing. And then how do you apply technology is up to you and your attitude. So I would say that if you have the attitude to learn, you could do anything, and not be challenged by anything that is coming across, right? Whether it is next gen sequencing, genomic selection, genome editing, it could be anything. Thank you.
Amitabh is actually heading the trait discovery and optimization team. It's a huge team, lots of very talented people doing research on high throughput functional genomics as well as genome editing. So perhaps Amitabh, if somebody wants to have a career in that particular line, what do you think are the required skills and how can they acquire those skills?
First thing again I will say basic, Babu said that, and Professor Pentel is here, maybe I should defer the question to him, maybe after adding a couple of points. So you need to have your fundamentals clear, right? If you are if you want to go to plant breeding, right, you got to have the fundamentals clear. You cannot use any modern tool if your fundamentals are not clear, right? Because those are advances of the basics, right? But if your base is not good, you can't have a multi-story building unless your base is right? Same way, if you really want to do good in molecular breeding or molecular biology, genome editing, you got to have the fundamentals right? You got to understand how the mechanism works, right? What is a gene, what is a promoter, all those basic, how transcription translation happens, which is taught in all our courses, right? You do not need to know this experimentally because there are protocols. Once you go do the stuff, we can teach you, okay? But if your fundamentals are not clear, even if you give a SOP, if somebody is instructing, you would never be able to excel. You may do a job, but you won't be able to excel, right? With that, maybe I defer this question to Professor Pentel, who has been our teacher.
So we will, he will have the last word. So Anand, would you like to go? Yeah.
So, I just wanted to add, Amitabh mentioned attitude, and I think that's the most important thing. And so, I can go back to my graduate school days and my, experience on trying to do this. And when I was given admission for a PhD at Kansas State, my professor told me, I'm giving you admission not because you're intelligent, I'm giving you admission because I saw you working in the field with these people in the morning and in the evening, and that's the attitude that I like. So again, I just want to go back to this point that you can learn things, but you have to have that basic dedication to what you're trying to do. And if you're a breeder, your basic dedication is to selection and making sure that you're trying to put your efforts in trying to learn some of these basics and fundamentals right. The other point I just wanted to emphasize was, in today's world, there are many places where things can be done. So students encourage you to think outside the box. So if your institute only has a gel machine, there are 100 other places where there is a SNPs available, where professors and institutes like IARI, like NBPGR, like private institutes, where people are willing to do this and can do it, and will be encouraging. So just reach out, expand your horizon so that you are able to learn it, because this is the best time that you can do it. So somebody working on SSRs, figure out if somebody will do SNPs for you, convince them that this is the right project where they can invest their resources, so that both the people realize value. So, and many people will be there helping you out on this.
Anand again has huge industry experience, so he's leading the research. He's a research director for Corn, and he, before Pioneer, he had tremendous experience in Monsanto in the United States. So I think he's again one of the important space to watch for in seed industry. And do you, like, if they want to acquire some skill sets relevant to what we discussed, where do they go, Anand? Like, what are the places they can go? So where they can learn these things?
Babu, there's no, and that's a great question. So, for us, we struggle, and I know you have done this, and we have done this, and we had great mentors here in places like IARI, and in our universities, PJTSAU and Angro, so that we could sort of go and say we wanted to learn this, and then we're given that opportunity. But in today's world, there is plenty of places where you can seek information, and
any online thing that you have seen come across.
Right. So many of these, many of the online databases that we have, everything in the public sphere, right from Grameen to, today Professor mentioned QTL cartographer, ASRML, most of these things, they and are. So all of these are public open source software. They have modules that are there that are now enabling both phenotypic analysis, genotypic analysis, sequence analysis, and everything from a very low complex basic ANOVA to a highly complex sequence alignment within integrated functions. So R, for example, is one of those most powerful software. I know a lot of people use it, and maybe somebody like Justin has a better idea of what would be available as a public tool, given his exposure to some of these things.
So Justin and Emerson, later on I'm also going to request both of you, because you, Dindo as well, so you come from a different country, so your perspectives on what we are discussing here.
And I would also like to add here is that, in terms of practical experience, DuPont Pioneer and DuPont is very open to collaborate. We do take a lot of interns. We have been taking, some years we have taken even a dozen of interns in our institutions. We also participate with VCIL internship program. So that's another area. If you want to learn something practically, we do provide that opportunity as well.
May I request Professor Aram Singh to briefly comment?
Professor Pentel, am I audible? Okay. Professor Pentel and other colleagues on the dais. I have been basically a teacher and then also a researcher. As a teacher, what I feel is that the basics of genetics and basics of plant breeding, which is taught at the undergraduate level, it forms the basis really for students who go for postgraduate and PhD program. And that way, there has to be a significant improvement in the teaching curriculum at the undergraduate level. Of course, postgraduate level, Dr. Prabhu has already mentioned certain things, and those are definitely very important. Even subjects, only genetics and plant breeding. At most of the places, these are the subjects which are taught. I remember when I joined BHU, there was a course like agricultural botany, where we used to teach about different crops, their foliar biology, when the flower opens, when should you do pollination, how should you do it, all these things were taught. How would you classify, how will you identify a particular plant? That is not being taught anywhere now, neither at +2 level, neither at undergraduate level. Straightway we come and start doing plant breeding. My real apprehension is that in the curriculum at the undergraduate level, I mean this is a policy matter, I would request from this forum that in most of the agricultural university across the country, there should be some kind of a training of the students in this domain, in this particular area. Of course, when a student goes for doing MSc or PhD on a particular crop or in the lab, depending upon the problem, gets familiarizes with the particular crop, that particular crop only. But that is not sufficient. You have crops, self-pollinated crops, cross-pollinated crops, the apomix and so forth, and each one of them they have a different kind of breeding behavior, and this is very important to my mind. The other point which was raised by one of the students, that the data recording has to be really done by the students themselves. The researcher, as far as possible, should monitor it, whether it is being done properly or not. Otherwise, we end up nowhere. What happens many times I have seen, many crosses are made in wheat at our place, and when you go and look at the segregating generation, there is no variation in the F2. And people make selection in that F2 even when you don't find any variation, you see. I mean, that's the very basic principle of selection, is that you must have variation. And when you have made a cross where which are very similar varieties you have taken, naturally there would be no genetic variation available. If there is any variation, it will be very small, and also some of it may be environmental. So this is also very important for the students to see what a segregating generation looks like. I mean, at the place where they are really working, whether MSc or PhD in different crops, so that they get a feel of it. I mean, we say variation, but then what it really means, this is not really easily conceived by the students. I used to give a example in the class that in this particular class here, the students particularly, each one, you look different, you see, even though your genome may be the same in terms of the amount of the genetic material, of course variation at a finer level, but that is what is the variation in this population here. Similarly, in the crops, when you are selecting, your selection of the parent is very, very important. Doesn't matter whether you are working with a self-pollinated crop or cross-pollinated crop. Particularly here, like I was looking at the inbreds here, which was being presented in case of Symposium maize. You could see the inbreds. Some of the inbreds are very good per se. When you look at the filling of the ear grain, some of them are perfect filled. Others are, half filled or something of that kind. So selection of those inbreds is very, very important. I've seen in Symposium, some of the inbreds are as good as some of the hybrids. Just inbred per se, they are very good. So therefore, selection of material in the breeding program is very, very important, and the data recording. Of course, analysis and all that things, the modern techniques are now available. I am not familiar with many of those techniques because we are the old, gods, let me say. But as far as the fundamental aspects are concerned, those are very, very important. Then also in the in relation to private public partnership, when we come to a plant breeder, now people go as a plant breeder and they look for a job in the sense either in the public sector or in the private sector. A point was raised here that there should be a kind of internship, and I would again make an appeal from this forum that from the agriculture universities, institutions, if a program is built in the program itself, that some of the students, those who are willing, or even all of them, as it is in case of other business courses, where people are attached with a particular industry or particular organization for a month or so to get the some kind of a training.
Yeah.
So similarly here, I would suggest that the persons who are at the MSc level, postgraduate level or PhD level, if they are associated with a organization, multinational organization, seed companies who are really looking for you, then that kind of expertise, I mean, you will, for a month you can gain a lot, and then you can deliver when you go to those organizations. Then another point I would like to make, it's not necessary that all of you should go to a industry. You should have the courage to take the risk, what we call risk taking ability. You must develop this thing in yourself, and when you go out, you are strong enough in your background, in your expertise now, with all the teaching which you have got, the experience you have got with organizations like some of the seed industries, you can open your own seed industry. Nobody can stop. I mean, you can do this thing, and you can really generate lot of job for others also in your own industry. And this is a thing which people should think. I have seen in other area, in IT sector, the boys are going and opening their own e-marketing, and they buy and sell on the just computer, and doing business of crores in Bangalore and other places in agricultural sector. I mean, they buy and sell. I mean, so this is the kind of thing. So you have all the expertise, and you can go and really do this thing. With those are some of the comments. Maybe I can come back.
Excellent, sir. Thank you very much. I think those are very important points. Building your entrepreneurial skills. That's a very important point that he made. So, do we request the chairman now, or is there anyone else who would like to speak up? Oh, sir, please go ahead.
Yeah. This has been a good learning for me itself, because
Sir, that way the camera is there, so you can. It's being webcasted.
That's not so important. The content of what I say, maybe if it has relevance, photograph is there or not. But anyway, thank you very much. And I never taught plant breeding in the division of genetics, so I did not really know much about plant breeding. What all I knew was that plant breeding means producing varieties. You don't have to know Mendel's laws for that. And that was the situation with our most famous plant breeders like V.S. Mathur, Ganga Prasad Rao, and so on and so forth. Then there was another to which Dr. Arunachalam, R.V. Mehra, and there was one more who belonged to the school of quantitative genetics. That was one area which I understood. The reason is that I did not know what purpose really all that analysis serves. And right from the beginning, I had a feeling that this is most probably a meaningless exercise, because it cannot lead you to a definite designing of a cross, identification of the parental materials, and selection procedure. The weakness I thought in the quantitative genetics was that all their studies, particularly the leading to PhD or MSc thesis, were based on a sample of 5 plants. and my understanding was that plant breeding is actually swimming like swimming in ocean, where you want to catch the most valuable material in a huge area, and then on the basis of 5 plants you want to make recommendations. So that was something which was incomprehensible to me then and even today. But as I saw in the discussions for 2 days, I found some people have made some sense out of that, I suppose, and they are able to make good selections. From a common sense point of view, I knew only that if you have to make selection, as you rightly pointed out, you have to go look at the plants one by one and then make selections according to your choice. Not because you asked, some class to go and put labels on a plant which look like this and look like that, and then I will see what happens later on. So that's the way plant breeding is done in the public sector institutions. I'm sure the picture is very different in the private sector, where the companies are very large, they have invested millions and millions, and they cannot afford any lapses. And ultimately they see the performance of an individual or a group on the basis of output, what product they give, and how much income their product generated for the company, because they have invested money and they work only for profits. So every small step there has to be looked profit earning. That's why all these diagrams and figures and principles you have shown, they are actually not new to anybody, any one of us. We know all of that. Ultimately, the point is that who will said that the ultimate work. You have to do really hard work. I have seen V.S. Mathur personally working in the field. He was a very unique character. Many of you have seen him for that long period. I was very friendly with him. So I knew some of his personal habits, the language he used, and he was once appointed as a microbial geneticist. When I joined IARI, he was appointed a microbial geneticist, and he did not know how to pronounce Escherichia coli. And he used to abuse this Escherichia coli because he could not pronounce it properly. But then as a workman, whenever he will go and talk to Dr. Swaminathan or Dr. Jain anywhere, he will pull out some wheat grains and show it to him that this is what I have seen and this is something. This was remarkable, and this impressed everybody else. And he was a near zero in genetics, naturally. He openly used to say, "Genes do not make your gunny bags. It is the grain which fills it." And he used to go to the field in the evening when the institute was closed and work there for at least about 2 hours or so and note down something in pencil. Then in the evening, he used to sit with a glass of whiskey. There, that pencil notings were converted into ink writing in his permanent diary. So that's how he worked, and this was his daily routine. Not that casually we just go there once a week or twice a week and then say that yes, we are doing a lot of work. So ultimately it is, I think there are two major steps in plant breeding. And if you have perfected, if you have mastered in those steps, I think you are going to be definitely a good plant breeder. The first step is identification of the parental materials. Usually it has been the practice that you make a cross between two good varieties and you will always end up with a good variety. You may end up with an equally good variety, but not necessarily a better variety, not significantly better variety. Somehow I attribute this tendency to the fact that over 45 years or 50 years of green revolution, the yield levels have not increased. Mind you, green revolution was created when the yield levels were increased by 2.5 to 3 times. In wheat, if we take 15 quintals, was the usual yield in those days, 50s and early 60s. And then all of a sudden it became around 50, almost 3. And at the same time, the hybrid programs in maize, in sorghum and bajra. So all these things combined together provided a great push. But after today, almost at the same round figure of 50 quintals. There are varieties. This does not mean that breeding has been a failure in all these periods. Developed. I can name some of those in wheat itself, although wheat was not my personal crop of choice or working. But there have been a sort of amendments in certain properties. And that has allowed to replace the by the new ones, and we have been able to sustain at least that level. But if it was possible to increase the yield levels in 1960 from 15 quintal to 50 quintal, from 50 quintal we have not been able to make it even 60 quintal, totally speaking. Which is in wheat, which is in cultivation, that is called Ganga Jamna or whatever name. It is Dr. Prabhu's variety. He is involved in that. So that is the most successful variety. But personal experience from the field, because I grow seed for a National Seed Corporation, etc. in my own village, and I know what's happening there. People are not very much interested in having this variety there, although this variety has created miracles in Punjab, Haryana and Western Uttar Pradesh. The reason is these areas are saturated with irrigation. They have assured water everywhere all the time, round the year. I talk of eastern parts of Rajasthan, irrigation is not assured. There are no canals there. Tube wells not working. Shallow tube wells have all failed. Now to go up to the height of depth of about 200 feet at least, minimum. Or some people go even up to 700 feet to make sure that the water will never dry up. So under those conditions where you have limited water, the variety like 2967 is not very much favored because they do not have that much water. And without that water, even a good variety will not give you the kind of results that you will get otherwise. So having a costly seed, putting a lot of fertilizer as required and no water or less water. If you provide in this variety, for instance, you provide enough fertilizers and minimum 6 irrigations, 6, not 5. With 6 irrigations, about 50 quintals of yield is guaranteed. That's my experience of 3-4 years. But everybody doesn't have that much water. I have a tube well, so I can ensure that much, and it is electrically operated. So the cost of irrigation is also minimized to a very, very small extent. These are the kind of things where you have to work out. So first step is selecting the donor parents, and the second one is the selection stage, whether you can really pick up superior individual plants using helicopters or those drones and all those kind of things, and then picking up individual plants in a 10-acre field. I don't know, I have no experience. You people can tell it better how this will be done. But if that kind of, at that scale, if you can work, raise your F2 population in 10 acres and select even if 15 plants which are really remarkable, you will always be successful. There is no question of being facing a failure in those cases. Besides that, everything else is almost a routine which plant breeders do it even otherwise, and all of you are doing maybe a little better because you are in a private industry, private enterprise. We are in public institutions. We have a lot of liberties. You don't have those kind of liberties which the government servants have. So we can work at leisure. You don't have leisure. So with that,
thank you very much, sir. Thank you. Thanks. Yeah.
Thank you. Now coming to the topic. Trust me, all of you who are doing research are industry ready. Anyone who is able to work in a team and is able to recognize the performance or ability of a team member on a particular job and trust him or her on that particular job, even a trait measurement or whatever, and then you do your job of your expertise and put that together and then come out with the next step logically, is industry ready. There is a myth that you are not industry ready and you are only public sector ready. There is no difference in the way you function, the way you do things, other than pure academics, like the teaching part, where you, like Dr. P.K. Gupta, for example. He is excellent in teaching, but I would not have the same confidence. I'm just candidly telling so that we understand what I am saying. I would not be having that faith that given a topic of breeding for resistance to a disease or breeding for improving drought tolerance in a variety, he would be able to do it with that kind of intensity with which he teaches and writes books. Same with Professor B.D. Singh. So that's the point. If you are in research, you have a problem at your head and then you are doing everything possible as dictated or at times say restricted by your chairperson. You are still doing something in the field, taking your decisions on that material at that point of time, at that spot. If you're doing it and committing a mistake and able to realize it's a mistake and do the correction and then get back to the line as you're supposed to follow, you are there for industry. Raman was a student here. He did excellently well as an ARS scientist. He did equally well in public, say, international public system, where there are others, the work culture is different and the targeting is different. And here he is in industry with so much of confidence that exudes out of him from the same system from which he grew, as a system where there was liberty to do nothing, as Chairperson just now said. In an industry you have liberty to do, you have no free liberty. In a public system you have liberty to do nothing also. With that system, to the other system, we see him functioning very well. And many of us, his colleague, his earlier boss, Prasanna, same thing. It's all like there's no question of none of you being ready for seed industry if you are doing research of any kind in plant breeding. For example, in this particular sense, plant breeding. The basic things you need to know is the logic of step 1 to step 2, whether it is understanding probability principles of what you get when you do an event, when it comes out with the possible classes, either it is exclusive or whatever, independent, whatever. The point is, if you know that you are doing something and the expected result out of it is this, is it coming out the same way or not? If it is not coming out, is it because of an error or it's expected variation that's going to happen because of the material on which you have done that experiment? Is all that you need to know. And when it comes to analysis, if you understand the basic analysis, setting hypothesis, and basic analysis of how do you partition the variation in simple ANOVA, and when it comes to identifying from the different sources of variation, different categories of sources, how do you orthogonally partition it, how do you use the replications, and how replications become samples and how samples become treatments. If you understand those distinctions in variable groups of analysis, that's what all this BLUP or any of these analysis are. There is nothing different. QTL cartographer, for example, for that matter, or QTL analysis for that matter, it's all just translating the qualitative data of say non-parametric nature into parameterized information you are collecting on the phenotype. It is what some people have even called it Mendelizing and those kinds of things. So it is just understanding the principles of how variation happens from parent to the offspring or crosses to the progeny, and how you track it down without causing any error in terms of cross-pollination where it should not, or self-pollination where it should not have. Things of this kind. If that is under control, you as a plant breeder have to understand that part. So it is that only, and rest of the things are just by learning, just by following the routines as they are supposed to be, and you are there. And I'll just end up by quoting an example, not quoting, narrating an example of one of my own students, Anupam Singh. He worked with me for about 12 years. He has his limitation. I'm just candidly telling, using as an example. I took the name
only to ensure or bring into you the confidence that I am telling the fact. He would not make a good teacher. He was attempting to get into, say, universities where he would have to teach. I knew it would be not a good option for him. He would do teaching and he would also get promoted to professor, all right, that's a different thing. But then successful when he would go back home at the end of the day, having achieved something, that would not happen if you are just a teacher. I knew that. So I tried my best to get him into an industry. Somehow the time when he was looking for a job was the weak time in the industry because of the problems we are having in the government or in the industry setup. There is some problem in industry setup, a lot of layoffs are happening, and that's why good companies were not hiring at that point of time. But still patience worked, and now he's in a very good company, and already he is on one rung up within 6 months of his joining it, because I knew he had everything that would make him industry ready, because you gave him a job, he would focus on it, he would not, even if he has not done it before, he would go and learn from the people, because in IARI we have that facility to get anybody, anyone who is an expert in something or the other. So he would go to that person, understand what is to be done, and go back and do it. And whenever he is in trouble, he would call me and tell me this is where it is. What can I do for it? So I would then again tell him to go to somebody else, and he would do that. So somehow the job is done. So that is what is required in an industry. You have a target, you have a timeline, and you have fund to get to that target, and flexibility how to operate with it. So you have all that thing, and therefore if that is given to anyone among you, you will all do well. So there is many of you, when the question was asked about how would you analyze 100 say lines of hybrids in an experimental design, each of you should have raised your hand. Professor Pentel said in a matter of say conjecture that all of you raised your hand, then he would not be able to find out who is the one who actually knows or who doesn't know. But in reality, all of you would know. It is not necessary that you have to do a blip analysis. You don't have to, you may not even have a random or fixed, you didn't say anything about it. Whether the lines which were used for the hybrid were random lines or they were fixed lines which had come from one set of sample population. Depending on that, you have to decide. But you should know how would you, if it is fixed, what would happen, if it is random, what would happen, and how do you classify. Things of this kind you are taught. You have to say yes, I would do it, then ask questions. They tell me this. Then you get into this. So because you didn't have this in your mind that he would possibly ask you, then show it. So therefore many of you did not raise, I suppose. But if you knew that you had the freedom, your job is to analyze, and you have been given that task, you would naturally come and ask him, please tell me where this 100 hybrids came from. What was this, whether they are from same maturity group, or things of this kind. You would start asking questions, then one by one, logically things will fit in. So don't hesitate. Your basic foundations in these simple probability theory, inheritance laws, and linkages, how they behave, how environment is able to interact with quantitative variation, and how do you distinguish between qualitative and quantitative variation, is all that is required for entire phenomena to function and unfold. Thank you so much.
Thank you very much, sir. Thank you. Thank you. May I request the chairman for his comments?
A lot of time has been taken, and I think we find the inaugural and the valedictories to be the most interesting sessions in which the population size is the largest. I have many points to say, but I am not going to go into all this because the time is very short.
Sir, we are cancelling the next session, so we have a little bit of time.
In any way, I don't want to fill these young people with a sense of gloom. I personally am facing a lot of gloom. After about 30 years work on mustard breeding, I am being told so many things. This is what you guys didn't do. This is the way to approach. Pure lines can be better than hybrids. Marker assisted breeding is a wastage of time and money. I mean, all kind of advice. So I am going to desist from giving you any advice. Only one or two points I want to make. One is that in agriculture policy, we are damn confused in this country at the moment. You heard what Pioneer is doing. But when they say industry, do you know what many other small industries are doing who have very little access to germplasm, who are basically running on the basis of marketing rather than any research work in-house? They don't have the resources, they don't have the ability to compete with multinationals. The public sector is very important. It was very interesting to listen to two talks in the morning. I missed, unfortunately, Ashok's talk and Dr. Gupta's talk. But the two talks on maize, one improving the nutritional quality in the public domain, the other one on how genomic selection is being practiced and how it is going to be useful for predictions for areas in which we want to take forward maize production or penicillium production and so on. Somehow I believe the fault is, do these two have any common ground? Are they interacting? Should we give material of public sectors free to the companies? To how many companies we should give? It's a very strange situation. Globally, there is so much consolidation. Monsanto, Bayer are coming together. China buys up one of the largest companies, Syngenta, and DuPont and BASF are also, I think, in some kind of dialogue to come together.
DuPont and Dow.
This has happened or not. So on one hand you see such tremendous consolidation. On the other hand, in India we have hundreds of countries, and Professor Saab has said you can also be an entrepreneur and start your own company. It's a very confusing situation. Now there is certainly, I am very clear that even though prophets of doom, and I don't want to name them, they say that plant breeding is not even required. The many health-related scientists, Pushpa Bhargav is one of them. He says insulin, recombinant insulin is very good because it saves lives. And you can recall it. But anything you put in a crop, you have put an atom bomb there in the crop, which will burst on any day on the horizon and kill all of you. So, if you feel confused, don't worry about it. I at this age am feeling very confused where our policy is going and what are our attitudes and what are our opinions. Now as students I understand, we like dabbu students. We don't like aggressive students. You guys have become little aggressive by working abroad. Or maybe IARI encourages some aggressiveness. Mostly we want you to be, ये टीचर हैं, आप पढ़ने वाले हैं, जो पढ़ा रहे हैं वो बड़ा important है. I had a hellish job in asking Delhi University teachers to review and revise their curriculum for botany course. कहते जी ट्रिडोफाइटा important नहीं है. आप कह रहे हैं molecular biology पढ़ाओ. Triidophyta is as important. Bryophytes are very important. Now, how do you, with these kind of tremendous advances in science, how do you accommodate everything? You will have to leave something out. And one thing I believe, you believe me, you cannot leave out is quantitative skills. Why people didn't understand Mendel, he died without being understood. Because most of the biologists were descriptive type. Even Darwin was a great description master. He described very accurately. But he was into description. He was not into any quantifying anything. Mendel was into quantification. And nobody understood him. So we have to improve the mathematical skills and computational skills of our agriculture students. Why can't we have, the second point, why we are saying the curricula are not good. One is our inertia. जो मैं पढ़ा रहा हूँ वो ठीक है, मुझे इतना ही आता है, मैं इससे ज़्यादा क्यों कोशिश करूँ. The second thing is 103 institutes in this country in agriculture, 56 universities. Maybe their number has increased since I found out last. In these institutes, which are supposed to be the best funded by the central government, there is no teaching. These guys are so busy in research, they have no time to teach. And in the teaching institutions, you have no money, so the teachers are doing no research. So where is the incentive to bring new things on the table? If I am doing no research, and I am asked to teach what he is propagating as genomic selection, I will have to first learn, but I have no incentive to learn it, because I am not practicing it. So there is a deep crisis which nobody is understanding. For political expediency, every minister who comes opens up 3-4 new institutions. Most of the money goes into brick and mortar. And we all know that the best learning is by doing. And this is what we are talking about. Do you know this? Have you taken field observations? Have you run a few gels? In my university, there was a big debate. Should recombinant DNA techniques be taught in the first year or in the 4th year or 3rd year? People who are not comfortable with this would say 3rd year. They should first learn the basics, rote. छोटा मारो कि restriction enzymes कैसे काम करते हैं, क्या होते हैं. Then you do the practical. Why can't they be done together side by side? So I think we need to improve whatever I have learned, and I reinforced in my mind, and I tell my students also, we don't have good quantitative skills. And that's why we have not been able to do as much as probably you will be asked to do in future. If you learn genetics by problem solving, you will be a much better geneticist than by rote. कि मेंडल के 2 law थे या 3 law थे. So we have to improve our curricula. I'm glad you are going around telling people what are the requirements for the future. We had great breakthroughs at the time of Green Revolution, and we are very mythology oriented people. So we believe we want miracles. 10-20% increase in yield हमें पसंद ही नहीं आता. We want the same jump as dwarf wheat or dwarf rice gave us. I call it the greatest jackpot that humankind hit in the last century, the dwarf genes. They had wonderful pleiotropic effect. They took us away from starvation. Pushpa Bhargav writes that they brought water hyacinth to this country. This is how negative atmosphere we are dealing with vis-a-vis agricultural research. And agriculture students and researchers have to stand up. We have to say what we learned is not enough. We have to learn more. We have to improve quantitative skills. We have to bring out the best researchers to teach and give little bit more to those who teach in terms of research so they remain up to date. And in fact, only one point where I disagree with you is don't ask these people to learn marketing. You hire so many MBAs running around to do your marketing. Some people may have flair for, I mean, if you look at the ICR system, very few people want to work on the bench. Everybody wants to make others work. Say, मेरा idea है, ये काम कर रहा है. So there are many people who are interested in running others' lives rather than being creative themselves. But as plant breeding students, please learn your subject. You will have to learn molecular biology. You will have to learn some biochemistry. You will have to learn some quantitative genetics. So you have many things to learn to become a good plant breeder for the coming times, where incremental increases will occur, not big jumps. Somehow, somewhere, if you are searching, you can hit a jackpot also, like dwarf genes. But in the last 40-50 years, that kind of jump has not come to us. So good luck. You are very important people, because this country requires plant breeders, agriculture scientists. For sure, let nobody discourage you that you are doing something useless. You are in a good field, which is going to be very important for this country. Today our population is 1.2 billion. our population is going to increase to 1.6 billion before it starts plateauing out. Who is going to feed all these people? It's only through better production, what I call as low input, high output agriculture. And I would invite you guys to interact with universities in teaching. Send your best guy who does genomic selections to teach a course for 4 or 5 days. Rather than a conference, come here and interact with the students. And I hope Dr. Prabhu, as the research dean, will invite you guys, the best people, to come and teach the students. This is the talent we have. You make anything out of them. You can make good people out of them, or we can say, ये कर लो, वो कर लो, तुम्हारे रिसर्च करने का फायदा क्या है, IAS में चले जाओ, बैंक क्लर्क बन जाओ. You know, we can do that, or we can encourage them to do better and better. Thank you for giving me a chance.
Thank you very much, sir. I think that was excellently summarized, and I'm sure you all would have got the point. So quantitative skills are very, very essential if you have to succeed. I once made the mistake of asking an animal breeder, what is the use of quantitative genetics? So he asked me back, if I give you 10 bulls, can you evaluate those bulls for their milk yield? So I was wondering, how can I evaluate the bulls for their milk yield? You can still do it. And that is the advantage of blobs. So I am sure you will go back and read. And we have Professor R.B. Singh here already, so for the valedictory section. So we are going to go ahead with our valedictory session next. And thank you very much once again, all the panel members and all the students. And okay, so sorry. Please go ahead. Breaking tea. Okay, so we are going to break for tea now, and then we will come back in 15 minutes. We are going to come back. Thank you.
बाहर बाहर और invite all the people inside. मुकेश सबको अंदर बुला लो, अंदर बुला लो बाहर से.
Good evening. I would like to request all the respected dignitaries to kindly grace the dais. Please, Professor R.B. Singh, sir, Dr. Sir.
Invite one by one. I don't worry. The group coming, you say.
I welcome you all for the valedictory program of 2 days Student Symposium on Genomics-based Next Generation Crop Improvement Approaches. We are very fortunate that the Professor R.B. Singh, Chancellor, Central Agricultural University, Imphal, Manipur, is here. He is here with us to chair the session. So he is a former chairman of ASRB, member of Farmer Commission, ADG FAO, former director of IARI, professor and head of genetics and plant breeding, Banaras Hindu University, Varanasi. So now I would like to request Dr. K.V. Prabhu, Joint Director Research, IARI, to kindly present brief report about the symposium, please.
Professor R.B. Singh, one of my first introducer to the science of genetics and my dean in my undergraduate period during 1975-1979 in Banaras Hindu University. My friend Dr. A.K. Singh, Dr. Anand and Dr. Abhinav. We have had 2 days of this workshop, which is a very novel kind of workshop where students are the major takers. It's not the faculty-faculty interaction, it is faculty-student interaction more, and student-student interaction also. It happened with a very good beginning by the person who has almost worked for 75 years in the science of genetics, Professor Swaminathan. So, and you are there, sir, for 50 years plus. So it is excellent organization from the introductory to the valedictory, where we have people who have contributed to the system so much in the science of genetics in one way or the other. So apart from that part, I would like to tell you, sir, that the introductory went off very well when Professor Swaminathan gave a very good lecture in his own inimitable style on food for all in the Anthropocene era, where he explained how we have living in a man-made environment, evolving because of the human interventions more than natural evolution. So the Anthropocene era, and how we are making it more difficult for enabling the food and nutritional security to all, and what we have to do in terms of genetics to provide the crop improvement part that gives the initial beginning for food security. The next session was one of the more exciting one, next two sessions actually, the one with the lunch that in between. One was when Dr. B.D. Singh explained through his academic understanding of the science of genetics, right from Mendel to the plant breeders' final outputs, how the two are linked. Unlike what Dr. Balaram Sharma was telling just now, that best plant breeders are not necessarily good geneticists, who not understand genetics, need not necessarily understand genetics. So Professor B.D. Singh connected the two very well. Then Raman, who is one of the coordinators here from DuPont, got into the new technology side and how the plant breeding tools have changed, and whether the sophistication is needless or it is a necessity of the current day in order to make it more precise, more efficient, and more resource economic. Then was the real technology session when one talked about deploying the molecular breeding option, which Emerson gave, and then Justin came with the sequence based breeding and the limitations where it can stop, and application where it can give you better outputs. Then was the two practical things which came up with Anand explaining the precision phenotyping and next generation, the next crop breeding frontier, how precision is as crucial in phenotyping as much as in the genotyping and the post genome sequence based information analysis in terms of informatics. Then was a very good, excellent, connected, experienced talk by Amitabh. Amitabh only gave that lecture on genome editing, where he connected it to cut and paste. He's there, Amitabh Mohanty is there. Yeah, he gave it and explained the genome editing, and now I understand on the demand, which is very encouraging from the students. They're going to have a workshop with I think genome editing as well as bluff analysis, both. Very good. After the valediction, as an exception. So very good for any academic intervention. No formalities have to be followed. This valedictory is over, so end of the show nahi hai. So we can have a brief workshop after this. After that, we had Dindo, who spoke about double haploids, which is something very important for India. We are now trying to do that in IARI, and we hope what was exposed by that lecture would help students to understand the importance of it, and maybe that drive will make us facilitate it. We'll do that. So that's what was done. And then there was an excellent series of posters. There were fantastic posters set up in the new lobby of the genetics division, brightly lit, and the posters' content were more brighter. So it was very good posters, and congratulations to all the students who did that contribution for the posters, and some of you will be acknowledged for the excellence done today at the end of the day. Then the application genomics part, which came today with Professor P.K. Gupta, giving a total overview from Mendelian genetics to the advancements that have been happening, with all new acronyms and antonyms, and then everything that can happen in the name of molecular techniques and how they have been used, or more candidly, how not being used in India, unlike other countries. He mentioned it in many cases where he expected India to do it. So that is for the students to understand that we need to put that kind of inputs in our research. Then Ashok gave the contrasting but fantastic, realistic use of molecular tools in turning into products. Evidently, through the 11 or 12 different varieties he has in his group has put forward, and how they work towards it, how they overcame the pitfalls using molecular markers, and then successfully were able to target and also achieve the targets that he gave using basmati breeding as an example. Then Firoz Hussain gave an equally exciting say display of how over the last 6 years they have been bringing in the quality improvement in maize, and how they are using the tools, especially the ones which are based on pathway genomics, and then accordingly controlling the expression of different traits and putting them together, which he did very well. Then Mahala, the one who gave a talk which sounds really not academic when he said commercial plant breeding. The topic was about how is a success defined in commercial plant breeding. So at the end of the talk, we had a long discussion on the point that there is nothing commercial in plant breeding. Plant breeding itself is commercial, and there is nothing like commercial plant breeding. And the marketing was being confused to commercial plant breeding, which is what the chairman of last session also mentioned apparently. But what Dr. Mahala was trying to explain was how best we can use the resources and see that the use of resources are properly utilized in turning out the targeted products without making the company lose money and making the company earn that money, and see that the science is not at any time compromised in doing that. That's how we put it, and it was a very good, maybe the name commercial plant breeding, like commercial heterosis, is the same thing as commercial. We teach commercial heterosis when we were teaching heterosis breeding, so it is the same way he put it in commercial plant breeding. So that is a very good one in terms of practical exposure to what happens in seed industry or in industry which has R&D, and improvement happens in that. Then came the next session where we had students direct interacting with us. Although the interaction was not really that much as one would expect, nevertheless it was there when we panelists were sitting there talking about, are the students seed industry ready, or at least industry ready? The genetic students, are they industry ready was the point, and many of them being exposed for the first time, and maybe some intimidated by some of the presence of their own teachers here, didn't raise their hands when some simple questions were asked, isn't it? That was one of the reasons, maybe. Otherwise, the first question could have been answered by all of you. Many of you did not. Second question one would understand, because it depends on the facility available in the research area where you are working, in the university where you are working. But the first question could have been answered by all of you, which I know you will all answer that. And Raman Babu was also in the same place as you are today now, and he was asking this question with us. So have that confidence, don't lose it at any point of time. As Raman also said, there is nothing which is silly. Every doubt is not small doubt or big doubt. A doubt is doubt, and it can come even to Einstein. So there is no problem about asking a question when you want to ask. That's what I tried to explain when Anupam, I said, was best suited for industry, because he would ask question without hesitation. And if you ask question and you know the logics in asking the question, you will get the answer and you can follow to solve the problem which you are questioning about. So that's what happened. It was a very good one, sir. So interestingly, this was webcast. The 2 days were webcast, or is being webcast even now. And then we had interesting questions being asked by people all around, from Dharwad, from Angrao, from Tirupati, from Telangana. We had questions coming from BHU, from Pusa. Questions are being asked, so which is good. So that is very encouraging, and that's what has to happen. And they had no faces to possibly face, therefore they were asking questions very directly, some very simple questions, but they were asking questions. So that's what happened, and it was very encouraging. And I must, with this point, thank DuPont for thinking of this, and also arranging for the webcasting, which Ashok did very well. So it was very ingenious to have virtual attendance of must be, this is about 100 crowd, that must be more than at least 500 to 600 students who are in research or genetics in their career, or at least in their mind, sitting and listening to many of us. Thank you very much, sir. This is how it was, and I just want to end up with the point that I enjoyed it, although I could not be present the whole day yesterday, but wherever I was, whatever I listened to, I enjoyed it. It was great, and now that we are in next generation genomics, the generation will advance, and all of you will get into it, and with more funding and more facilitation now coming from organizations like DuPont, we will be better off. Thank you very much, sir.
Thank you, sir, for briefing the symposium. Now I would like to call upon students to give their remarks. So first I would like to call Mr. Navdeep Singh Jamwal, PhD scholar from CSK HPKV Palampur, followed by Mr. Vidya Sagar, PhD scholar from IARI, New Delhi.
Very good evening to all of you presented on dais and my fellow mates and teachers. So I am Navdeep Singh Jamwal and I am working on wheat molecular cytogenetics. So as a student, we all know that we always stuck to our own subject, our own field, and most of time we are unaware of what is happening around the world. So this kind of symposiums, this gave us opportunity to learn the things. And I must say that this symposium was different in terms of that we met the father as Dr. Swaminathan, and the terms we have started our science with the Dr. P.K. Gupta's books, and as agriculture graduates, Dr. B.D. Singh. So it was a complete package for us, starting with the views or vision of a father. How a father thinks means he always thinks about a whole mankind, and then Dr. P.K. Gupta knowledge and theoretical knowledge of Dr. B.D. Singh, as in his books and as well as in his lecture we have seen this. And I must say that we are not judged in this symposium. Like everything starts with the fundamental of starting with the Mendel, and then gradually move to the advancements, molecular biology, CRISPR, the whole thing, double haploidy things, which starting with the initial and the movement up to the application part. Today it was done by Dr. A.K. Singh, and Dr. P.K. Gupta also teaches us that. And further the panel discussion was quite good for us. It's more entertaining. And I want to congratulate the organizer IARI. It was my first formal visit to IARI, and I must say that it was a very nice, and I'm overwhelmed with your hospitality, and I will say big hand to all of you. And I want to congratulate our sponsors which have organized this event, and they have sponsored our visits also. So, and finally, I want to pay a tribute to G.J. Mendel. Because of his science, we all are under one umbrella today. And this auditorium, B.P.Pal, because this auditorium has special, because similar kind of auditorium is in PAU, and I was graduated from there. I'm alumni of PAU. So same auditorium I have entered in PAU and become a graduate master, and now I am doing PhD, and again under the same name. So it's my tribute to Dr. B.P.Pal and Mendel. Thank you all of you for your listening. Thank you.
Good evening all, especially sitting on the dais, my teachers, friends, students present over here for the 2 days symposium on next generation genomics based next generation crop improvement approaches. First of all, I would like to say thanks to the DuPont Pioneer and Division of Genetics for inviting them to IARI and for sponsoring this symposium on genomics based crop improvement approaches. Several part of session during these 2 days impart us with great wealth of knowledge regarding tools and practices in genomics and post-genomic era through high-put genotyping and phenotyping facilities. Plant breeding and molecular technology are taught in the classes. However, this symposium has given opportunity to us to learn from the people who are actually practicing these techniques in the field and producing a commercial product. We also interacted with Professor Swaminathan, Dr. B.D. Singh, Dr. P.K. Gupta, whose books are read by every student sitting here as a plant breeder, as a molecular biologist, as a cytogenetist. So it was a great opportunity for us. The lecture given by Dr. Raman Babu, Emerson, Justin, Anand, and to name all, some of them are very mesmerizing, and it was to take away some message from those lectures. The way you have heterotic pooling and development of hybrid, which are covering more than 30 million hectares of area, is remarkable, sir. The phenotyping facility you people utilize are way ahead than what we practice at our institute. Like you are using drone technology, high velocity wind machine, etc. It feels very good to us to see the application of CRISPR technology that has shifted from model organism to commercial crop, and you have developed a variety of vaxicorn with this technology. Although several queries were made from students and faculties just to gain insight into how you people proceed for development of your products, somehow research personnel from your side didn't responded to the point. Maybe you have some restriction from your sides. It will be very helpful if students will get all the slides in the PDF form or PowerPoint presentation by the scientists who have presented in the symposia. It will be very helpful to search all over the references and get insight again into the contents of this symposium. I would also like to ask one thing, sir. Does there any training facility available at DuPont Pioneer, especially regarding data analysis? Because we are generating data, but we don't have that technical skill for analysis of data. So if you provide us that training facility, we will especially like to visit your facility for that, as we are knowledge seeker and we would like to get some from your side, sir. At the last, after listening all the discussion, I would like to say that in spite of immense technological advancement, the breeder's 2 eye, left and right eye, are still the best marker used for plant breeding and selection of commercial plant. Thank you, sir.
Thank you, friends, for giving your feedback. Now I would like to request Dr. Anand Pandravada to brief about poster session and to announce the most awaited best poster awards, please.
Thank you so much. The only other comment I have is, the breeder sometimes can develop long sight or short sight, so he may need eyeglasses, and your markers are the eyeglasses that he will need. So, it was great seeing all the students, and I can relate back to the first student symposium we did in PAU, where Dr. Kuldeep Singh and Dr. Dhillon very much supported it, and then at PJT-SAU, where Dr. Praveen Rao, the current Vice Chancellor, was fully supportive of it, and now here where Dr. A.K. Singh and Dr. Prabhu were fully supportive of getting this visibility to a lot more people. So I appreciate this, and one of the one of the new things that happened here was the presence of a well organized poster session. And I wish we had a lot more time to look at some of the excellent work that these students have done, because as part of this, I think we have presented about 39 posters from IARI, 26 alone from the division of genetics, and remaining from all the other divisions put together. There were 22 posters coming from all of the students here who are representing the different universities that are sitting in this room, not to mention the students who are listening to this session through webcasting. Some of the students include students from BCKV and UBKV West Bengal, Assam Agriculture University, CPGS, then Haryana, Rajendra Prasad Agriculture University Pusa, Palampur, BAU, BHU, CCS, Meerut, Pantnagar, RAU, and then MPUAT. I'm sorry if I missed any of the universities, but the work that you guys have presented on the posters and the enthusiasm you have shown is sort of contagious. It just takes me back to the days where we were students, and also gets us new vigor that we should be even more energetic in the research that we do, what we are doing today. And the work ranged from everything from germplasm characterization to using molecular markers for disease tolerance, for abiotic stress tolerance, association analysis, expression studies. There was a couple of posters on Cas9 genome editing, and also plant microbe interactions, to name a few. So excellent range of topics, wide variety of crops, wheat, there are horticulture plants. I saw a poster that was talking about how to process seed of almonds. So it was completely diverse, but very, very unique and important projects that the students are working on. So congratulations to all of those students. And what I would do now is announce the 6 posters that the jury selected as the best, as were among the best. All of them were best, but these were the 6 that got selected. And I would request Professor R.B. Singh to hand over the posters to the certificates to the students that we call on. So the first one is the poster was on marker-aided incorporation of BLB, bacterial blight and blast resistant genes in elite rice variety Pusa Basmati 1509. And V. Sagar, the lead author, and there's a group of people, Gopal, Divedi, Dhawan, Amit, and belonging to all the way to Dr. Prabhu and AK Singh. So V. Sagar, if you're here, please come onto the dais to receive the certificate from Dr. R.B. Singh. Yeah. The next poster is on genome-wide association mapping for identification of QTLs for micronutrients and protein content in wheat. The lead author is J. Kumar, G. Saripalli, V. Gehloot, P.K. Sharma, and Dr. P.K. Gupta. Congratulations. The next poster is development of biofortified multinutrient maize using marker-assisted selection for nutrition security. This is by A. Das, Firoz Hussain, Vignesh, Rajkumar, Hema, Baveja, Saha, and H.S. Gupta. The next poster is the identification of white rust resistant gene in Brassica gentiana. This is by H. Arora, Pradhan, and Dr. Pental. The next poster is on the cytogenetic characterization molecular tagging of Triticum miltinae derived leaf rust resistance in bread wheat. This is by Nataraj V, Sharma JB and Vinod. So, and the last poster is on marker trait association for low light intensity tolerance in rice genotypes from eastern India. Again, a very good characterization effort. This is by S.S. Datta, G. Pale, Polong, Achon and A. Pandey. So, excellent set of work, guys, and for all of those people who were not called onto the stage, again, your work is no less important. It was beautiful. Keep asking questions, extend what you're doing, and try to do something that is beyond what is just there. And then I think that's where you will see the real fruit of your work. So, great work, everybody, and a lovely set of posters. Thank you.
Congratulations to all the winners. As a mark of respect and regards to all the eminent speakers in the symposium, we would like to present the mementos. Now I would like to call upon, Professor B.D. Singh sir was the first speaker. Now he has left, but we would like to acknowledge his talk. Now we have Dr. Raman Babu who gave the second speech. So kindly, I request Professor R.B. Singh sir to kindly give the memento to Dr. Raman Babu sir. Next, I would like to call upon Dr. Emerson Limburger from DuPont Pioneer. Next is Dr. Justin Gerke. Dr. Justin Gerke, please. From the session 2 on enabling technologies, we had speakers. Dr. Anand Pandravada. The next speaker is Dr. Amitabh Mohanty. Next is Dr. Dindo Tabano. On session 3, we had Professor P.K. Gupta. So we also would like to acknowledge his wonderful talk. Now we had Dr. A.K. Singh sir, who is also the organizer. Thank you. Next is Dr. Firez Hussain sir. Dr. R.S. Mahala. Next we have Dr. Deepak Pental, who chaired the Dr. R.M. Singh sir. Dr. K.V. Prabhu. Dr. Raman Babu sir. So thank you sir for giving away the mementos. Now I would like to request Dr. A.K. Singh sir to give the memento to Professor R.B. Singh sir. Thank you, sir. Now I would like to request Dr. Raman Babu sir and also Dr. A.K. Singh sir to present the vote of thanks, please. Sorry for that. Now I would like to request Professor R.B. Singh sir to give his chairman remarks, please. Sir.
A very good afternoon to each one of you. On my left and right, when I see the outstanding scientists, nothing will please me more than that. Dr. Vinod Prabhu, my student, Dr. A.K. Singh, another of my students, and of course, Raman, his wife was in my class. He was also in the same class. So Raman was in the same batch, in the same class. And of course, Anand, here you are. So you see how delighted am I that I am flanked by the outstanding people who have contributed and who are here today to share their experiences with each one of you. In my front, I see Rishi. Rishi was also my student. I taught him the first course in MSc in botany, genetics, and breeding. So, and Balram was my classmate, as a matter of fact. So you can imagine, and of course in the audience, Tara, here you are. I know you are in the same class, and that really makes me so happy to see that we are here together. And several of you who came out from that group of education system are leading one of the best private sector initiatives in the world, which has served the humanity with tremendous distinction. DuPont Pioneer, I had the privilege of being at the headquarters of DuPont Pioneer last year, and in advance, and Manjit Mishra, the director of the seed program, took me there, and I spent half a day, met your top people at the company, and I was so pleased to see that when you go to Iowa and you see DuPont headquarters, DuPont Pioneer headquarters, you start realizing that probably some of those systems are bigger than the state agriculture universities here, or the land grant university system like Iowa State University seems dwarfer in comparison to the setup and the systems that DuPont Pioneer has there. So I was so pleased really to see how much is the human resource for these various initiatives, and also your 65 platforms in 5 continents, and 2 of those, one was mentioned at Punjab Agriculture University, second one being held here, probably speak volumes for your devotion and dedication to the system. As a matter of fact, it gives you a kind of a nostalgic feeling in this room, particularly when I remember in 1965, when I was with the Punjab Agriculture University as the economic botanist cotton and associate professor there. In 65, I sat somewhere in this hall when the 100 years of Mendel centenary celebration was happening, right here in this hall. And do you know Dr. B.P.Pal, of course, Dr. Swaminathan, Dr. A.B. Joshi, all the distinguished Dr. S.K. Jain, you name it, all of them here with tremendous deal enthusiasm. And a very large number of people from outside, all over the world, whom in those days you must have been reading about classical genetics, population genetics, quantitative genetics, you name it. All of them, more or less, right from the leading, in those days, the leading cytogenesis. Professor Kihara was here, Munsing was there, and all you think of it, Gustafson was here. You think of those people, Dr. Robinson was there. Some of you, those who might be using the North Carolina design 1, 2, 3, and if you know the reciprocal recurrent selection, the father of reciprocal recurrent selection in quantitative genetics, Dr. H.F. Robinson was here in this hall. Dr. Cummings from, then he was the dean here at this institution. Can you believe it? So this is the kind of history, as someone said, father, son, son to son, and grandson. He mentioned it the way it is, and it's nostalgic feeling that you get. Now, what is more important, when you go through these various historical points, milestones, each one of those have a message for you. You very think of those very names, you think of the conditions under which Mendel discovered the principles and law of heredity, the fundamental principles of heredity, and those conditions not being himself trained for, but think of the innovative way of doing, the designing of the experiment, use of mathematics, counting and accounting for, and then keeping your discrete records, which made him really come up with the kind of things for which the whole world must be absolutely grateful to that really unparalleled monk and priest which the world ever had experienced. The humanity at large must have not benefited from any other discovery than the fundamental principles of heredity and genetics which Mendel was able to bring forth, which I think is absolutely, and therefore I congratulate both DuPont Pioneer and IARI and each one of you who are able to participate to count those milestones and travel through those posts. And I think it will be worthwhile, Vinod and Ashok, both of you, if you will be able to put through those milestones. In that context, maybe it's not that I am doing any publicity or doing any advertisement of anything. The National Academy of Agricultural Sciences has come up with a book, and that book is titled as 100 Years of Agricultural Sciences in India. And both of these gentlemen on my left and right are authors of the very first chapter, which deals with the crop improvement, about 100 pages, and all those landmarks in that book of about 600 pages out is indicated here, and I can see other colleagues here. Gopala is also one of the authors. So I thought it is so very important for us to make a mention that how those journeys, right from the beginning, when you think of Madsen, Muller, you think of Mather, all those people in the classical sense worked on after the rediscovery of Mendel's laws in 1900 onwards, which led to the foundation which has given you the various kinds of revolutions today. We have the green, yellow, white, blue revolution, all owe their origin to those Mendel's basic principles of genetics. World today is food secure simply because of those laws, and you can imagine how important you all are here because you are following to go through the path which possibly will bring further fortification of the efforts made by Mendel. The science, technology, policy, market, farmer, all these linkages, private sector, every one of you, those who link together to give the ambience where this technology will be able to operate and unfold their potential is so very important. Therefore, I admire the partnership which you are building today, which will take you further on in the process to congrue, to converge, to synergize, to make 1 plus 1 11, not only 2, which is the game which you are playing today, which I think is absolutely, and I give my best of the best wishes to each one of you to be on this. When I really look at teaching programs and also see where we are, and we look at the world today, had this not been really discovered, even today, if you feel, somewhere complacent and so on, you have no reason. Even today, 2 billion people, can you believe it, are undernourished. 1 billion, almost 800 to 900 million people are really hungry. Various kinds of hunger they suffer with. 160 million children, less than 5 years of age, are really stunted. A very large proportion of them are wasted. It's not a commodity. A child is not a commodity to be wasted. But in the human parlor, it is called wasted children, who are both stunted as well as underweight, and these two combinations make them really wasted children. Think of epigenetics. You are all students of genetics. If the epigenetic effect of perpetuation of that real handicap continues, we will breed stunted, will breed stunted. If that is true, are we prepared for that? Is this not that enough? Should we really look back what Mendel did and what he said, we be able to absorb some of those messages and come through that we don't allow this to happen. And do you know the largest share of these very underprivileged are the people who have been swimming in the back waters of distortion of their life, are the ones who 1/4 of those hungry and poor live in India. 40% of the stunted, wasted children of the world live in India. On one side we want tall India, on the other side we have stunted children. How can we combine the two? Child or the human resource is the greatest treasure and capital of any system. If that is stunted, the country gets stunted. And of course, I am saying that please break this unholy alliance. Only through the laws of genetics we can do it. Therefore, I will say that please, the various cutting-edge technologies which you are all pursuing today, I can see right from the classical genetics, as I mentioned to you, then the various cytogenetical developments, starting, if you start from Mendel to Mather to Muller to McClintock, you keep going like that, and then you come to Jacob and Manoz, and then in between you have the Watson and Crick, and now you have the genome editing and genomics, all the omics. You have reached this stage. If this is the journey that you have, I think you should be able to meet the destined, devoted goals of the world today, and zero hunger. A world free of hunger must be the objective. It should be possible. As you know, Brazil was able to do it in 10 years' time with President Lula at the single-minded approach, where genetics played also a very, very important role. The climate smart agriculture, the productivity, the resilience, the mitigation, the three components of developments of developing a climate smart agriculture in face of the shrinking land, water, biodiversity, resources, where the MLM, more from less for more, should be the equation. And if that equation is allowed to work and made to work through genetics, it is genetics which will make it happen. Nothing else with that proportion will make it happen. I will say, please stand for it. Inequity is the largest handicap in this country. A farmer is 1/4. The average income of a farmer in this country is 1/4 who is not a farmer. A farmer, non-farmer, the one who feeds us, the one who practices genetics, the one who converts the rules of genetics into food, are the one who are hungry. What a paradox. I think this paradox must be removed. The iniquity which is breeding depression should be allowed not to further multiply itself. So please look into this in this fast changing world. If we have to look into the possibilities of meeting the requirements of 2050 onward, these sustainable development goals are there, Paris Declaration is there, the population increase which was mentioned at, in the previous session. I heard Dr. Deepak Pental mentioning this will be the population growth for world and the India, and the doubling, almost doubling of food requirement from less and less water, biodiversity and land. In other words, science will have to work only. The efficiency, more from less for more, will come through the good science like Mendel's science and Mendel. So I would say that in this changing world, so many new things are to be done, and the political economy is changing. The trend is already trumped, and you know what is happening in the US, what will happen in the rest of the world, how things will shape in this oscillating world, in this fast changing world, how free, equitable access to technology, genetic resources, genomically edited products would be available. This is all a question which the private sector would be asking, the public sector will be interacting, and what kind of political economy will come in the face. You all students, who are students today, we have the largest bulge of young people of the world here in this country, and that is our treasure. We look at you as our treasure, and if this treasure has to really make a dent for India tomorrow, then look at the total political economy, the world economy. You live in world changing, and world grant is your grant, not land grant university system. You live in a world grant university system. You live in a world grant institution system, and therefore think beyond your SAUs and also think beyond your province. It is global, local are no more poles apart. Global is local, local can be global, and you have to look into the totality. So become partners, work together so that it is one collective voice which touches the humanity. If science has no human face, then obviously that science is non-productive, is not useful. So science for human face has to have these changes and the new changes which must come into the picture. How many of you, if I have another opportunity to teach my first course in genetics and plant breeding, I will again go back to Mendel laws. I will go back to how the polygenes, how the pleiotropy, how the multiple alleles, how the allele discoveries in those days can be linked with principles of genetics discovered by Mendel. Connect your best of so-called allele mining, polymorphism, polymorphic arrays, you always put so many of those. You go back, look at what is polymorphism defined by Dobzhansky. What did he mean by Muller? How did they define? If those classical genetics have to be taught in the realm of Mendel's discovery, then connect the genome editing today, the pleiotropism, polymorphism today, the polygeny today with the classical genetics of Mendel. Can you do that? This is a challenge for my teachers as well as for my students to work together. So please you understand, and there is connectivity of those basic, fundamental, real life sciences. Life is life. The code is code, it's written in DNA. How you are able to really manage it, handle through the process is absolutely interconnected. If you know this connectivity, you will commit least mistake, and then if you do the work in your field, in your lab, you will run to the field. Somebody said you have left and right eye, use the eyes, ball rolling also, as Borlaug said, you go close to the plants, they will whisper with you, they will listen to you, you can listen to them. Now those carry your DNA profiles, carry all your mappings, carry all your markers to the field. Don't remain yourself only with the internet, only with the bioinformatics, only with the kind of test tubes and those beautiful designs and so on and so forth. Take it to the field. Work with the plant, animal, fish, whatever is your ultimate. Connect it. Learn from that. Come back to your own interpretation of your data, your material. If you understand what is polymorphism, what is pleiotropism, what is the polygene, then you will be able to interact quickly and you will be able to make much greater sense. So keep your eyes open, bring these changes in your curriculum. I happen to be also the chairman of the Dean's Committee. I have suggested these changes in the curriculum of the universities. IARI will also have to probably change several of those things which possibly have to be done differently, teach differently, equip our students differently, equip our teachers differently, so that these realities are internalized in our teaching system. If we can do that, then it is a real respect we are paying to mentors. With these words, I am so delighted, my friends, that you gave me the opportunity to touch hands of my students on both sides. And to be here, I thank you very much for this opportunity.
Thank you, sir, for the remarkable talk, inspiring young breeders and minds for better India and better world. And I would like to request Dr. Raman Babu and Dr. A.K. Singh to present the vote of thanks, please.
Thank you very much to Dr. R.B. Singh, Professor R.B. Singh, who took time to come and chair this valedictory session. Even when he used to be director IARI, he used to make it a point to come and teach us. So I think that's tremendous commitment, and we have greatly benefited out of it. So I think students, thank you very much. I think the major purpose of this 2-day symposium was to expose you to some of the technological developments. Technology has pervaded all the spheres of our lives, and agricultural research is not going to be an exception, right? So we use cell phones, we use Google, we use online transactions, we use driverless cars. We are going to use driverless cars in future. So if all those technologies are going to affect you, agricultural research also will evolve. So this is just a small primer for you to know that technological developments are happening in our own field, in the plant breeding field. And this is just to expose you to all those developments so that you will go back and refer to all those things, the literature available in public domain, and benefit out of that. So, before I propose a formal vote of thanks, 2 issues raised by students from their feedback. One, can we distribute PowerPoints, right? So that was one of the requests. So I would really request you to understand this. In any organization, there are certain principles, basic procedures and ways of functioning that are very well established. So perhaps we are not allowed to distribute this, for and companies that operate in multiple countries, we have multiple host country sensitivities. And as I told you, to be a successful plant breeder, you should have a very good legal knowledge of whether you are infringing upon somebody else's product or whether somebody else is infringing upon your rights. So I think legal aspects are really, really important, and there are certain principles laid out in the organization, so we should respect that. So that may not be possible for us to distribute. Number one, I just want to be open about it. Number two, data analysis. So this is something very, very doable, but the data analysis that is done within the organization is not like 1 or 2 different tools. It is an ecosystem of tools. There are at least 30 or 40 tools which are done one by one, and just teaching you 1 or 2 different tools will not make. So that's why what we did today, what we couldn't do it today, we had a session on demonstrations on calculating blobs, genomic selection, the whole genome prediction, and also some of the genome editing stuff, so which we couldn't do because of the paucity of time. But we planned that based on open source tools that are readily available in public domain. So you can make use of those tools, and you can certainly get all the results based on those tools. So these are very much doable, and some students have expressed interest that they want to still go through the demo. We are certainly open to it. We have made all the preparation. So if there is time and if there is interest, we will certainly take it up after the valedictory function. Yeah. So with that, let me propose a formal vote of thanks. And as I told earlier, I'm incredibly happy that this finally happened. And coming back to IARI, being an alumnus is a great feeling. And my first thanks goes to all the students. When you made it possible. I know the kind of chaos and pandemonium we were going through with the currency, what do you call that, the surgical strike on black currency, right? So I think some of the students left without any money. I think just with the tickets they left their universities. So I think we are really, really grateful to you that you could make it all the way. Students and the online community which watched us. I think that those were a tremendous audience, and we even were receiving, today being Saturday, still there was tremendous interest and we were getting questions online. So that was very good to see and tremendous interest. We would like to thank Professor M.S. Swaminathan. I mean, at this age, coming to such places and still being a keynote speaker, I think that speaks volumes of his commitment, and even his mere presence was such a big inspiration for us. I'm sure you would agree with me. And very, 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 very special thanks to Dr. A.K. Singh. He made it really happen. So he, very big hand. I think we need a very big hand. Dr. A.K. Singh, he made it really happen, this one. So he used to be a very, very friendly teacher to all of us. So we had that kind of freedom with him all the time. So from the word go, he was behind all the arrangements, and the scale and the perfection with which it was done is unimaginable for us, and it was excellent, and thanks a lot, sir. And also I would like to thank all the team members, Dr. A.K. Singh's team members, Firoz, Tara, Talukdar. I'm sure there are many more. I don't know the names. And also we would like to thank the management of IARI, joint directors, director, dean. I think they could attend most of the sessions, and that was really very encouraging for all of us. The keynote speakers, Professor B.D. Singh as well as Dr. P.K. Gupta. So that was again excellent talks, and I would like to also thank all our former teachers who were there, and it was such an inspiration to see all of them. And last but not least, my dear pioneer colleagues. I think they have been great support for the last, not only for the last 2 days, but I think over the last 2 or 3 months. I think whatever we presented, you would have realized that this is all team effort. A lot of those slides were not even made by us. There are people, a very good team in Johnston. So we have a nucleus of team working there, and they all readily helped. And I would like to place a special thanks to Dr. Tabare Abadi, who is our research effectiveness lead, and he is spearheading this across all over the globe, the conducting of the symposium series. And we would like to place special thanks, and I would like to thank my pioneer colleagues, Dr. Anand Pandravada, who is here. As I told you, he is our research director, and he is not only directing CON, he is leading us on many of the other spheres as well. Despite a personal emergency at home, I think he is here still. So I think we are really grateful for Anand. I'd like to thank Dr. Mahala, who is our very senior breeder, and you heard from him what commercial plant breeding is. And I would like to place thanks to Dr. Amitabh, who is our very senior research leader, trait optimization and discovery team. I'd like to thank all my scientists, Emerson Limburger, Justin Gerke, who have come all the way from the United States, Dindo from Philippines. And I would like to thank Dr. Rakesh Arora, another very, very, very senior breeder with respect to Brassica, and Rakesh, Yogesh, as well as Manoj, all his team members. I am sure I am leaving somebody. So I would like to thank our senior leadership, Dr. Hans Bharadwaj, Dr. Sandra Milach, and also Jeff Graham. So I think I am sure there are many other people who have contributed to this. And I would like to once again thank every one of you, and this has been a great success, at least for us, in terms of getting your valuable feedback. And if there is a way to get some kind of feedback, we will try to do that as well through email or any other means. We should have done a feedback form. Sorry about that. So if there is still interest, we are here. So after this, if you want to take up the demo, we can do that. Otherwise, I'm sure we can do it anytime, somewhere, in some other occasion. Thank you very much once again. Thanks.
Thank you, Raman. I don't think there is anything left now. You have covered almost all the key issues and the
stakeholders and their commitment for this program. In any successful program, and this has been one very successful program, what goes into it is, 3P, I say, the planning for it. And this program, the very first idea for commemorating 150 years of Mendel's laws of inheritance was given by Dr. Swaminathan to me in April this year, when he had come for the Foundation Day lecture of IARI. And it was our good fortune that in July, Raman came and he proposed that if IARI could organize a symposium on genomics-based crop breeding approaches. And it was a God-given opportunity. We thought we will definitely do it, and we had several rounds of meeting. So that was planning part in terms of prioritization, who will be the stakeholders, the students from how many universities, because he had already done one in Hyderabad and one in Punjab Agriculture University, Ludhiana. So we thought we'll focus more on the northeastern and western part. So we had 15 universities, approximately 2 students each who participated from Central Agriculture University in Manipur to Palampur. And more importantly, as I said, the webcasting of this program to all 73 agriculture universities. We wrote to the respective vice chancellors, and they were kind enough to make the arrangement, and also to all the crop-based research institutes, directors, to make available the program for their scientists and also the seed industry. So through this, because still it is on live webcast, I would like to thank the entire viewership who are watching the program for being with us for last 2 days to take the benefit of this program. I am sure this is not the last but the first program with Pioneer DuPont that we have, and I hope and I wish this becomes a kind of regular feature. As a matter of fact, the greatest beneficiary of the plant breeding teaching program that happens in the university in the public sector, the greatest beneficiary is the private sector, because all the HR that you get in the private sector, they are the product from public sector institution. And therefore, there is a sort of corporate social responsibility from the private sector to enrich the education in the public sector institution. I wish many more, industry partners take this kind of clue and the inspiration from what Pioneer DuPont is doing, and they come forward to sponsor many more such symposium. We will be very happy to be in touch and continue this as a sort of regular program. In terms of, this has been very educating, and the differences that I find is that most of our marker-assisted breeding program as of now are focused on handling with the simply inherited characters. The kind of focus that has been given on phenotyping, high-throughput genotyping to handle the complex quantitative trait is very, very enriching. And this is definitely going to be the order of the day in times to come. The speakers from Pioneer DuPont, I would like to add my own words of thanks to Raman, Emerson, Justin, Anand, Amitav, Dindo, and Dr. Mehla for making very excellent presentation. And I made a request to very senior faculty, senior citizen, many of them, Professor P.K. Gupta, just one call, Professor R.M. Singh here, Professor Deepak Pintal, and Professor B.D. Singh. And they did not take a minute to say yes to participate in this program in spite of several commitments. Professor Gupta was in Uttar Pradesh, in Lucknow, attending another meeting, and from there he came direct to this place. Likewise, Dr. Swaminathan traveled day before the inauguration. He traveled to Mumbai. Evening he came late night, and then he was with us. So my big thanks to each one of them for honoring our request and participating in this program. My colleagues in the division of genetics, and particularly the one person who has worked behind the scene is Firoz, very systematically, and the entire, if I say the what you see on the backdrop, what you see on the memento, everything done by him with, of course, help of his excellent team to support him Dr. Prabhu for all the support that he has provided, Dr. D.K. Adav for providing excellent support to transport and food, Dr. Bharadwaj, Dr. Tara, she has been doing the management of the entire stage, hall management, etc. Ranjit in designing the logo for the backdrop, he worked hard and came out with many innovative ideas. Ramaprasad, Dr. Binod was solely responsible for organizing the entire poster session, which was a sight to see in the Division of Genetics corridor yesterday, very well organized. And Sailendra, Dr. Gopal, for recording many of the very important photographs, which will become, in fact, history for all time to remember. And I think, and finally, Professor R.B. Singh. In fact, I requested him to be the chair of the inaugural session because Professor Swaminathan was coming. I requested him, but he had committed for a program of the convocation program of Central Agriculture University in Manipur, and therefore he could not be with us that day. But today morning I called him and I learned that he has come back from Imphal, and he didn't take any time. He said, I am there, you take it from me. And thank you very much, sir, for being with us and for giving us very valuable advice and support. Your blessings will go a long way in completing the targets that we have, the targets that you have given to us, and we will certainly do it with the help of all the students community present here. I thank each one of you, including Bansalji sitting there, who has been managing the entire PowerPoint presentation and the hall. Each one of you who are directly and indirectly involved with the successful completion of this program. Thank you very much.
मैं तो तुम्हारे यूनिवर्सिटी से क्यों नहीं आया अभी